Good morning and good afternoon, everyone. My name is Mahalaya Marshall, and I'm with the IQ Consortium Secretariat. On behalf of IQ Consortium, I'd like to thank you for joining today's workshop entitled Machine Intelligence for Quantitative Modeling in Drug Discovery and Development Applications. Before we get started, I'd like to take care of a few housekeeping items. First, all attendees will be on listen-only mode. Next, depending on your viewing method, please enter questions at any time during the workshop using the question module located in the control panel on the right-hand side of your screen, or click the question mark on your control panel. If possible, please preface all of your questions with the speaker name and the slide number so that we may properly direct your question. This webinar will be recorded, and a copy of the recording and slides will be circulated after the workshop concludes. Due to fluctuating bandwidth demands, attendees may experience intermittent delays in slide transition and/or audio disruption. Click the handout icon to view the connection and audio tips handout for possible solutions. Thank you for joining today's workshop. I'll now turn things over to our workshop lead, Dr. James Liu.
Hi, good morning and good afternoon, everyone. My name is James Liu. And on behalf of the IQ Consortium Working Group on AI/ML, I would like to welcome you to this workshop. So this is an overview of the workshop program. Today, we will focus on drug development applications by starting the program with a keynote, followed by the 3 sessions shown here. And the workshop will continue tomorrow with a half-day program on drug discovery applications. Next slide, please. Today, we will start off this workshop with a keynote presentation from Dr. Qi Liu. So Dr. Qi Liu is currently the Associate Director of Innovation and Partnership in the Office of Clinical Pharmacology at the FDA. During her time at the FDA, Dr. Liu has contributed to over 200 NDAs and SADAs reviews, as well as numerous BLA and IND reviews. She has helped to develop the OCP's capacity on machine learning and real-world evidence and digital health technologies. She's also on the editorial board of many journals, including AAPS, Clinical Pharmacology, and Therapeutics, and so on. She's also the member of the FDA Digital Health Advisory Board, the AI/ML subcommittee. It is therefore our privilege today to hear Dr. Liu give us some of her perspectives on the evolving regulatory considerations for AI/ML in drug development. I will now turn things over to Dr. Liu.
Thanks, Dr. Liu. Good morning, everyone. It's my great pleasure to be here today and to share with you my views on the application of artificial intelligence and machine learning in drug development. I would like to start my talk today with a discussion on why we need to talk about AI and machine learning today. This is because the application of AI and machine learning in drug development is expanding very rapidly in recent years. They can be very powerful tools with the potential to improve the efficiency of drug development and advance precision medicine, but they also have unique challenges. Both opportunities and challenges lie ahead, so it will be very helpful if all of us can have some understanding of what AI and machine learning are, what they can do, where they can add value, and what we need to be cautious when we use them in drug development. Here's the outline of my talk today. I will first give a very brief background about AI/ML, followed by a landscape analysis of the AI/ML related submissions at CDER FDA. After that, I will discuss challenges related to the applications of AI/ML and my personal view of the regulatory considerations for their application in drug development. Then I will include 2 examples of machine learning application in drug development. Lastly, I will share some AI/ML related activities in FDA's Office of Clinical Pharmacology. Although many people use the term AI and ML interchangeably, the 2 terms are related but not the same. I will start with the definition of machine learning first, because it's more clearly defined. Machine learning is a field of study that gives computer the ability to learn without being explicitly programmed. During the machine learning process, a computer program is learning from experience with respect to a task if its performance at this task improves with experience. If we think about experience as data, this simply means the performance of the algorithm improves when it is presented with more data. Now, I bet this sounds familiar to you, and many of you are probably wondering whether you have done it before. The answer is probably yes. In fact, machine learning has a lot of overlap with statistical or pharmacometric modeling. Most high school students who have done linear regression can say they actually have done some machine learning. So what exactly is new this time? In general, the traditional statistical or pharmacometric modeling use explicit model structures defined with domain knowledge and assumptions, while the machine learning approach uses very general and flexible model structure with less reliance on domain knowledge and assumptions. In general, the traditional statistical or pharmacometric modeling tends to excel at handling data with a small or moderate number of independent variables, while the machine learning approach are very good at handling data with a large number of independent variables, and it can also handle new types of data such as medical imaging. In general, the traditional statistical or pharmacometric models are easier to interpret or explain or draw statistical inference, while machine learning such as neural network are harder to interpret or explain or draw statistical inference. Instead, they are more focused on predicted performance. When we talk about machine learning, it's important to understand a few related terminology. For example, artificial intelligence is a big word. It is very often used interchangeably with machine learning. However, artificial intelligence is a much broader field as compared to machine learning. It's a term coined by John McCarthy in 1955 as the science and engineering of making intelligent machines. In the HHS AI strategy document published last year, AI is defined as the theory and development of computer systems able to perform tasks normally requiring human intelligence in order to deliver solutions that can automate routine tasks, draw database insights, or augment human activities. Within the machine learning field, there are many different algorithms. The artificial neural network is a popular one, and many of the recent breakthroughs in the AI/ML field are applications of neural networks. This is a mathematical model developed under the inspiration of the network of neurons which make up the brain. It uses layers of mathematical processing units, the so-called neurons, to analyze the data. The data goes into the input layer and then travels through the collecting lines, or the so-called edges, to neurons in the hidden layers. These collections are weighted. The higher the weight, the greater influence one neuron has on another. In the neurons of the hidden layers and output layers, 2 functions will happen. One is to calculate the total net summation of all the inputs into this neuron, and the second one is the activation function, which is usually a nonlinear transformation of the net input into a new output. This new output is then sent out over one or more edges to the next layer of neurons. This cycle continues until the output layer calculations are completed and output is made. Then based on the difference between the neuron network output and the target output, in other words, the observed data, the weights on the collecting lines will be updated, and we call this a learning process of the neural network. Deep neural network refers to a neural network with many hidden layers. Traditional neural networks only contain 2 to 3 hidden layers, while deep neural networks can have more than 100 hidden layers. Most deep learning methods use deep neural network architecture. Therefore, many people use deep neural network and deep learning interchangeably, although strictly speaking, they are not the same. Some of the most impressive applications in the deep learning field include using convolutional neural network for computer vision and recurrent neural network for speech recognition. Machine learning can be used to aggregate data, synthesize information, seek patterns, and optimize decisions. When used in drug development, it can help us understand the disease in targets, generate and evaluate drug candidates and combinations, improve trial design, and advance precision medicine by improving diagnosis and treatment. Now, I will talk about the regulatory submissions with AI/ML components that CDER FDA have received in the past few years. At CDER, the submissions with AI/ML components have been increasing very quickly in recent years. As you can see from the figure in the table here, before 2018, there were very few submissions with AI/ML components at CDER. In 2016 and 2017, we identified only one such submissions each year. From 2017 to 2020, the numbers increased about 2-fold to 10-fold yearly. And then in 2021, the number increased sharply to 132. The most common type of these submissions is IND, although there are also NDA, BLA, ANDA, drug development tools, and critical path innovation meeting packages. This figure here illustrates the distribution of the submissions by therapeutic area. Oncology, psychiatry, GI, and neurology were the disciplines with the most AI/ML related submissions from 2016 to 2021. This figure summarizes the distribution of these submissions by the stage of drug development life cycle. In these submissions, most of the AI/ML applications happened at the clinical drug development stage, but it also happened at drug discovery, preclinical drug development, and post-marketing stage. It's important to note that the frequency by which AI/ML is mentioned in regulatory submissions to FDA likely only represents a fraction of its increasing use in drug discovery. We also summarized common analysis types of AI/ML application in the submissions. They were often used for prediction of clinical outcome, including disease prognosis and treatment response based on characteristics of the patients as well as the treatment. AI/ML were also commonly used for covariate selection and confounding adjustment. For example, in some submissions, AI algorithms were used to screen through a large amount of baseline information such as patient demographic data and lab values to find important factors that can impact patients' prognosis as well as exposure or response to a drug. AI/ML have also been used for pharmacometric modeling, anomaly detection, the analysis of imaging data, videos, or voices. Some submissions explore the use of AI/ML for natural language processing and the phenotyping of real-world data. We also summarized the common objectives for the use of AI/ML in the submissions. AI/ML have been demonstrated to be a very useful tool for drug discovery and repurposing. We have seen submissions where sponsors stated that the selection of the therapeutic targets or drug candidates were supported by AI/ML. In some submissions, AI/ML was used to predict the potential safety risk of a drug based on its structure, physicochemical properties, or affinity to certain targets. One common application of AI/ML in drug development is to facilitate enrichment trial design. Generally speaking, there are three categories of enrichment strategies: decreasing variability, prognostic enrichment strategy, and predictive enrichment strategy. AI/ML have been used for all three enrichment strategies. Certain submissions explore the use of AI/ML for patient risk stratification and management. AI/ML were also proposed for dose selection or optimization. AI/ML-based monitoring platforms were used in trials to aid and confirm adherence to the investigational drug regimen. AI/ML has also been proposed to generate synthetic controls, endpoint or biomarker assessment, or post-marketing surveillance. AI and machine learning have lots of potential to make our world better, but they also bring many challenges. Here I will focus on three technical challenges that are especially relevant to our regulatory review: bias, generalizability, and opacity. It's important to know that these technical challenges can lead to issues of disparity, fairness, and trustworthiness. Bias can be introduced in many ways, but one of the most common ways is through bias in the data we use to develop the model. We need to remember that the model can only be as good as the data, otherwise it will just be garbage in, garbage out. Some of you have probably heard about this story in the news. Amazon had been building computer programs since 2014 to review job applicants' resumes with the aim of automating the search for top talents. But by 2015, the company realized the AI program showed bias against women. Why did this happen? This is because AI was learning from a biased dataset. Amazon's AI models were trained to vet applicants by observing patterns in resumes submitted to the company over a 10-year period. As you may know, US tech companies have yet to close the gender gap in hiring. This disparity is most pronounced among technical roles, where men far outnumber women. Amazon's AI engine just followed the same pattern. The lessons we can learn from this case is that training the AI algorithm with a biased dataset will produce a biased algorithm. Generalizability can be another big issue. It refers to how well a trained model can perform on previously unseen data. I guess many of you have probably heard about the AI apps that detect skin cancer, but do you know many of those apps only works well for white people? This is because the data used to develop those models were mostly images from white people. So when the app is presented with images from patients with darker skin tones, they may not perform as well. Another common issue is that many AI machine learning models were built with data from a single institution and may not be generalizable to data from other institutions. This is because different institutions may have differences in patient population, the data collection, or even medical practices. Data sharing and collaboration among different institutions are critical, but can be challenging. Some of the AI/ML algorithms suffer from opacity, in other words, lack of explainability and interpretability. In general, the more complex a model is, the less likely we understand the relationships between input variables and model prediction. It is the main reason why some machine learning models are called black boxes. Now, I would like to discuss my personal view of the regulatory consideration for the application of AI/ML in drug development. First, when we use AI/ML in drug development, we need to clearly define the context of use of the model. Proper validation of the algorithm is critical. However, our expectation should be fit for purpose and risk-based. Ensuring that the algorithm is generalizable is also important. To the extent possible, the training data should be unbiased, diverse, and inclusive of the intended population. Methods need to be developed for performance guarantee. The black box nature of many AI/ML systems, such as artificial neural networks, can sometimes discourage their application. We should encourage the development of methods to increase interpretability and explainability. In addition, we need to develop best practice for AI/ML, such as how to split the data for training, validation, and testing. In the previous slide, I talked about fit for purpose and risk-based validation for AI/ML applications. Here I'm going to expand on it, showing how we have borrowed concepts from the modeling credibility assessment framework in our review of AI/ML applications. Here the term credibility refers to trust in the predictive capability of a model for a particular context of use. Based on this framework, model risk guides the level of credibility evidence needed. In this framework, model risk is determined by both the influence of the model on a decision, we call it model influence, and the consequence of adverse outcome from an incorrect decision, we call it decision consequence. An increase in either factor can lead to an increase in overall model risk. Today, I will share two examples where machine learning was applied during drug development. In the first example, machine learning was used for patient risk stratification and management. First, let us look at the context of use. Drug A is under development for life-threatening disease. In previous trials, all patients went through inpatient monitoring after dosing due to the concern of a potentially life-threatening drug-related adverse event. The sponsor proposed to use a machine learning model to predict a patient's risk for this AE based on baseline characteristics and lab values. If someone is predicted to be of low risk, then he or she will have outpatient monitoring instead of inpatient monitoring. It's worth mentioning that the sponsor was not clear on some components of the context of use. For example, do they plan to use this algorithm in clinical trials only or in both the trial and the real-world application? FDA sent the sponsor relevant comments. In addition, the sponsor also mentioned that this model could also be used for dosing regimen optimization. If so, it will need a different model risk analysis. FDA's model risk analysis has two dimensions, model influence and decision risk. In this case, we rank the model influence to be high, as the model prediction will be the sole factor to determine whether a patient will go through inpatient or outpatient monitoring after dosing. For decision risk, we ranked it medium or high. If a model erroneously predicts a low-risk patient to be moderate or high risk for this AE, it may not be of major concern, as this patient will go through inpatient monitoring even though it may not be necessary. If the model erroneously predicts a moderate or high-risk patient to be low risk, it will be of a major concern, as this patient will not have the necessary inpatient monitoring. The analysis of the decision risk is also very helpful for us to figure out which performance metric will be most critical. Here, we concluded that the negative predicted value will be the major performance metric to determine the acceptance of the model from a regulatory perspective. Now I'm going to share the second example in which machine learning was used for enrichment trial design. Again, let's start with the context of use. In the phase 2 trial, drug B failed to reach statistical significance in the pre-specified primary efficacy analysis. Their analysis revealed unexpectedly large improvement in the primary efficacy endpoint from baseline for the placebo-treated cohort. This motivated sponsors to explore machine learning methods to identify patients with abnormal patterns. The sponsor is now proposing a machine learning-based enrichment strategy for 2 planned phase 3 studies, which aims to decrease inter-subject variability prior to randomization. They propose to select canonical subjects whose symptoms were characterized by greater similarity to the typical patient in the target population. The dataset for the model development include the phase 2 trial of drug B and several trials of drug C for the same indication. The non-mean score is calculated by applying a trained isolation forest model on the components of the efficacy endpoint at screening and baseline. This algorithm is an unsupervised machine learning algorithm. The non-mean score for a given subject indicates how easily he or she can be isolated from the rest of the subjects. If someone's anonymous score is less than a pre-specified cutoff value, he or she will be considered as a canonical subject and included into the trial. Otherwise, he or she will be considered as an anonymous subject and excluded from the trial. Encouraging trend was observed when machine learning-based inclusion criteria were applied retrospectively to the phase 2 data of drug B. We observed a larger effect size after this inclusion criterion is applied. Similar trends were observed when it was applied to trials of drug C, another drug for the same indication. Here I want to discuss one of the major comments from the FDA. The sponsor proposed to use the machine learning-based inclusion criteria in 2 phase 3 studies. FDA suggested that sponsors use this criteria in only one of the 2 planned phase 3 studies. We made this recommendation due to the potential challenges related to the explainability of the model and generalizability of the result. It would be helpful to include the anonymous patients in at least one of the 2 phase 3 studies. Now, I would also like to introduce some of the AI/ML related activities in the Office of Clinical Pharmacology. We launched an innovative data analytics program in 2019 to help build capacity within OCP for exploring and leveraging the application of AI/ML, digital health tools, and real-world evidence, and other emerging data science tools to support advances in drug development and regulation. Last year, our office also established a machine learning review team. I would also like to briefly mention some AI/ML related research activities in our office. We have conducted some landscape analysis, methodological exploration, and their application in therapeutic optimization and individualization. Here I listed some reference articles in case you're interested. Currently, AI/ML related review and research have become an integral part of OCP's function. I was often asked how we made it happen, so today I will share our story with you. Back in 2017, three of my friends in OCP and myself noticed the scientific development in AI and machine learning. We quickly realized there was a great potential for the application of AI/ML in drug development and precision medicine. So we decided to form a machine learning study group. We met on weekends and used resources such as Coursera machine learning course and other online training. We call ourselves the Geek Squad. At this point, it was a grassroots effort purely based on scientific curiosity. I think the vision for our office leadership was critical in turning this grassroots interest into something strategic and impactful. In March 2018, our office director, Dr. Isang Zalei, tasked me to build a program for the office, starting from educational efforts. Dr. Xiumei Huang, our deputy director, also gave us a lot of support, especially funding to support our research projects. Remember, this was back in 2018 when there were very few submissions with AI machine learning components at CEDAR. I really appreciate the vision of our leadership and their trust in us. At that time, Isang's ORAS fellow, Dr. Daphne Zhen, also volunteered to work with me. Together with Stephanie, Doctors Yaning Wang, Chao Liu, and Hao Zhu, we officially launched the IDEA program in 2019. Under the IDEA program, we focused on education, research, collaboration, and fellowship. All these activities helped develop our capacity in AI and machine learning. Then in 2021, when a huge increase of AI/ML related submission happened, we were ready, and it was a logical and painless step for us to establish this machine learning review team and take on the new challenges. I hope our experience can be of some help to you. Here are the take-home messages of my talk today. AI and machine learning are playing an increasingly impactful role in drug development and regulation. They have potentials to improve the efficiency of drug development and advance precision medicine, but they also have unique challenges. Both opportunities and challenges lie ahead. More education, research, and collaboration are needed to help move the field forward, and I hope many of you will be part of this effort. Last but not least, I would like to thank all my colleagues who have contributed to this presentation and our AI/ML related projects, and thank you for your attention.
Good morning, everyone. My name is Kumpal Madrasi, and I would be introducing you to our session 1, which is AI/ML enabled analytics and pharmacometrics workflows. I'm an associate director of division of PKDM in Sanofi, but prior to that, I've had experience in various pharmacometric roles for some time. So for this session, we will know the role of artificial intelligence in increasing automation and causing a greater efficiency in data analysis. But there's a risk of AI/ML overfitting data compared to more traditional methods. So we will know in more details during this session. The first speaker is Meng Hu from FDA. He is team lead at the Division of Quantitative Methods and Modeling in the Office of Research and Standards of OGD, CEDAR. Dr. Hu received his BE in Biomedical Engineering and PhD in Physics from Zhejiang University, China. And he did a postdoctoral fellowship at Drexel University in Philadelphia. He then joined FDA CDER as a staff fellow in 2015 and rose up to the rank of team lead in DQMM under ORS. His main research interests include the development and application of advanced data analytic tools to promote business intelligence in government big data management, generation of real-world evidence, and quantitative methods to facilitate assessment for in vitro bioequivalence studies. And around these topics, he has many publications. I welcome Meng to give his talk. Meng, please take it away.
Hello, everyone. It's my great privilege to be here and talk about leveraging artificial intelligence, AI, and machine learning to support generic drug development and regulatory efficiency. This presentation reflects the views of the author, should not be construed to represent FDA's views and policies. Here's the outline. First, I will introduce the definition of AI and its prevalence. Second, I will discuss the opportunities offered by AI for facilitating generic drug development and regulatory assessment. As the third and fourth points, I will talk about our efforts to answer the opportunity call as well as the highlighted case example. Lastly, I will summarize the learned challenges and lessons. AI is everywhere. What is AI? In 1950, Alan Turing, the father of theoretical computer science and artificial intelligence, proposed the famous Turing test. Turing proposed a computer can be said to possess AI if it can mimic a human response under specific condition. As shown in the right side figure, A refers to a computer, B, C represent two humans. During the test, C functions as a questioner, while A and B function as a respondent. If after a preset number of questions, C cannot determine the difference between A and B, then the computer A is considered to have an AI. Another definition comes from John McCarthy, one of the founders of the discipline of artificial intelligence. He said, it's a science and engineering of the making intelligent machines, especially intelligent computer programs. Nowadays, AI and machine learning have become buzzwords. You can hear them almost everywhere. In general, machine learning is considered as a subcategory of AI. The recent decade has witnessed the exponential growth of AI. Truly, the thriving AI community should thank the advance in information technologies and the powerful chips, including big data, data analytical tools, and HPC, high-performance computer. Indeed, AI is transforming many areas of everyday life, including smartphone, autopilot, chatbot, personalized recommendation, and ATM. Given the prevalence of AI, we also see AI offers opportunities to facilitate generic drug development and regulatory assessment. The first aspect of AI can help is development of automation tools, such as for streamlining labor-intensive work. With automated process, we expect to see enhanced efficiency, improved consistency, and high-quality deliverable. The second aspect we see the benefit of using AI is to borrow strengths of recently developed advanced data analytical methods to promote business intelligence, like modeling, optimizing business process. In addition, the advanced quantitative analysis model can also be used for supporting regulatory assessment, like API sameness assessment. This slide shows our efforts. The effort from Division of Quantitative Modeling and Method under the Office of Research and Standards in OGD to answer the opportunity call. First, It's about development of automation tool. We are having two ongoing projects in this regard. One is about development of a bioequivalence assessment tool aiming to streamline the labor-intensive, less human intelligence-involved task during the bioequivalence assessment. Another project aims to develop a data analytical tool based on machine learning and natural language processing to facilitate product-specific guidance development. Moving to the right side, we also conduct research to employ the advanced data analytical method, for example, machine learning, for promoting business intelligence. Here, I list the 3 ongoing projects from this perspective. They are prediction of AI submission, heterogeneous treatment effect analysis to inform impact of PSG, and modeling the process of ANDA assessment. The advanced data analytical method, for example, multivariate analysis method, can also be used to support regulatory assessment. 2 examples I list here are equivalence assessment for complex particle size distribution and multivariate analysis method to facilitate active pharmaceutical ingredient sameness assessment. In the following slides, I will use the highlighted 2 projects as case examples. The first case example I want to share is about machine learning-based heterogeneous treatment effect analysis. Heterogeneous treatment effect, HTE analysis, has drawn growing attention in a variety of fields, from economics to medicine. In the economy and the marketing field, the HTE analysis may go by the alias uplift model. In the big data era, dramatically increased data volume and complexity pose significant challenges for HTE analysis, especially using conventional methods. Recently, machine learning methodology have been employed in the HTE analysis, show their merits in handling complex data, given their nature of no assumption on data. To introduce this advancement to the community, we conduct a systemic performance evaluation for the machine learning method against the conventional method by simulating various complex scenarios. What's HTE analysis? Compared to the response analysis that predicts the outcome itself, HTE analysis focuses on examining varying treatment effects for individuals or subgroups in a population. Figure A here shows a case without treatment heterogeneity. In the figure, blue and gray curves show outcomes according to treatment and control, respectively, over all the individuals. Here, we can see that treatment can lead to different outcomes over the population, shown as a blue curve. However, if we look at the treatment effect, which is the difference between treatment and the control, the treatment effect remains the same for all the individuals. This is a hypothetical situation, but shows the scenario without treatment heterogeneity. Okay, then if we look at the figure B, we can clearly see the HTE over the population in this figure. Importantly, to conduct HTE analysis, one unique challenge is the treatment effect is often not explicitly observed on a given data, as each subject can often only be exposed to one of the treatments, which is also known as a fundamental problem of a causal inference. To conduct HTE analysis, one conventional method is called a 2-step method. Very briefly, with the 2-step method, we will build separate models for each treatment groups and estimate the treatment effect by calculating outcome difference between models. Another development direction is a machine learning-based method. The first attempt was the tree-based method. Now it has been evolving to the forest-based method, causal forest, which is the method we would like to introduce in this work. The table provides a general pros and cons of 2 methods. For 2-step method, the pros is it is intuitive and easy to implement. But the cons are the 2-step method is not a necessarily accurate model, and it's often based on ordinary regression model. And also user need sufficient domain knowledge to introduce interaction between the variables. For causal forests, the pros are this method is capable of handling complex practical problems, and it was developed to overcome issues of a single tree method. Also, causal forests will inherit all the machine learning methodologies issue, like a black box, and its dependency on the quality of data. To conduct performance evaluation, simulations were performed. We learned interaction between treatment and the covariates often lead to HTE among the study population. As shown in this equation, Y, X, and T refers to outcome, covariates, and treatment respectively. Fx and Gx refer to functions of covariates. Of note, Gx and T have interaction. And the below table shows 4 models, 1 to 4, simulating scenarios with different level of HTE complexity. Model 1 represents scenario without HTE because there's no interaction between T and X. Model 2 and 3 correspond to linear and complex HTE. Model 4 uses the basic format of Model 2, which is a linear HTE but with a much bigger number of covariates. And the number of covariates exceeds the number of observations, which impose significant challenge to the conventional regression-based method. In summary, we expect to see no difference in performance for Model 1 and 2, but better performance of machine learning method for Model 3 and 4. 2 evaluation methods were applied for performance check. They are root mean square error and the Gini curve. IMSE is quite straightforward, which directly compare predicted value and the true value. Due to the time limit, I will have to skip the description how to generate the Gini curve. But importantly, Gini curve describes the treatment effect of regrouped population based on the model predicted individual treatment effect, shown as the red curve in this figure. In the same figure, the black dotted line shows the benchmark curve, which is constructed by random generated subgroup from population. Thus, the more effective HTE method is, the more the Gini curve will separate from the benchmark curve in the plot. Of note, the value of the Gini curve at 100% population is always equal to estimated average treatment effect for the whole population. By subtracting the area under the benchmark curve from the area under the Gini curve of HTE method, the Gini coefficient can be obtained. This slide shows an exemplified case generated by a nonlinear HTE model as shown in this equation. There are 10 covariates, but only x1, x2 have interaction with treatment and in a nonlinear way. The heat map A shows the true treatment effect distribution in the space with coordinates x1 and x2. We can see the pattern is quite complex. And the heat map B and C show the treatment effect distribution recovered by causal forest and the two-step method, respectively. It's clear two-step method only capture the general trend of the true distribution, while causal forest restore the details of the true distribution. This page shows the results from a systemical test by the 4 models using RMSE. We can see that for model 1 and 2 methods, performance have no significant difference. However, for model 3 and 4, two-step method produce significantly higher RMSE than the causal forest. Now we are looking at the results of Gini curve from model 1 to 4. In each panel, the gray, red, and blue curves refer to true treatment effect, causal forest, and the two-step method, respectively. We can see that, as expected, for model 1 and 2, the 3 curves overlap together. For model 3 and 4, none of the methods can perform as good as the true values. However, the red curve, which is from causal forest, is more closer to the true value than the two-step method. Model interpretability is always a topic for machine learning-based method. Causal forest offers a useful variable importance algorithm to facilitate the identification of important variable. The figure here shows an example from Model 4. By design, in Model 4, there are 500 covariates, but only the first 5 covariates have interaction with treatment. From this figure, we can see the variable importance algorithm can accurately capture the first 5 covariates that have interaction with the treatment. As a recap, the above results suggest causal first, a machine learning-based method, is a promising tool in real-world application for HTE analysis. It can potentially be used to improve business intelligence in the agency if treatment and outcome are carefully designed. As the second highlighted case, I would like to share some experience challenges learned from an ongoing project. This is a contract focusing on applying text analysis and machine learning to facilitate product-specific guidance (PSG) development. The left side diagram displays a high-level thinking on this project. According to our analysis, to develop a regular PSG for immediately released product, a developer usually spends 50 to 60% effort time to conduct information retrieval from public and internal data sources. Use 20 to 30% of effort time for information summarization. Based on all the collected information, the developer will draw conclusion, recommendation, and etc. Given the current advance in natural language processing, the project team is working on streamlining the information retrieval and the information summary part. If we finally succeed, a significant amount of time and efforts will be saved for developer so that more time can be invested on the human intelligence involved task. Meanwhile, the project team was and is facing a few challenges, including evolving layouts of a source document, need for information retrieval based on semantic understanding, capturing information from unstructured text, choosing a proper NLP model. The next slide will show an example of information retrieval based on semantic understanding. In this project, one specific task is to extract paragraphs with ADME information from drug labeling. However, in some drug labeling, rule-based searching, for example, keyword searching, does not work. This is an example of a semantic understanding-based information retrieval from drug labeling for food effect. The two labels, as shown here, contain no keyword for food effect paragraph, although the word food appears in the paragraph, but keyword searching for food will lead to high false positive rate. The NLP algorithm has been applied to implement the semantic understanding-based information retrieval. We are now able to correctly label the paragraph with food effect information in drug labeling. As a progress of the project, the state-of-the-art BERT model was used for this NLP application. NLP pipeline was developed to extract drug product information, for example, ADME information from drug labeling with minimal human intervention. More information can be found in the published paper. To wrap up my presentation, here are takeaways. AI technologies bring opportunities to advance development and regulatory assessment of generic drugs. AI have been applied to facilitate BE assessment, PSG development, business intelligence, and regulatory assessment, etc. AI will play a more important role in providing high-quality generic drugs for US public as more challenges get addressed. That concludes my presentation. Thanks for your attention.
Thank you, Meng. So there's no question from the audience, but I had a question for the bioequivalent assessments tool that you mentioned in the presentation. What's the, are you including both PK and PD bioequivalence, or is that only PK bioequivalence?
For the BE assessment, whether PK or PD study will include, it's product specific. So we cannot provide any general say whether it's a PD, PK, PD, or both. It's it's a definitely product specific. I guess you are talking about the HTE approach applied to the PK/PD study, right? Are you talking about that?
Well, about the bioequivalent assessment tool that you mentioned that you're not presenting, and also for the
That.
Yeah.
That it's more like focusing on the streamlining the label intensive work during the BE assessment, something like information retrieval. For example, our reviewer, in order to conduct the review, our reviewer has to collect a lot of information from applicants submitted the document, and also some information from drug labeling and whatever information related. Those work actually it's more label intensive work and less human intelligence involved. So we are, but it's very time consuming. So we are developing tool to streamline those works. So far we have 3 steps to finish that. 3 parts information can be covered. The first one it's a submitted PK data streamlining to make the submitted data executable for our internal reviewers. And the second part is we call it the table conversion, because we will receive a lot of table information from submission from the applicant. Then we make those information smoothly and automatically transfer to the document or whatever files, so that our review can directly start work on. And the third part is we make the routine PK statistical analysis automatically, make it automatically around the statistical analysis, so that our review can focus on their a review comments on the results. Yeah, that's general description about the project.
Thank you, Meng. So now we will move to the second presentation. Our second speaker will be Nadia Taranova from EMD Saronno. She is a biomedical engineer by training with a PhD in bioengineering and bioinformatics, and currently serves as the head of advanced data analytics in quantitative pharmacology at EMD Saronno. She joined the company in 2013, and here Dr. Taranova made impactful contributions to the company and R&D organization in systems pharmacology models, pharmacometrics applications of advanced machine learning techniques to complex and multivariate drug development problems, across oncology, immuno-oncology, and multiple sclerosis programs. And around these topics, Dr. Taranova is a leading author of several papers in selected journals. She is a Page Liu-Shiner awardee and a mentor of the CPT editorial board, the JPKPD editorial advisory board, and ASCPT pharmacometrics and pharmacokinetic steering committee. With this, I would like to welcome Dr. Taranova, and please take it away.
Thank you for the introduction. It's my pleasure being here today to share our work on explainable machine learning modeling for prediction of disease activity in patients with multiple sclerosis. Here the outline of my presentation. I will start by giving some background on the analysis objectives and clinical data used for the analysis. I will then go through the machine learning workflow and present results from model prediction of disease activity in MS patients. I will conclude by making some remarks. So this work was recently published in CPT PSP journal, and is the results of a great teamwork and cross-functional collaboration between our Merck quantitative pharmacology team and global medical affairs colleagues. So the team brought to us the clear patient-focused ambition of enhancing understanding of underlying mechanism of disease activity in MS and the factors driving it through its early prediction. This has indeed a great potential in enabling better patient monitoring, treatment planning, and potentially patient selection. So to this purpose, we integrated demographics, response data, MRI, and neurological assessments collected in Cladribine trials to predict whether a patient will have disease activity in 3 and 6 months. Or if you want, try to identify which covariates collected 3 or 6 months earlier can inform whether a patient will experience disease activity. We use data coming from Cladribine trials. Cladribine is an oral disease-modifying drug for use in MS treatment. The recommended dose is 3.5 mg per kg, consisting of 2 annual courses, each comprising 2 treatment weeks, 1 month apart, starting at the beginning of 2 consecutive years, for a total of 4 treatment weeks. We combine data from 3 phase 3 pivotal MS trials, Clarity, Clarity Extension, and Oracle. The pooled dataset resulted in a richly informative population of more than 1,900 patients receiving placebo, Cladribine 3.5 mg per kg recommended dose, or the 5.25 mg per kg higher dose, which was also explored in these trials. And we had a very long observation period of more than 6 years. So with heterogeneous baseline and time-varying covariates that can enable the exploration of the relationship of multiple efficacy endpoints with patient baseline and time-varying characteristics, as well as treatment effects. Our endpoint was a disease activity event, which was defined as a composite of well-known clinical endpoints measuring various dimensions of disease activity, qualifying relapses, T1 gadolinium enhancing lesions, T2 lesions, EDSS, progression over 3 months, as well as the switch to other disease-modifying treatment, which in fact, the switch, it's a marker of treatment persistence and can be driven by the lack of efficacy or tolerability issues. Indeed, disease progression in MS is quite complex, and thanks for the introduction and for inviting me to share our work on machine learning-enabled pharmacometric workflows. Big data and technological innovation have really revolutionized medicine and healthcare over the last decade. So these advances in biology, biomedical engineering, and computational sciences have resulted in an explosive increase in our ability to generate and store multidimensional data coming from diverse sources, laboratory data, clinical trial, real-world, omics, images. And these open up for big opportunities for a consistent real-time integration of these data into model-informed drug development approaches. Indeed, this can help us to advance precision medicine and to improve understanding of the disease, underlying physiological and biological underpinnings, drug mechanism of action, identification of promising combination, as well as characterization of sources of population variability in patient response, enhancing trial design and operational efficiency, diagnosis, individualized treatment, and precision dosing solution. So having the right tools in place to manage, interpret, and exploit the vast amount of data that are being generated, it's absolutely critical to unlock the value of the data and clearly demonstrate it to the patients and their caregivers by making a positive impact on patient outcomes. So advanced analytics are a strategic enabler of translational medicine. With multiple data sources at its center, translational medicine relies on quantitative integration powered by multiple advanced analytical solutions. These include also AI, machine learning, so artificial intelligence and machine learning, to innovatively support forward and reverse translation while building confidence in the 3 strategic pillars of translational medicine: target, patients, and dose. So focusing on model-informed drug development, application of AI and machine learning into population drug disease modeling, translational system pharmacology, mechanism models, it's really becoming crucial to accelerate analysis and enable the integration of a variety of data amenable to AI and machine learning. And in fact, there are multiple opportunities to advance MIDD at various levels through integration of AI and machine learning. So these approaches can be used to automatize workflow for modeling and data extraction by making them more efficient. But beyond efficiency and computational cost, several efforts have been made already to advance our models by increasing the confidence and the accuracy of the prediction. So integrating, for example, machine learning derived metrics or signatures that can help us to better understand certain mechanisms and factors influencing disease evolution, or even going above and beyond through machine learning models forecasting disease progression based on early high-dimensional data. So with the number of publications coming out over the last year in this field, you see highlighted in colors those coming from our Merck Quantitative Pharmacology Advanced Data Analytics team. And you can see that these go across all levels, and we have been even pioneering advancement in certain areas. So in this specific session today, I will provide an overview of the works highlighted in blue, which cover the application of AI machine learning to enhance standard pharmacometry workflows towards more efficient model building and covariate screening involving high-dimensional datasets. But later today, in the second session of the workshop, in fact, I will also cover the work highlighted in pink. So among the techniques available in MIDD, population modeling, in particular nonlinear mixed effect modeling, it's a tool that allows us a complete characterization of PK/PD and response in a patient population by quantifying the sources and extent of variability and uncertainty in the underlying responses. So building these kind of models, it's a step-by-step approach towards a fit-for-purpose model. So first, we build a base model which reflects a physiological understanding coupled by a statistical model reflecting the observed variability. Here, a certain number of model structure is assessed, and then the variability of the model parameter is modeled to reflect the observed differences across patient profiles. Then, based on this base model, we proceed with the covariate assessment to investigate really which factor lead to different patients' profile, what is the inter-individual variability, and how we can explain it by looking at these factors. So the outcome of this leads to a final model where the statistically and clinically significant covariates are retained on model parameters for which they can explain part of the observed variability. But this workflow can be repetitive and time-consuming step-by-step process, which is done by the analyst, especially in the case of model selection. On top, standard methods cannot handle high-dimensional dataset, thus limiting us to expand the assessment to large datasets without making prior assumptions. So we have tackled both challenges in the different steps of the model building by developing dedicated machine learning frameworks, which we tested in a population PK setting and then also use in real case studies. So in particular, for the base model building, we have assessed the use of genetic algorithm and neural network for model selection. So specifically, the AI genetic algorithm was used to automatize the model assessment by integrating genetic algorithm-based selection of models, still evaluating on a pharmacometry software, which was Monolix. In less than a day, our base model was selected from a library of structural and statistical models with a computational performance which was remarkable compared to the days and weeks that this procedure takes usually to the analyst, depending on the model complexity and runtime. So basically, we had this in less than a day. But even more impressive was the performance of the neural network models, which were able to infer the value of a model parameter or just the presence or absence of a specific component in the PK model by giving in input the data profiles. So in few minutes, we got our model advised by the neural network. This work has been published in JPKPD. But let's move now to the other step of the standard stepwise model building, which is the covariate assessment, and which is the main focus of my presentation today. So here we look in a parallel analysis, which is described in another paper, still published in JPKPD. Here we aim at assessing machine learning methods like random forest, neural network, and support vector machine regression for covariate screening, and compare the results to traditional pharmacometry selection-based methods in terms of accuracy and computational cost. The pharmacometry approaches that we use and we refer to were lasso, stepwise covariate modeling, so SCM, InnoMem through PSN, and COSAC, conditional sampling used for stepwise approach, which is implemented in monoliths coupled by Samba. So the performance of the machine learning and pharmacometry methods in identifying covariates being true factors explaining the variability across patients was assessed under various scenarios, which differ in the complexity level or the definition of our true model. So starting from a simple one-compartment PK model with oral absorption, we generated virtual populations, so individual PK profiles over time, according to 38 true models or scenarios. And for each of these scenarios, we predefine a number of true categorical and continuous covariates on clearance, false categorical and continuous covariates, and a certain correlation across the covariates, effect size, and the shrinkage. So for example, for scenario 2E, we had 2 true covariates, 1 categorical, 1 continuous, 2 false covariates, 1 categorical and 1 continuous. We assume high correlation among covariates, but also a small effect of the true covariates. So based on these scenarios, we generated PK profile for virtual population, 100 virtual population for each of the single scenarios, and these were input along with false and true covariates into the machine learning and pharmacometry models to then assess the selection of the covariates from these methods. So pharmacometry methods provide already a covariate selection, which is based on p-values for forward selection, backward deletion processes. But for machine learning, we use criteria. We had to set up criteria based on the importance scores coming from the methods to make a covariate selection. Once we have such selection of covariates for all methods, we can then compute some metrics based on the confusion matrix. For example, the true positive rate, so the rate of the true covariates which were correctly selected as significant or important, or a false positive rate, so the rate of false covariates wrongly predicted as significant or important, and so on. So we can derive sensitivity, specificity, and then calculate, for example, for machine learning methods, these metrics for various thresholds of covariate selection. Based on this, we can then construct the ROC, the receiving operating curve, and calculate the area, the EUROC, which provides a measure of how well a method separates the classes, so the true and the false covariates, with EUROC being 0.5 for random guessing towards 1 for excellent outcomes. So SCM, COSAC, lasso provide only one covariate selection once parameters are set, so like p-values. So the ROC generation, it's not very practical for pharmacometry methods. So for this reason, to compare machine learning with pharmacometry covariate selections, we compute F1 scores, which then allows to compare these methods. And this score range from 0 to 1, with 1 indicating perfect precision and recall. So let's look at some results now. So with respect to EUROC, similar results were obtained across all machine learning methods, with neural networks showing slightly better performance. So in this slide, we have an example of 6 scenarios, with 2 categorical, 2 continuous true covariates, 5 false categorical covariates, and 10 false continuous covariates, where we explore 6 different levels of covariate correlation and effect size. We can see that the scenarios with high effect size, so scenario B, D, and F, achieve EUROC equal to 1, which means a perfect separation of the classes or identification of true versus false covariates. Overall, across all scenarios, we could see less precise selections when we had low effect size. So somehow the signal was not so clear, right? It was low. So it expected. And the number of overall false covariates, which can be seen as a noise, did not impact the ability of machine learning to correctly identify true and false covariates in these scenarios. Overall, for all machine learning methods, we had very good EUROCs with an average of 0.9 and a perfect separation, so EUROC equal to 1 in several cases. But let's now look at the comparison across machine learning and pharmacometry methods by looking at F1 scores. So in this table, you can see the average F1 scores as well as the number of scenarios where a perfect separation F1 equal 1 was obtained across all the explored scenarios. We can see that similar performance in terms of average F1 score is achieved across machine learning methods and also pharmacometry, although machine learning slightly outperform SEM or has a similar accuracy. SEM and COSAC provide similar results. However, only machine learning methods provided a perfect selection F1 equal 1 for all the 100 dataset within certain scenarios. And in particular, in 18 scenarios for random forest, 16 scenarios for neural network, we had a perfect selection out of the 38 scenarios. So when assessing the ability of SEM and COSAC to recover on top of the true covariates, also the true covariate parameter relationship, of course, F1 score decrease. This is expected, right? Then, with respect to the components that we had in the scenarios, the size of the covariate effect and the shrinkage are those having higher impact on the results. But if all the models perform extremely well, the computational gain with machine learning was really notable, from simple to complex scenarios. So despite the additional computational resources, the SEN runtime exceed 3 days for some scenarios, whereas machine learning methods are run even in less than 3 hours, even in the complex scenarios. On average, we could see that COSAC was about 4 times faster than SEM, with the computational cost ratio between SEM and COSAC increasing with the number of covariates, so suggesting that COSAC it's more quick basically than SEM, also when increasing the number of covariates. So machine learning, it's at least 8 up to 225 times faster than SEM and 2 to 61 times faster than COSAC, with random forest really providing covariate selection in few minutes for both simple and complex scenarios. So this machine learning performance was also confirmed in more complex scenarios out of the 38 scenarios involving 100 covariates, and random forest could provide accurate covariate selection in 1 hour and a half, while SEM even failed as the run continued for more than 18 days. But also, we tested this framework in a real use case, where we ran pharmacometry and machine learning-based covariate selection for a more complex 2-compartment PK model with Michaelis-Menten and linear elimination describing the PK of cetuximab. We had 30 covariates assessed on clearance and central volumes, and the machine learning methods provide very consistent results with similar selection of covariates in less than 10 minutes. But we could not make any comparison with SEM as the runtime really exceeded the 19 days. So taken together, these results really indicate how machine learning methods can greatly increase the efficiency of population covariate model building in the case of large data sets or complex models that require long run times. with accuracy equal or higher to SEM and COSAC. In fact, machine learning methods could provide accurate covariate selection in few minutes to hours, depending on the complexity of scenario, while pharmacometry models will require much longer time, even more than a day. So this kind of machine learning framework indeed can efficiently be used as part of our pharmacometry pipeline. For example, to provide a fast and initial covariate screening, which can then be followed by more conventional pharmacometry approaches to assess the clinical relevance of the selected covariates and then build the final model. And in fact, this is what we have done in a real study. I would like to take the last few minutes to sharing with you the successful use of such machine learning methods to assess prognostic and predictive factors of long-term overall survival and tumor growth dynamics in one of our avelumab studies. So the Javelin Gastric 100 phase 3 trial did not meet its primary objective of demonstrating superior overall survival with avelumab maintenance versus chemotherapy in patients with advanced gastric cancer. However, we could see that the OS, so overall survival rate, was much higher with avelumab at time points after 12 months. So these results raise the question to the team about whether a subpopulation exists that could benefit from avelumab. To this purpose, then, we developed time-to-event model for overall survival and a model for tumor growth dynamics, and then use machine learning to expand the investigation to a vast set of baseline and longitudinal covariates in the prediction of these outcomes. We use random forest and site screens to assess prognostic and predictive markers from a pool of 89 covariates for overall survival and 52 covariates for tumor growth dynamics. Of note, this assessment included longitudinal covariates as well. So based on the variable importance coming from machine learning methods, we then included these covariates into our time to event and tumor sites models to assess the clinical relevance. So at the end, no subpopulation for which avelumab was superior to chemotherapy could be identified, but this was reassuring the team decision on the gastric cancer trial. And with respect to the overall model, this could be used to support the lack of differences in overall survival and tumor sites between Asian and non-Asian population, which is extremely valuable to inform the design of Asia-inclusive trial in gastric cancer in post-induction setting. So to conclude, by using machine learning, we really have the opportunity to reinvent the pharmacometry model building pipeline to mine large data sets in more automatic and efficient way. And in fact, depending on the question and the stage of the model building process, these approaches can be used to improve model selection and covariate screening separately, or in fact, altogether with an end-to-end approach advising the model base and the covariate selection at once. Or in fact, if we are interested in the prediction of specific outcomes, irrespectively of the underlying mechanism and model assumptions, machine learning can be used directly without any pharmacometry model building. And with these acknowledgements, I'm now happy to take any question. Thanks for your attention.
Thank you, Nadia, for your presentation. We have one question from Simon Carter. The question is, when taking the top X of covariates to then use in a pharmacometrics model from slide 16, where machine learning methods rerun, as all the covariate inputs may interact non-linearly, could this affect the output and conclusions?
Yeah, just reading. So basically, I mean, that's part accounted somehow in the machine learning model. It's also something we do directly, and with pharmacometry somehow we filter out. I mean, we have, in fact, the correlations was something we did test into the various scenarios to understand how these was going to have an impact on the results. And we could see, so with different levels, you can see that there was low correlation up to very high correlation across true false covariates and among all of them. And in fact, there was no impact of the correlation across covariates on the results. And this was the case across all methods we tested.
Okay, there's another question from Anna Victoria Pons Barbadella. Can this method be used for time-dependent covariates?
That's a good question. So the framework, as we did develop directly, I don't think can move to that. In our case, we really were looking at baseline or like a constant time-invariant covariates. But definitely, we will see also maybe in the next talk, there are ways to take into account and describe profiles over time by using machine learning. So in this specific case, it was really a proof of concept of how machine learning is positioned with respect to standard conventional pharmacometry approaches, but definitely it can be expanded beyond that. Yes, it's a very good point. Thank you.
And then the next question is, are the machine learning methods being applied within a LLME estimation framework or being applied to individual estimates of clearance from the base PK model?
Yes, so they are applied to the true model. So basically, we did generate virtual profiles, assuming that we have really running PK study. And by using true models, we do generate these profiles, and we also have true estimates at the individual level. So this is what we are using as an input. So somehow it's how much the model is able to recover the true parameter for each individual, and then you look at which factors, which covariates are providing the most contribution into that prediction.
Okay, the next question is, do you see integration of machine learning model with compartmental model for better prediction? Could you please provide your comment?
Sorry, can you say again?
I think we're running out of time, but the question is, do you see integration of machine learning model with compartmental model for better prediction?
Yeah, definitely. I mean, I think maybe we will take this in the panel because it's gonna take some time to answer fully. But yes, we are seeing various attempts within the community, right, to leverage and make more efficient our workflows. And indeed, that there has been some work also with respect to compartmental analysis and integration of machine learning on that.
Okay. All right, we'll move on to the next talk.
Thank you.
And I would like to hand over the mic to my colleague Pawan for introducing the next speaker.
Yeah, thanks, Kumpal. Hi, everyone. Good morning. My name is Pawan Badadi. I'm a Senior Director and Head of Advanced Pharmacometrics at Daiichi Sankyo. I'm one of the co-moderators for session 1, and will be introducing the next set of speakers and moderating the question and answers at the end of their respective talks. I encourage all of you to type in your questions as the presentation is ongoing, and note that for questions that are not answered during the session, we'll send them to the speakers and panelists offline. With that, next up, we have Dr. James Liu. Dr. Liu is a principal AI scientist in modeling and simulation clinical pharmacology at Genentech, where he currently leads a team of AI/ML scientists in applying and developing advanced algorithms to clinical data. Prior to that, he worked in modeling roles at Roche in Basel, Switzerland, and AstraZeneca in Cambridge, UK. He's an applied mathematician by training and obtained a bachelor's degree in mathematics and PhD in computational fluid dynamics from MIT. After his PhD, he performed postdoctoral research in mathematical and systems biology within both academia and industry. Over the past years, he has contributed to QSP and AI/ML efforts from both the safety as well as efficacy perspectives in various therapeutic areas, ranging from lipid metabolism and ophthalmology to oncology. He has authored over 30 refereed publications in various technical journals, and he's the co-inventor of several AI/ML patents. He's given presentations at major conferences, including PAGE, and he and his team have been selected to receive the ACOP Quality Abstract Awards in 2021-22 based on their work applying deep learning to clinical PK and PK/PD data. The title of his talk is Neural PK/PD as a Pharmacology-Informed Deep Learning Architecture. I'll now turn it over to Dr. Liu. Please take it away.
Hello, my name is James Liu. It's my pleasure to present to you at the IQ workshop. In this presentation, I'll discuss our work on the methodology Neural PK/PD, which is a pharmacology-informed deep learning architecture. As motivation, why do we need to consider machine learning approaches in data modeling? One key reason is that we are currently facing the challenge of needing to draw insights from an ever-increasing amount of data, thanks to advancements in technology and digitization. This leads to a growth in, firstly, the number of patients. As opposed to just working with clinical trials, we are seeing an increase in the number of patients available from real-world data. Secondly, through the availability of wearable devices that can collect data continuously, there's going to be a huge increase in the number of longitudinal measurements. Finally, through advancements in imaging and sequencing technology and so on, biological data at ever finer resolutions are becoming available. Therefore, we need algorithms that can scale computationally with these technological advancements. The concept of neural network has been around for quite some time, and to my knowledge, the proposal for using it for PK and PK/PD modeling first came about in 1997. In this paper by Goldberg et al., a 3-layer feed-forward network was proposed, with the inputs including quantities like time, dose, and PK, and the output being the PD variable of interest. And in between the input and output is a single hidden layer that learns to perform the input to output mapping correctly. A similar approach was proposed by Chow et al. To perform PubPK analysis. Here, the inputs are covariates such as age, gender, and so on, and a single hidden layer similarly was used to predict the drug concentration. Since then, there have been some follow-up efforts as well, notably by Major et al. And Khan et al. And others. As we know, in the recent decade or so, there has been a deep learning revolution whereby neural networks consisting of many hidden layers were able to achieve predictive performances that were previously unprecedented. This gave rise to a renewed interest in using novel architectures to carry out PK/PD modeling. This includes the use of recurrent neural networks, including LSTM, to model PK/PD data, as pioneered by Liu et al. And more recently, we've developed methodologies based on neural ODEs, which can directly express concepts of dynamical systems and pharmacology that we believe are important for PK and PD applications. As we aim to develop novel algorithms for PK and PK/PD modeling, we need to view them from a dynamical systems perspective. From this perspective, it's important to distinguish between the two types of dynamical systems, that of autonomous systems, where a given time-invariant law governs its evolution, versus that of non-autonomous systems, where there is an explicit time dependence. From this perspective, it turns out that PK/PD are piecewise autonomous systems whereby dosing introduces perturbations at the times of dosing. As we see from the equation below, apart from the times of dosing, the system evolves autonomously. Furthermore, in pharmacological modeling, it is usually assumed that there is a causal chain from dose to PK and then to PD. And this is a concept that needs to be made explicit as one considers machine or deep learning models. Over the recent years, there has been a huge growth in the application of deep learning models for time series data, including in fields such as finance and tech and others. However, I argue that PK-PD modeling differs from many such application areas. In particular, the standard deep learning architecture for time series such as LSTM do not explicitly encode causality relationships between dose, PK, and PD, which we believe is crucial to incorporate. In particular, in LSTM, such as the one schematically represented on the right, treats all input data on an equal footing as simply a vector X, with no specific meaning between the variables being encapsulated. It turns out that this causes challenges in performing counterfactual or what-if simulations. This issue has motivated the development of LSTM-based architectures by Brehm et al. That explicitly separates out the concentration data, for instance in PK, versus the dose data. While this approach provides a possible avenue, we would like to consider other neural network architectures that are pharmacologically informed. We believe the right approach is to merge deep learning with dynamical systems at a fundamental level. On the one hand, we would like to leverage the ability of deep learning to excel in approximating high-dimensional nonlinear functions, and doing so in a way that improves the model as the amount of data increases. On the other hand, we would like to build in concepts of dynamical systems that enables assumptions of specific causal dynamic relationships. Crucially, we would like to implement dosing as a perturbation in the system state, as we've seen previously. The solution that we propose is neural PK-PD, which seamlessly intertwines these two complementary concepts. And the result is a pharmacology-informed dynamical system that learns from data and enables counterfactual dosing simulations. So what are the key components of the neural PK-PD architecture? First of all, the PK-PD data at the patient level is passed to an encoder, which has the task of performing data abstraction at the patient level. As we'll see later, we aim to do so in a pharmacology-informed manner. And the output of the encoder is a low-dimensional embedding vector denoted here as P for each patient. And this vector P then goes into the decoder, which is an ODE system with the right-hand side represented by a neural network. The aim of the decoder is to perform predictions using a single system of ODEs, taking as input the patient embedding vector P. In particular, the dose amount can correspond to the actual values given to the patient in question or to simulate a novel dosing regimen. We believe there are several benefits to this pharmacology-informed encoder-decoder architecture. Namely, this provides explicit control on the dimensionalities of parameter space and state space, respectively, while the neural network may involve tens, if not hundreds of thousands of trainable weights. And finally, the explicit dosing port in the decoder ensures that counterfactual simulations can be performed. As a comparison between the standard formulation of PK-PD models versus neural PK-PD, we note that in the human generator model, the equations are expressed in analytical forms based on human experience and fit to the data. In contrast, in the neural network model, the network weights are learned from data under the specified constraints on the number of linear layers and activation layers in between. In particular, the same architecture could be used to model PK-PD data from different drugs, whereas in the human generator model, it has to be adapted to the data and assumptions for each drug of interest. We've previously mentioned that causality assumption is a key part of what we would like to capture. How is that encoded into the neural PK-PD architecture? This is done in the following manner. On the left, we have a patient's PK-PD data in a tabular form. Of these, we are using the first three columns to feed into the PK encoder with the aim of quantifying the dose to PK relationship. And once the vector P for the PK model is computed, it then goes into the neural PK model with the given doses as the driving force of the system. And once this is done, we then consider the PD part of the model. In this case, the time, PK, and PD columns go into the PD encoder with the aim of quantifying the PK to PD relationship. And once the vector P for the PD model is computed, it goes into the neural PK-PD model with a time-dependent PK state, providing a causal influence on the PD. This general architecture ensures that dose is what drives PK and PK is what drives PD in the causality chain. We've shown that the neural PK-PD model can learn from data to produce dynamics that is, encouragingly, qualitatively similar to the state-of-the-art PK-PD model for platelet dynamics that have been finely tuned to the data. In particular, comparison of the dashed green line together with the solid blue lines show much similarities in the decay and recovery dynamics across the 6 patients shown here. Quite notably, the steady decline in the peak values of predicted platelet count over long-term dosing is captured by both models. While the qualitative aspects of the two models are similar, on exactly the same trend and test splits, we show that the neural PK-PD model can give rise to superior predicted performance at the individual patient level. And this is true independent of how much observation data is used across cycles 1, 2, 3. We believe that the reason that the neural network approach is able to effectively search over a wider space of equations explains this improvement in accuracy. This benefit of the deep learning approach may also open up future applications in precision medicine. As we previously mentioned, one key use of PK-PD models is to simulate untested dosing regimens. Hence, assessing the ability of neural PK-PD models to do so is of the paramount importance. Through the architectural design, this can be performed in the following manner. While the encoder is fed the actual doses and PK data, the decoder can be driven by the counterfactual dosing regimen. And we show that the model can simulate what if all the patients were treated once every 3 weeks or once every week and show predictions that are consistent with the data. In addition, it can also be used to simulate a totally novel scenario, once every 3 days dosing, which is a novel regimen that no patient has been treated with. The model predictions are showing the expected behavior of PK and PD dynamics, which reflects the benefits of pharmacology-informed neural network architecture. The question of whether the architectural choice of machine and deep learning models affect the ability to generalize was explored in this article we published in iScience. In it, we compared a few models, including LightGBM, which is a tree-based machine learning model, and LSTM, which is a commonly used neural network model for time series data, and finally Neuro-ODE. These different models differ in how the input data is used, and hence associated with them the different inductive biases in making generalizations. When we compare the results, we showed that all of the 3 machine deep learning models, ranging from LightGBM to Neuro-ODE, have similar predictive performance when the train and test set are from the same distribution, as measured by metrics such as RMSE and R². However, when the train and test set are from different dosing regimens, the 2 models, LightGBM and LSTM, do not generalize well. We believe that this is primarily due to the fact that causality relationship between dose and PK is not incorporated into these formalisms. And the lack of dose explicitly driving PK is also apparent when one looks at simulations on the right, where we've compared PK predictions for the nominal dosing versus when dosing is stopped at time equal to 2,500 hours. We see that the ODE-based models with dosing being the forcing function have PK correctly going to 0 after time equal to 2,500 hours. However, for LightGBM and LSTM, that's not the case. Finally, I would like to end the presentation with some concluding remarks. Firstly, due to the rise of complex PK/PD data and the increase in its volume, we'll clearly need to develop AI methodologies. Secondly, while there are many alternatives for machine and deep learning methodologies, we believe neural PK/PD has some clear benefits in terms of it being a pharmacology-informed deep learning architecture that encapsulates the dose concentration effect relationship within an encoder-decoder architecture. We believe these offer advantages in terms of explainability as well as generalizability. Finally, I would like to acknowledge colleagues at Genentech and collaborators at the University of Michigan and Ann Arbor Algorithms for their contributions. I'll be happy to take questions. Thank you for your attention.
Thanks, James. Couple of quick questions. So the first one is, what type of deep learning network is the encoder-decoder?
Okay, so because we need to deal with PK/PD data that have arbitrary number of rows, we need a RNN or recurrent neural network type architecture. And on top of that, one could add different mechanisms like attention to be able to deal with differences in dose and PK versus PD measurements. So that's what we've done.
Thank you. Another quick question is, can you comment on the applications for this approach in simulation? The need for PK/PD data as an input layer would seem to be limiting beyond the training population.
Okay, so yeah, certainly. Working with a limited set of data is one challenge in deep learning in general. However, there are approaches that one could potentially leverage based on transfer learning just like a human modeler seeing PK/PD data is not seeing that for the very first time. There are certain components that one can use a pre-trained model to start off with. So perhaps that can help.
Okay, thank you. Yeah, I see a lot of questions, but we'll try to answer them offline. So let's move on. Thanks again, Dr. Let's move on to the next speaker. So next up, we have Dr. Cambrien Poles. Dr. Poles is a senior scientist in the early clinical development QSP group, where she supports the oncology research unit. She obtained her PhD in biostatistics from Harvard in 2022. Her dissertation focused on building tumor evolution models to identify optimal drug dosing schedules in combinational therapy. After her PhD, she joined Pfizer ECD QSP as a postdoc, where she was able to refine her skills with mechanistic models of cancer and drug response. Using her training in statistics, she has been able to employ more data-driven approaches when building or calibrating mechanistic models at Pfizer. Her interests primarily lie in refining her knowledge in machine learning and combining ML methods with QSP or mechanistic models for oncology research. Today, she's going to talk about a machine learning-based approach for toxicity predictions in immuno-oncology. Dr. Poles, please take it.
Thank you very much for the introduction. I will be the first live presentation for this workshop. I will go ahead and start. So the title of my talk today is machine learning approach, machine learning-based approach for toxicity predictions in immuno-oncology. I am a senior scientist in the early clinical development quantitative systems pharmacology group, or QSP for short. And I will start off my talk first about taking a step back and introducing really what we're doing at QSP. So this diagram, which we shamelessly took from a presentation of the FDA a while ago, shows the expanded modeling and simulation approaches in support of model-informed drug development. As you can see, among these different methods, we have the exposure response, PBPK, PKPD, as we've talked about. But here's where I currently sit in the QSP field. QSP is a field of biomedical research that aims to model the mechanisms behind disease progression and quantify the pharmacokinetics and pharmacodynamics of pharmaceuticals using mathematical computer models. The QSP model presents a system that can be any biological or population process related to a disease, such as the physiological consequences of such disease, a specific disease pathway, like signal transduction. We can also model systems within the omics field, like genomics, proteomics, metabolics, etc. QSP models are constructed carefully in a bottom-up approach, and we rely heavily on biological and biophysical theory. So when these models are being constructed, they are usually in collaboration with other experimentalists and as well as physicians, biologists that can provide their input. QSP not only can predict pharmacodynamics, clinical efficacy outcomes, or toxicity events, but also it can provide an insight into the system being modeled when perturbations of interest are incorporated. So in the context of the symposium, our goal in QSP is not to use AI ML to replace our methods in QSP, since they're so carefully constructed with multiple collaborations typically, but also it is to combine them. And so we quote this nice sentence from this article where I've included the link at the bottom. So combining the best of both worlds of QSP plus ML and AI. Investigators suggest that when scientists from the machine learning and mechanistic modeling disciplines interact and learn from each other, they will find many overlapping concepts, equations, and ways to visualize model structure architectures. And the machine learning models and more traditional mechanistic ones may complement each other to improve the efficiency of drug development and optimize treatments for individual patients. So it is one of my main interests as I was introduced of combining QSP and machine learning methods to take our drug development to the next level. So in this application where we are interested in modeling toxicity in immuno-oncology, I will present our motivational example, the cytokine release syndrome, or CRS for short. This can occur during bispecific antibody treatment for cancer patients, also seen in other therapies such as CAR-T, etc. The mechanism of how this works is that the bispecific antibody links a T cell and a cancer cell. And this binding proximity of the T cell and the cancer cell can triggers T cell activation, release tumor killing agents, but also this T cell activation can cause a release of toxic cytokines. Starting off with an on-target activation will lead to then non-target activations, and the excessive cytokine, an excessive amount of cytokines are released. This usually leads to CRS, and in clinic, this adverse event is graded is a graded response. So there's still a disjoint between what we understand of what will lead to a CRS event and how it is graded or detected or classified, I should say, in clinic. But as I mentioned, there's a CRS grading in clinic, starting with grade 1, very light symptoms such as fever, and then proceeding all the way to the severe grades, which starts at grade 3, where there's some hypoxia, as well as some organ failure. So we, at Pfizer, especially in our QSP group, we are thankful for the opportunity of being able to mentor interns almost every summer. And I will need to first start off by thanking Miriam Bell that she joined us last year as a summer intern. She was getting her obtaining her PhD from UC San Diego, has obtained it since. But she led this this project of constructing this QSP model of CRS with priming. And this model was constructed with in a preclinical environment, where it included key biological features such as the tumor killing, cytokine release. We've also included the more complex features such as cytokine feedback, where IL-10, specifically an anti-inflammatory cytokine, will inhibit the production of other cytokines. As well, there was an implementation of an internal inhibition mechanism for T cell desensitization. And then the last key feature is the, which has been explained in literature that is referenced below this slide, is that the dose priming, in this case starting at lower amounts of drug, can reduce CRS, but obviously the goal is to maintain efficacy. And so going to the diagram to the right, this is a schema of the model that Miriam had put together. It can capture key features for drug efficacy and cytokine release. It's a mechanistic, traditional model. However, there is this, as I mentioned before, the mechanism of dose priming is not completely understood. So we have an area on the right side of that is boxed in yellow that is more of a phenomenological model component here, that we can capture the effect, the dose priming dynamics, but we are lacking the biological basis here. So we are not able to, we can simply attempt to describe the relationship of the inputs, the synapse, IL-10, into then the output, which is this inhibition we have here of the cytokine release. But we cannot extend past that point. We cannot describe why the variables interact the way they do. So now the interesting part is that well, I think everything is interesting here, but where AI and ML can help us out. Well, this is a example of a workflow of hybrid QSP AI modeling for antibody therapy. On the left side, I have boxed the clinical efficacy in CRQSP models. So the model I showed before, I mentioned that it was built in the preclinical stage. So we need to take it to the next phase, to the clinical model. So there are more sophisticated QSP models of efficacy for such therapies that are published in literature. And here I'm showing a schema of one published in 2018. But these efficacy models are composed of different compartments to model to more closely represent drug trajectory and tumor growth response dynamics in larger organisms. Through the merging of QSP models, in this case, this clinical efficacy model I'm showing right here, and then this cytokine release mechanistic model, we can acquire a more insightful QSP model as long as we keep supporting biological theory. So then this combined enhanced QSP clinical model can output efficacy endpoints, as well as biomarker trajectories related to CRS. These biomarker trajectories are the cytokines, as you can see in the center of the screen. And where we can use artificial intelligence and machine learning in our shown framework, or the first area that we've identified, is in this boxed dashed red component, and that is using a CRS classifier. This CRS classifier would be a machine learning model trained and tested on clinical as well as maybe preclinical data, and we can take those learnings from this machine learning model and input QSP model outputs to this and predict CRS. So again, going back to the amazing opportunity we have with our summer student interns, we had another student intern with us this summer, Emily Nieves, who is working on her PhD at MIT. She spearheaded this analysis by using machine learning methods to analyze clinical data for cytokine to grade mapping. The proof of concept here is to show that cytokine signaling may be sufficient for CRS prediction, and therefore implemented in our workflow that I showed it before. She applied a variety of different methods, PCA, to show that good separation between patients with CRS from those that do not have CRS, as well as running random forests to identify important variables to classify CRS grades. We've identified that dynamics rather than amounts of these cytokines are better predictors. And then we identified, and this has been already established in literature, that IL-6 and IL-10 are key predictors for CRS, and this is what we have therefore included in our CRS model that I showed a couple of slides back. And we've had a good prediction. There are some limitations in the way how adverse event grading is classified out there. So that is something that is in the process of being better understood. So that was the first application of AI ML methods, the good old prediction power of such models. Now, another component that we've in our workflow that we can use machine learning is right here, going back to our mechanistic models and uncover the biology. Can we use more novel AI ML methods to discover structure of mechanisms? And so again, this is carried by Emily. who I should say comes from Chris Rocca's lab in MIT. So this is some of the work that has come from Chris's lab. They have they propose this automated model discovery using data to learn missing terms in a system of ODEs. By the universal approximation theorem, we know that neural networks can represent a wide variety of interesting functions when given appropriate weights. So neural nets can approximate a function very well, just like the Taylor series, only that it is neural nets can do it multidimensionally or in very high dimensions. So I think I will skip over my description of neural networks, as it has been explained before in the previous talks. But before going on to the methodology, I like to address a few novel things about this specific approach that came from Chris's lab. Other popular methods for model discovery exist out there. They approximate derivatives using spline over the data points and then imply symbolic regression to the full equation. The use of interpolating spline techniques for derivative estimates require a dense enough data for accurate predictions. With these methodologies, as it has been shown in this preprint quoted here, we are able to not rely on so much data to train the model. So a very quick review of how these methods work. First, you have an ODE system, and there are some parts that we do not know how a function looks. So we replace those parts with neural nets and optimized neural nets. So the output of the ODE model matches the data. Before fully replacing these functions in our ODE, we apply sparse regression to recover the equations or enough equations of this additional term needed to reproduce the data. This method or this approach is usually implemented in Julia's science machine learning ecosystem, and they can fit the parameters of the neural networks within an ODE problem. By the way, this SciML tool for mixing the information of physical laws and scientific models with data-driven machine learning approaches is quite efficient. So I'd like to show an example of where this has been applied and how it would, closely speak to our application. So this is an example of where automated model discovery supported mechanistic modeling. In this published paper in 2021, they've used the example of modeling a term related to quarantine strength from COVID-19 data using the automated model discovery presented in the slide before. They've used an adapted SEER epidemiological model to include effects of quarantine. For those who don't know, this SIR, which stands for Susceptible, Infected, Recovered model, is a very popular epidemiological model to represent infectious diseases or model infectious diseases. In this case, they use automated model discovery to learn a function of the strength of quarantine. Here it is denoted with a capital Q and a function of T, which is time. And this QT may be using CDC data for different US states. That implies that there might be different functions for each of the states. Q of T is a composite of testing rate and quarantine policies. So then in the lower left, you will see the system of ODE equations that they've used. Notice that all areas where this quarantine rate have been replaced by NN, standing for neural nets. W represents the weight, and U represents the input, which would be the the dynamics of this ODE system itself. And as something new about this, this model from the traditional SIR is the last equation, which is the T, and that represents the population that was quarantined. So they applied the methods I explained before, and they were able then to formulate QTs, or these functions in this for this ODE system, and they the advantage of this is that they're able to incorporate and completely understood and complex mechanism into models with data without the opacity of pure ML methods. So regarding this last clause, the opacity of pure ML methods, that this has been a disadvantage that typically we are not able to infer or describe the interaction of variables between variables when in these models. But once we have, or once in this case the authors understood this epidemiological system, in other words, they diagnosed the strength of the quarantine function, they could address questions such as how many infected cases would have been reduced had the worst affected cases not reopened early. So it would be exploring counterfactual scenarios, and that is a strength that we have with our QSP models, is that we are able to extrapolate or provide more insights than other traditional statistical models. So this is a active collaboration with our summer student intern Emily Nieves and her advisor. Apply this automated model discovery system to add mechanistic detail to dose priming component and improve fits. We would learn the inhibition term that varies with time from data. The inhibition term will likely be a composite of T cell receptor downregulation and downstream mechanisms related to desensitization. Right now, what we have with this phenomenological model or component in this whole QSP process, it's we are not able to provide much more insight than that. Yeah, and then lastly, the sparse regression would help us recover those necessary equations from neural networks. That way, we can still have a simpler, structure where we are able to describe what is happening within the system. Okay, I'd like to acknowledge these members here, especially both of our student interns, Emily Nieves and Miriam Bell, as well as my Pfizer colleagues, my supervisor Berta Stille, who helped me a lot to contribute to these slides or the content of these slides. And I will now hand it over for questions.
Pavan, we are unable to hear you.
I'm sorry. Yeah, I was saying thank you for your presentation. In the interest of time, maybe we'll skip the questions to the end of panel discussion. But I think through your talk, you've answered some of the questions that came in from the prior talks, which was about, can this be generalized for larger ODE, especially for QSP models. So thank you for your talk. With that, maybe I'll hand it over to Dr. Didier Renard for the panel discussion.
Thanks, Pavan. So yeah, I think we heard 5 interesting talks so far, covering a pretty broad range of AI ML applications in drug development, and we also heard from different perspectives, including from FDA and industry. So hello everyone, my name is Didier Renard. I'm a pharmacometrics group head in the immunology area at Novartis, and I will lead you through this panel discussion. So I will first ask for all the panelists and speakers to join me. So could you please all switch on your camera? And I will actually first introduce the new panelists. So we have first Dr. Qi Liu from FDA, whom we've already heard in the opening presentation, so I won't introduce her again here. It's also my pleasure to introduce Dr. Jonathan French, who is a Fellow 2 and Science Advisor at Metrum Research Group, where he leads statistical excellence initiatives as well as R&D projects. He's a biostatistician by training and received his PhD in biostatistics from Harvard University in the area of Bayesian methods with application to missing data. In his previous career, he worked more than 10 years in Pfizer, where he held positions of increasing responsibility, including pharmacometrics group leader for both inflammation and oncology products. And Jonathan has developed expertise in decision analysis, model-based meta-analysis, and disease progression modeling. He's a fellow of the International Society of Pharmacometrics and a member of CPT PSP editorial board. It is also my, sorry, I've got some disturbance on the screen here. Also my pleasure to introduce Dr. Sarah Kim, who is an assistant professor in the Department of Pharmaceutics at the University of Florida, College of Pharmacy. She did her postdoc training in pharmaceutical sciences, focused on pharmacometrics, and has received recognition from the international MID3 community, including the David Goldstein Award and Presidential Trainee Awards from the American Society for Clinical Pharmacology and Therapeutics. As an early stage principal investigator, she is currently leading several computational modeling projects to create and innovate quantitative solutions in healthcare. And using AI, her team is further developing an imaging analysis and informatics tool to improve understanding of disease progression using imaging. And since 2020, she has served as an AI initiative committee member at the University of Florida College of Pharmacy. So welcome everyone, so to our panelists, and welcome back to our speakers. And so, yeah, maybe a quick reminder before we kick off the discussions. I think all the questions that the audience ask will be answered offline. So, right, so we will take them and I guess make some answers available later on. So feel free to continue asking questions in the chat. So even if we don't take them right now. All right, I will kick off the discussions with a question to Shi. So in your talk, you have touched upon some important points about regulatory considerations on AI/ML, such as the context of use, fit for purpose and risk-based validation, generalizability and explainability. You also mentioned the need to develop best practices. And so my question to you would be, well, could you give us maybe some practical example of best practices, which in your opinion should be adopted by the AI/ML community in drug development applications?
Thank you, DJ. That's a really excellent question, very important. So far, at least at CEDAR, we haven't have a written best practice document yet. But our colleagues at CDRH, last year, they have published 10 guiding principles for good machine learning practice together with other regulatory agencies. Although those guiding principles are developed mostly for medical device development with AI components, many of them are also applicable for drug evaluation as well. For example, the first guiding principle they emphasized was multidisciplinary collaboration, which is also very, very important for when we use AI for drug development. And they also mentioned like the clinical study participants and data sets needs to be representative of the intended population, which is exactly matches our expectation when we use AI and machine learning in drug development and clinical trial as well. Other things such as the training data set and the validation or testing data set needs to be independent of each other, which is also what we think is very important as well. There's other things such as the testing needs to evaluate the model performance during the clinical relevant condition, those are also actually really relevant to us. And also to emphasize that the user will need to be provided very clear essential informations, which we also think it's very, very important. I want to mention that actually the regulatory considerations for the AI and machine learning in drug development is still evolving, and we need input from all stakeholders in different disciplines. And in order to promote responsible innovation, we need more collaboration. And actually, maybe I can put this back to IQ. So maybe IQ could also think about maybe there are some roles IQ could play to facilitate the discussion. And maybe as a community, we can come up with our own guiding principles for the use of AI and machine learning in drug development. Thank you.
Yeah, that's certainly something I guess we can take up following this workshop. I also just wanted to ask Meng if you have any further insight, comments, also from an FDA perspective, perhaps.
Sure. Thank you. Yeah, that's an excellent question, and I fully agree what the Qi provided. Just want to add a little bit things. I personally consider the evaluation of AI machine model with no much big difference with other models. They are pretty much should be similar, rule or criteria to evaluate, like reproducibility, like transparency. Needless to say, reproducibility, but for the transparency, it could be a little bit unique to the AI machine learning because they are built this imbued property for the AI machine learning. So another aspect I want to mention is about the presentation of the data, because we all know for AI machine learning, there's a very famous saying, it's a garbage in, garbage out. So we need to be very careful about the presentation of the data. For example, for imbalanced data, we all know the machine learning algorithm will more favor to the majority subgroup in the population. So we want to present data in a very good way to show the data is valid and kind of a representative. That's my input. Thanks.
All right, thanks a lot, Meng. Maybe I forgot to mention actually that due to this issue in the video for Nadia's presentation, that we will extend by 5 minutes the Q&A, right? So we will probably wrap up around 12:40 instead of 12:35. Our apologies for this inconvenience. So maybe my next question would be for Jonathan. So as I mentioned, I think today we've already heard about a number of different applications here in drug development, and you are certainly someone as a pharmacometrician with long drug development experience. I wonder what is your perspective on how AI ML will change the way we do pharmacometrics work, maybe in 5 years from now?
Yeah, that's a great question, Didier. Well, first, I just want to say thanks to all the presenters for such great talks. Really think that the work that you're doing now is really on the cutting edge of where we're thinking about AI and ML and applications in pharmacometrics. And so I think that was really fantastic. So in terms of pharmacometrics, in the next five, let's say, I'll even say maybe out to 10 years, I think about where pharmacometrics really adds value to what we do. And it's in kind of both understanding sort of how and why things happen. So like some of the more mechanistic models that we saw today, as well as helping inform decisions that get made either at the FDA or within a pharmaceutical company or in terms of patient care. The other thing we saw is that we think about AI and ML are really good at generating predictions or finding associations between things. So those are a little bit in tension, I think, sometimes are used for what pharmacometrics is really good at and what AI ML is really good at. So I think if I had to sort of prognosticate, I'd say in the next 5 to 10 years, we'll probably see a lot of uptake in terms of areas where we're already doing empirical modeling and we don't care so much about understanding kind of the fundamental biological and pharmacological processes. So I'm thinking there about, sort of more mechanistic models, maybe some exposure response models, for example, or identifying covariates, like some of the examples that Nadia was was showing us earlier. I can see a lot of uptake in that quickly. I think what's going to be important in that context, though, is that we're really sticking to good statistical principles. It's not surprising, maybe hearing that coming from me as a biostatistician, where I think defining our estimands, maybe focusing on things like targeted maximum likelihood, and thinking very clearly about how we're using machine learning models is going to be important. I'm really excited about the work that James is doing and that Chris Rakalkas and others are doing in terms of integrating the work of machine learning and AI into sort of more mechanistic modeling and thinking about how we can leverage those tools. Personally, I think that what we're seeing now are going to be stepping stones to getting to where ultimately we want to be, and that sort of the tools are going to continue to develop and get better. And so maybe it'll take a little longer for the adoption of those, including the kind of AI ML and for more mechanistic models, because they don't, right now what we're getting doesn't really give us kind of the insight that we're looking for in terms of biological processes, but I think that's coming.
All right, thanks for the very insightful answer, Jonathan. And I'd like to follow up with kind of a related questions to Nadia. You showed a very nice picture of a pyramid in your talk to illustrate how AI/ML can advance MIDD at different levels. And this picture really resonated a lot with me. I think with the various applications we discussed today, we probably tackle pretty much the first and the second level. And so my question to you would be, what do you think is the key to unlock the power and make of AI/ML a driver of innovation in drug development? So in other words, how do we reach that top level of the pyramid?
Yeah. Thank you, Didier. That's a great question. Of course, I mean, there is not a single key, right? I mean, there are multiple keys to get there. But let's start from the basis that I think we will all agree that these are the data, right? So data right now, I mean, are in silos and are not easily findable. And we have also access within, issues within the organization, right? So the first step is really to have AI-ready data. So the FAIR data, where FAIR it's really meeting the principles of findability, accessibility, interoperability, and reusability. So that's the very first step. And of course, it comes with platforms, so infrastructure, but also a culture of open data. And as also Chi was mentioning, new cross-functional collaborations. That's really key. And it's a change that we need to have within the organization, across organizations. Then another key aspects from my point of view, and there is a lot going on there, it's about trust, right? We need to build trust. So what is that? It's really the ability to explain the outcome of a model so that the downstream decisions that are going to be taken are really conscious, right? So it's not only about interpretability, we will touch base in the second session today, but it's also about generalizability, and she also touched base on that. So really we need to build models that are not just explainable, but also free from biases. We could see the example from Chi on Amazon, right? And so trustworthiness of AI/ML, it's really crucial for scalability and building really the confidence into the results. Then we can apply and make informed decisions. And third, but not least, I think we really need to take now the time to perform an accurate analysis of the cost-benefit trade-off of applying, to demystifying the hype that we are having of AI/ML in drug development, right? So there is a lot of excitement, very high expectation, but I think we need to make an assessment and really demonstrate that the cost-benefit trade-off of applying this AI machine learning, it's much better than the conventional method. So we really need to bootstrap some investment, because of course the inclusion of all these applications comes with a cost. It's a cost of infrastructure, it's a cost of acquisition of larger volumes of data, but also building internal capabilities, making strategic investments with important companies that are working in specific fields. So we really need to demonstrate now the value. And of course, on all this, I think the engagement and the synergistic collaboration with the regulatory agency is going to be key towards developing best practices. So she has mentioned few, and for example, here in Europe, there is going to be also an AI Act on best practices, so how to perform data science and applying AI machine learning in across various domains. So I think the opportunities are there, but we really need to take some actions to move forward.
All right, thanks, Nadia. James, I believe you were the author of this pyramid, so I'm sure you probably also have some thoughts to share, and maybe also to the questions I raised to Jonathan about the future uptake.
Oh yeah, sure. Thanks, TDA. So I think right now we are seeing this huge amount of data, and there are data modalities like digital health that is really not so amenable to the more traditional pharmacometrics analysis. And there may be a lot of insights hiding in this large amounts of data. And we humans, it's okay for us to visualize data in 3 dimensions. If we make plots, PK versus PD. But as data gets high dimensional, more voluminous, a lot of measurements, I think we lose this ability to really gain intuitions from these data. And I think this is an area where AI could act as innovators to really uncover, unlock the data value, uncover new insights that would be hard for us to do kind of by human mind alone. So I see that as a huge opportunity as we tap into these new data types that have been emerging.
All right, thank you. So we've heard about regulatory and industry perspective. Time to move to academia. So Sarah, you're up now. So yeah, I mean, based on your experience at the frontier between pharmacometrics and AI/ML, I mean, yeah, could you share your thoughts maybe on where academia should focus its efforts to advance this field?
Yes. Can you hear me?
Yes.
So yeah, it's an honor to be at the frontier between pharmacometrics and AI in this academic setting. And I have the vocation to raise the next generation while making the new road with you all together. So regarding where academia should focus on our efforts to advance the field, I'd like to share my thoughts with respect to 2 key pillars, teaching and research. So for teaching, thankfully, the trainees in our pharmacometrics field are already motivated for AI and machine learning, and many of them already have quantitative knowledge and computational solid skill set ready, so which are essential for the AI research. So I think academia should focus on our effort to give them the opportunity to explore the AI applications in a pharmacy, as well as to provide tutorial-like sessions to build the skill sets. So as part of this effort, the faculty in our college is developing a graduate-level AI-focused course. It is a team effort, and throughout the course, multiple faculty mentors and invited guest speakers share their ongoing research using AI from drug discovery to development to increase students' exposure to AI application in pharmacy and build a thinking muscle to create a new research ideas. In addition to it, UF is developing AI curriculum across the entire university, and UF research computing team is providing faculty and trainees a series of hands-on tutorial sessions with support and partnership with NVIDIA. I think it is a great effort toward the direction in education. And regarding research in academic setting, the faculty investigator can pursue academic project aims driven by their research own interest, and they are quite flexible to plan on deliverable due dates. Of course, it depends on different funding source and types of grant. Even in academia, we are also always having tight timelines, but still we have more flexibility compared to the industry setting. It allows us to pursue research projects that benefit drug development in long term. So in my humble opinion, academia should pursue research aims, not only project benefit the drug development quickly, but also to the basic science that slowly but surely advance the field. So this is the type of research I consider we should pursue in academia. Thank you.
All right, thanks. So let me close the loop with Cameron then, who is a fresh PhD graduate who recently joined the industry. So you have a feet on both sides, I guess. So yeah, what's maybe also your perspective on this?
Yes. So on the industry side, things definitely move a lot quicker, as Sarah alluded to, that in academia, there might be less, there are still some timelines, but less, less tight compared to industry. So that could mean more time to develop algorithms, as well as, in the academic setting, there is an ability to be more open or present your work as soon as it comes out, like through preprints, for example, which is something that has saved me many times and has helped me for my work. So to address the question, in the fewest words as possible, I'd say that the collaboration between academia and industry is very important. I think that in academia, maybe the strongest algorithms or the more thoughtful would be able to come, or the more advanced machine learning. And then in industry, through constant collaboration, constant communication, we would be able to really test these in a more realistic scenario with real data, and through the drug pipeline, where there are timelines that are closer.
All right, thanks. For maybe the next few minutes, I will reach out to Kumpal and Pavan, who have been screening some of the questions from the audience. So maybe could you, maybe select 2 or 3 of them?
No, I can go first. So I think, thanks, first of all. I thoroughly enjoyed the panel discussion and all the great insights. One question is, how can we best turn these data-driven relationships, which are identified through AI/ML, into more mechanistic and causal applications, or causal explanations, I should say. This is probably to some of the people who are probably doing both, like maybe Cameron, Sarah, and I welcome others to comment as well. Cameron, do you want to go?
So could you repeat the question?
Sure. So how can we best turn these data-driven relationships, which are typically identified through the AI/ML, into mechanistic and causal explanations?
Yes. So these, well, as a method that we've presented with this automated model discovery, that is one, one example of turning this data-driven observed or a data-driven measured relationship into something that we can describe with words. So that would be, one example of the many other methods that are emerging in this area. So the automated model discovery neural networks.
I think I was going to add on top of what Cameron said. So I think nowadays we have a lot of knowledge graphs, and as example of OD models that I presented earlier, we assume every variable can be connected to every other, and this is not the case in many situations. If you know the set of gene interactions, or if these are variables of different cell types, some interact and some do not. And I think building in some of this graphical knowledge may be one way to build in both the mechanistic aspect and as well as neural network and its ability to approximate high-dimensional data.
Let's take perhaps one final question, Kumpal.
Yeah, hello. So I have a question to Jonathan or James. What steps should pharmacometricians be taking to make use of AI/ML explainable to scientists and clinicians?
That's a really fundamentally important question. I guess it's probably going to depend a little bit on where we're applying AI and ML and what kinds of, to which kinds of questions we're trying to answer. So I think for kind of the more empirical models, there are sets of tools out there that can help explain things like Shapley values that we can use to help kind of explain effects of predictors in sort of these more black boxy types of models like neural networks, for example, or deep networks. I don't know, for other types of models that are more ODE based, where we're kind of plugging in a black box in the middle of an ODE. I'm not really sure. Maybe James, you'd probably have some thoughts on that.
Yeah, certainly. I think visualizations can be a very powerful idea. So for instance, in ODE models, if the system is two-dimensional, you can plot the ODE the vector field to see whether the system is flowing, are there any attracted states, and so on. And of course, in higher dimensions, one may need to do this by slices. But I think rather than, in AI, we don't have explicit analytical functions being used, but they are nonlinear functions, and visualizing the functional nonlinearities in appropriate manners may already go a long way in conveying what the model entails.
All right, I think it's time to wrap up our session. Unfortunately, we are already extending by more than 5 minutes. So yeah, I hope that the session was enjoyable and insightful enough, and all the talks that were given here. And well, I hope that perhaps some of you may have new ideas about how to combining pharmacometrics and AI/ML approaches to support drug development. So I would like to thank, of course, a lot all the speakers and the panelists for joining us today. I think we have now slightly less than an hour break before the next session. So I hope you can still take some time to grab some food and come back here at 1:35 p.m. for the session number 2, which will focus on explainable machine learning for disease progression and digital twins. Thanks a lot. Bye-bye.
Thank you. Bye-bye.
Hello.
Hello, hi.
Hi.
Nadia, how are you?
Good, good. Thank you. What about you?
Yeah, I'm doing well, thanks.
Great. I
Are
just
we
saw
alone
you.
here?
No, I think people are connecting.
Okay.
hear me? Audio okay?
Sujata, it sounds okay to me.
Sounds good to me.
Thank you.
All right, I think it's 1:35, so we can go ahead and get started. Good morning and good afternoon, everyone, depending on where you are located, and welcome to our session, the second session of this workshop about explainable machine learning for disease progression and digital twins. My name is Mohammed Shaheen. I'm a clinical pharmacology lead at Pfizer, and it's my pleasure to moderate the session with my colleague Sujata Menon, who also works at Pfizer in clinical pharmacology global product development. Let me see that I'm making sure that I'm showing the right screen. Before we begin, I would like to take care of a few housekeeping items. First of all, this session includes 3 presentation speakers. We'll have 5 minutes after each presentation to answer questions. As shown on the screen, all attendees will be on listen-only mode and hand raising is disabled. For questions, please enter them at any time during the session in the question module located in the control panel, or click question mark at the top of your screen. If possible, please preface your question with speaker name and slide number. And this webinar will be recorded, so for your information, and a copy of the slides and recording will be circulated in 2 to 3 days after the webinar. And due to fluctuating local bandwidth demands, attendees may experience intermittent delays in the slide transition or audio distribution. So please click on the handout that's available on the toolbar to your right-hand side here to know more how to handle any trouble in connection. So as mentioned earlier, we have 3 speakers for this session. And it's my pleasure to start introducing our first speaker, Dr. James Kozlowski. So Dr. Kozlowski is a principal research scientist at IBM and leads IBM Department of Hybrid Biological AI Modeling, working with external collaborators in different fields, including neuroscience, cardiology, and pharmacometrics. As a manager, James oversees efforts to use AI to solve stochastic inverse problems for biophysical models of disease and drug mechanism of action. His team focuses on quantitative systems pharmacology and PKPD for model-based therapeutic design and triaging, applying deep learning methods and generative models to population parameter inference. To date, he has over 250 issued patents in the area of neuroscience, neuroscience, neurotechnology, and computer science. Without further ado, James, the floor is yours.
Okay, thank you, Mohammed. And thank you to the organizers, as well as to all, for inviting me, as well as to all attendees for your attention. So let me share my screen. So the title of my talk is Constructing Virtual Cohorts and Coherent Data Distributions with Generative Adversarial Networks. And I'd like to make the point from the beginning that we believe generative adversarial networks provide a particularly useful means to construct digital twins, virtual cohorts of patients, and do them in a way that is explainable, as I hope my presentation will illuminate for you. So first of all, the motivation for our approach to virtual patient cohort creation is this idea that we've identified, and you all are very familiar with, that divergence in therapeutic design. That is, while we look at phenotypes, for example, in the example I'll show you in a minute, in electrophysiological phenotypes of neurons, there's a great deal of variability across those neurons, across the tissues they comprise, and across the patients who are suffering, for example, from Huntington's disease due to neurodegeneration in the neural tissue known as striatum. But that variability, in this case, observed in the phenotype of pyramidal cells in a paper by Scala et al., is more compounded by variability in the expression even of the genes that are responsible for encoding these different phenotypes shown on the right. So these gene variabilities map onto the phenotype variabilities in a way that's non-trivial. And we believe the deep problem in missing the mark, so to speak, in drug design is often that this nonlinear mapping has not been adequately addressed through machine learning. So in the case of Huntington's disease, we looked at the Pfizer Amaryllis trial, which failed to provide any meaningful endpoints in relieving motor symptoms in Huntington's disease. And we went back to the preclinical data that was published in Neuron from Huntington's mouse model neurons showing that the Huntington's phenotype occupied a more excitable region of the MSN, medium spiny neuron phenotype, shown by this red convex hull, when compared to the wild type phenotype shown by this green convex hull. And the application of PDE10, phosphodiesterase 10 inhibitor, was the basis for moving this drug into trial, because it did show a movement from the Huntington's phenotype towards the wild type phenotype. But it was not a direct hit. And so what I'd like to share with you is the results from a paper that we published, which got us into this area of virtual population modeling. But in this case, we were looking at constructing virtual drugs. This was published in iScience in 2021. And as you can see, our methods for constructing virtual drugs involve taking the phenotypes from the Huntington's and the wild type neuronal populations and applying a virtual drug in parameter space represented by a vector, in this case in one dimension, meaning a single target vector of a single ion channel. And while this moved the population towards the wild type population, it did so in an inadequate way. We tried things such as solving for a system of linear equations or finding the normal to a decision boundary between the phenotypes. Ultimately, what worked the best was to take an ad hoc method with difference of means between the parameter distributions. And this taught us that by finding this way to have a direct hit constructing virtual drugs in the parameter space of ion channel densities in the membrane, that this method needed to be explored further. In particular, we took the approach of doing parameter inference for mechanistic models based on an understanding of machine learning. In particular, the model parameters shown here as X, when fed through a biophysical model, lead to a clinical observation or some modeled clinical observation Y. And it's the role of our machine learning at this point to not simply find one instance of X that fits an observation, as we were doing to create construct populations of models in the previous paper, but instead to find a coherent distribution of all possible parameter sets that are reproducing the clinical observations or the preclinical observations in the case of some of our work, given the data. And so this can be thought of as a parameter inference problem, where you apply, for example, a Bayesian perspective, where the inverse model is sampling from this conditional probability given the forward model, which represents this conditional probability. And in our case, though, we have deterministic models, which are represented by the biophysical model here. Therefore, they do not have stochasticity, and the Bayesian approach becomes confounded by the Borel paradox. In addition, these are non-invertible models, at least not trivially invertible, and therefore the problem becomes one of sampling stochastically from a distribution of model parameters given a single observation and then doing it across the entire population. This Bayesian approach is further confounded by the problem that even in a case of a simple model, such as a constant plus the exponential of X, the uniform prior PX based on the ranges of some model parameters that we might observe will greatly oversample in a way that's non-physically biased from a region of the output space that happens to be overrepresented. And so we took this as very serious sort of challenge in terms of both the non-invertibility, the deterministic nature of our mechanistic models, as well as this confounding non-physical bias and parameter inference, and approach this as a problem as a stochastic inverse problem. In particular, we frame the stochastic inverse problem as constrained optimization, shown here, wherein the prior distribution of parameters, the experimental observations, and the model are subject to two constraints. One, to minimize the divergence between the generated parameters and the prior parameters, as well as to minimize the divergence or maintain zero divergence between the generated model output distribution and the actual experimental observations or features thereof. And so this parameter inference problem formulated in this way is novel to our team. And I'd like to share with you how we do it using generative adversarial networks. Some of you may know generative adversarial networks as a minimax problem in machine learning, wherein a generator and a discriminator are subject to a base variable distribution in the case of the generator. The generator produces outputs that the discriminator then compares to some target. And it's the role of the generator to learn to fool the discriminator into thinking its outputs are actual samples from the target data, whereas it's the role of the discriminator to always get it right and determine which is target and which is so-called fake. This has been applied trivially, well, not trivially, but I should say in a way that's not related to drug development in terms of things like face generation and various forms of artistic generation. In our case, we're using the generator to generate model parameters. Model parameters meaning parameters of the mechanistic model, which when run through the model, generate model outputs that the discriminator is then challenged to discriminate between that and the target data. And so this, together with the discriminator, which is discriminating between the model parameters that are generated and the prior on the parameters, gives us this dual discriminator GAN, which formulates this parameter inference problem as a constrained optimization. So the first example I'd like to show you, which applies this so-called RGAN, is that of modeling virtual pharmacokinetic populations, specifically with the human cytochrome P450 allele conditioner. And so in order to do that, we have to modify the RGAN slightly and add an auxiliary variable, which both the generator and the discriminator has access to. This auxiliary variable, in our case, for the pharmacokinetic problem, is one that is used to condition the parameters of the model, that is the parameters of the pharmacokinetic model that fit the data, given different, as you'll see, different CYP alleles. This is a very powerful modification to the RGAN in that it allows us to have any arbitrary data from patient populations act as a conditioning variable to the constrained optimization I described to you. So in a very simple way at IBM, we took a very simple PK model and used it to generate both synthetic data as well as to fit data from a very recent and quite interesting study of the CYP allele as a conditioner to pharmacokinetic model parameters. So I'm sure many of you are familiar with this. I'm not going to go into it in a great deal. This is a two-compartment model, and we are inferring parameters such as the kinetic parameters between the compartments, as well as the using the features from the decay of the simulated drug input to provide the synthetic inputs to our parameter inference GAN. And so just to show you how this works in a synthetic data set, I just wanted to point out that in our case, the density of the parameters that are targeted are selected by us, run through the model, and then we use the GAN in a naive way to infer those parameters. And so you can see that, first of all, the features of the outputs alpha and beta from the pharmacokinetic model are fit very well by the GAN, indicated by the red samples overlapping the black target data. This is a non-uniform distribution in both dimensions, and I think interestingly, the GAN's able to sample from the joint distribution with a high deal of accuracy. Because of the constraint, the constrained optimization, the prior was uniform in our case. Therefore, we were giving it a naive sort of view into what's possible, and that uniform distribution allowed the GAN to find other solutions in terms of the ground truth parameters that were used here in black. This is interesting when you then take away that constraint, how you can see that the GAN, while it gets the correct distribution of output features, does a very poor job of even overlapping with the ground truth parameter distribution. This is a word of caution in terms of in non-invertible models. You might get the right output, but there's no guarantee without a formal means to approximate the total coherent distribution that you actually have parameters that correspond to the parameters that generated the data. And so what we did next was looked at a data set from Frank 2009, again, dealing with the cytochrome CYP 2D6 alleles. And the first thing to observe from these data is that there was substantial individual variability within the AS group. So the three features that were extracted from these data, max concentration, time to max concentration, and time constant for the exponential fit to decay, what a subsequent paper this year showed from Rudesheim et al. Is that log normal sort of constraint on the parameter distribution could provide a good means to construct a virtual population. We took this as a challenge in that we have no such constraint on the GAN, and we went ahead and used what I've referred to previously as our conditioned RGAN, CRGAN, where the auxiliary variables are the CYP allele dependent activity scores, which is sort of a coarse view into the actual genomics of the patient, but those activity scores were what Rudesheim conditioned on, and so therefore we did as well. In addition, the data was quite sparse, and so we used a Gaussian mixture model to fit the patient data, both in terms of the CYP scores as well as the ground truth PK features, which were extracted from the curves I showed you on the previous plot. So what you can see is that our GAN was able to make accurate predictions of the dextromethorphan concentrations given the conditioning inputs from the activity scores shown here. This allowed us to generate populations of models, but in addition, allowed us to take a virtual population of pharmacokinetic subjects and interpolate between activity scores in the space of both parameters shown here and in terms of the features that are the extracted features of the PK traces. This just shows one of the advantages of these conditioning variables, that they can take a discrete conditioner such as genomics and turn it into a continuous discriminator. But in addition, I should point out that there's nothing that prevents us from embedding the actual genomic sequence of a patient and using that as the actual input to the CGAN for this type of a parameter inference problem. This has great potential. Next example I want to show you is that of modeling cardiac inotropes. In particular, a paper published last year from my team, led by Slava Gurev, made use of another modification to the RGAN, which is a little more complicated, but I'll walk you through it. This was a study that was targeted at interpreting the drug mechanism of action using population of models over the inotrope omecamtiv mecarbil. And so in this case, we have actually 4 discriminators. The 4 discriminators are, again, discriminating 2 sets of priors, and in this case, 2 sets of outputs of the model compared to real-world data. And what's important is that the reason we have these splits between the 2 discriminators types is that one is looking at a set of parameters which represent the control condition, and another set of parameters that represents the drugged condition. Often we have data in pharmacometrics that involves populations that are non-overlapping. In other words, one population was the control and the other was the drugged. And in this case, we use that in the GAN we separate the GAN into 2 discriminators in order to tackle this problem. But interestingly, there's also a generator which is generating parameters which are shared by both the control and the drugged condition. These are the so-called non-druggable parameters of the model. And this is prior information that affects the structure of the GAN. In this case, for omecamtiv mecarbil, we know that that drug was not affecting calcium trafficking in terms of contractility effects on the myocyte. And so we made those the non-druggable parameters and allowed only those involved in cross-bridge cycling, which are going to be affected by the inotrope, to be either in the control or the drug generator's outputs. The model itself was quite complex. It allowed us to take a stochastic approach to cross-bridge cycling, as we thought this was the right way to solve for the drug mechanism of action. And it actually paid off. For omecamtiv mecarbil, we were able to replicate data from the GAN using parameters from the GAN and the model I showed you that fit both the omecamtiv condition and the control condition for forced calcium curves. This was previously unexplained, and it lent credibility to the mechanisms that we were describing, in that it would be very expensive to do this with skin preparations. In this case, the model was based on data that was collected from myocytes in a dish, which were subjected to the drug and measured for their force time curves. So in the end, this TGAN, the so-called TGAN, allows us to look at 2 populations of parameters which represent the effects of the drug on the population that are measured, as well as 2 populations of model outputs which represent the drugged and the undrugged cohorts, which were separate in this case. And so the results are shown here. The experimental data, as I mentioned, are sarcomere length versus time for both the control and the omecamtiv condition. And our model simulations, you can see, not only fits the form of the result, but also very nicely the distribution, better seen here in terms of these features of the output of the contraction trace in multiple dimensions. And in fact, in the joint distribution, there was a great deal of correspondence between the actual data represented by the contour plot and the samples from the GAN represented by the points. But more importantly, the dotted line here represents those undruggable parameters that yet were still sampled arbitrarily, but in a way that was yoked together between the 2 generators in the previous slide. This shows that we were able to adhere to the constraint and the prior that the calcium trafficking parameters were not changing by the drug. And this then focused the GAN in on changing only those parameters which the model could use in order to approximate the effects of omecamtiv mecarbil on cross-bridge cycling in these experiments. The next example I'd like to talk to you about is a CD8 lymphocyte control study of SIV infection during antiretroviral therapy. And one of our speakers earlier is actually the person who pointed us to this study as a challenge, Yufeng Cao, who published this work, I believe, prior to joining the industry. This is a study which had 3 interventions for monkeys who were subjected to infection with SIV. They began antiretroviral therapy, and then they were given an anti-CD8, which created this sort of paradoxical rebound in viremia, which had been explained and debated in the literature. But what Yufeng and team proposed was a fairly sophisticated set of ODEs, which were quite complex and difficult to invert, which had an anti-CD8 affecting an exhausted and pool of CD8 effector cells, which were involved in cytolytic killing, such that when the anti-CD8 was applied, the population had already been returned from the exhausted state due to the antiretroviral therapy. The biology isn't as important as the troubles that we had with this problem. And in turn, we had to improve the GAN regularization to deal with the problem that target samples can fall in a very small volume of the prior, and this can cause us to get lost in the high dimensional space of such a complex model. What we found as solutions were either using normalizing flows to replace the GAN generator. What I'd like to talk to you about is projections into the low dimensional space for the discriminator, or to improve the prior with other methods such as ABC, which have their own computational demands that we steered away from. So in this case, we're projecting the prior into low dimensional space and allowing a sort of cascade of discriminators to classify the projected samples based on the prior distributions. And in this way, we created a custom layer to perform the calculation of this discriminator efficiently. And in fact, we solved the problem. So in this case, you can see roughly a dozen or more monkeys and their response in this population. We took a single monkey and were able to generate a population of models that is a virtual cohort from a single patient, in a sense, wherein the virtual cohort is constructed from multiple samples from our generator trend in the way I mentioned. And as you can see, we can not only look at features in Y, but also parameters in X and see how the distribution of those parameters play out, as well as look at the distribution in terms of a t-distributed stochastic neighbor embedding of X, which shows that there's a quite complex structure in this X parameter sampling. So this was exciting to us. We take it as an example of how we can invert very complex models, and we look forward to doing this in other QSP models. The conclusions that I have for you stem from the previous 2 examples that I showed you, 3 examples. First being the example from pharmacokinetic population modeling with the CYP allele conditioners. The second being the omecamtiv mecarbil mechanistic model inversion, which allowed us to infer a drug mechanism of action, given the observations of 2 populations, one drugged and one undrugged. And then lastly, in terms of the anti-CD8 therapy following ART therapy into a population of monkeys. And in that case, the problem that we addressed had to do with the complexity of the model and the high dimensional parameter space, which forced us to modify the regularization of our GAN. So in closing, I'd just like to say we've addressed the problem of non-invertibility of mechanistic models and solved them as a stochastic inverse problem using generative adversarial networks, which also affords us the ability to I think in a way explain the model in terms of the mechanistic model parameters, as well as explain the AI and the observations on the data in cases where the model is complex and the sampling over the full distribution of parameters that explains the data might be challenging. We had populations of models for both disease and drug mechanism understanding, and we feel that applying these virtual populations to rational drug cohort and design and assessment is possible now in areas such as clinical translation, drug triaging, dose response, therapeutic window identification, and prognostic biomarkers. And I'd like to leave you with this little diagram. Given the inference engines that we can create, we think that from response phenotypes, we can now create virtual drugs which can be used, for example, to take you from a healthy control to a disease control population and back again. We also believe that the drugs themselves can be tested, real world drugs, in order to create inverse drugs that take you from a diseased drug condition back to the, from a healthy drug condition back to a disease drug condition, or from a disease control condition to a disease drug condition in a way that's both rational and based on population modeling. So I'd like to thank you and open it up to questions.
Thank you so much, James, for this very interesting talk and sharing with us the capabilities of GANs, really to do this fantastic work. So we have time for one question. So perhaps I can ask, out of curiosity, what metrics might one look at to demonstrate that GAN-based approach, the one that you showed us, for virtual population generation performs better than other existing methodologies?
This is a great question. Thank you, Mohammed. So we use Jensen-Shannon divergence for much of our studies in order to understand how in our synthetic data sort of examples, because we have view into the ground truth of the parameters, how we're doing. And so JS divergence has provided us a very good means to understand in the sort of complex measure of parameter distribution coherence, how we're doing. And we find that the performance is equal to that of computationally intensive and somewhat constrained methods that we're all familiar with, like MCMC. So the method is performing at the same level, but it's solving problems that go well beyond MCMC's capabilities.
That's great. Thank you so much, James, one more time for this excellent presentation. And now I'm going to be moving to my colleague Sujatha to present and to introduce our next speaker.
Thank you, Mohammed. I'd like to introduce our next speaker, Dr. Nadia Terranova. She was introduced this morning, but our audience may be different now, so I'm going to reintroduce her. She serves as the head of Quantitative Pharmacology and Advanced Data Analytics at Merck Serono. Dr. Terranova is a biomedical engineer by training, having received her PhD in bioengineering and bioinformatics. She joined Merck Serono in 2013. Her contributions to the scientific community and to Merck's R&D range from impactful systems pharmacology modeling, pharmacometrics, and applications to advanced machine learning techniques applied to complex and multivariate drug development problems. And this is across spanning several disease areas: oncology, immuno-oncology, and multiple sclerosis, etc. Dr. Terranova is a leading author of several papers in selected journals and in international conference proceedings. She's a Page Lou Shiner awardee and a member of the CPT and JPKPD editorial boards, as well as she serves on the ASCPT, Pharmacometrics, and Pharmacokinetic Steering Committee. So today she'll be talking to us about predicting disease activity in multiple sclerosis patients using explainable machine learning models. Take it away, Dr. Terranova.
Thank you for the introduction. It's my pleasure being here today to share our work on explainable machine learning modeling for prediction of disease activity in patients with multiple sclerosis. Here the outline of my presentation. I will start by giving some background on the analysis objectives and clinical data used for the analysis. I will then go through the machine learning workflow and present results from model prediction of disease activity in MS patients. I will conclude by making some remarks. So this work was recently published in CPT PSP journal and is the results of a great teamwork and cross-functional collaboration between our Merck quantitative pharmacology team and global medical affairs colleagues. So the team brought to us the clear patient-focused ambition of enhancing understanding of underlying mechanism of disease activity in MS and the factors driving it through its early prediction. This has indeed a great potential in enabling better patient monitoring, treatment planning, and potentially patient selection. So to this purpose, we integrated demographics, response data, MRI, and neurological assessments collected in cadmium trials to predict whether a patient will have disease activity in 3 and 6 months. Or if you want, try to identify which covariates collected 3 or 6 months earlier can inform whether a patient will experience disease activity. We use data coming from cladribine trials. Cladribine is an oral disease-modifying drug for use in MS treatment. The recommended dose is 3.5 mg per kg, consisting of 2 annual courses, each comprising 2 treatment weeks, 1 month apart, starting at the beginning of 2 consecutive years, for a total of 4 treatment weeks. We combine data from 3 phase 3 pivotal MS trials, Clarity, Clarity Extension, and Oracle. The pooled dataset resulted in a richly informative population of more than 1,900 patients receiving placebo, cladribine 3.5 mg per kg recommended dose, or the 5.25 mg per kg higher dose, which was also explored in these trials. And we had a very long observation period of more than 6 years. So with heterogeneous baseline and time-varying covariates that can enable the exploration of the relationship of multiple efficacy endpoints with patient baseline and time-varying characteristics, as well as treatment effects. Our endpoint was a disease activity event, which was defined as a composite of well-known clinical endpoints measuring various dimensions of disease activity: qualifying relapses, T1 gadolinium-enhancing lesions, T2 lesions, EDSS, progression over 3 months, as well as the switch to other disease-modifying treatment, which in fact, this switch, it's a marker of treatment persistence and can be driven by the lack of efficacy or tolerability issues. Indeed, disease progression in MS is quite complex and has multidimensional aspects. So there is strong interest in studying composite endpoints that combine several clinical endpoints to then address better treatment planning and MS patient monitoring. And to note, our analysis involved all patients in the cladribine trial, so including those in the placebo arm not receiving cladribine. Here in the plot, you can see Kaplan-Meier curves of the time to the first disease activity event for the 3 arms. Higher prevalence of disease activity among patients is observed in the placebo arm compared to the 2 cladribine treatment arms. And these results are completely in accordance with observation in the study, demonstrating the efficacy of cladribine versus placebo in reducing clinical relapses, disability progression, MRI-assessed disease activity. Out of our combined population of more than 1,900 patients over 6-year observation, we could see that approximately 25% of the patients met one of the criteria, at least one of the criteria for disease activity. And on top of this, that 80% of observed events fall within the first 2 years out of the 6 years observations. Let's move now to the machine learning analysis framework. We use a supervised multivariate machine learning method to train and validate models to predict which patient will have disease activity 3 months and 6 months in advance of their clinical determination. This time frame was selected based on the frequency of the patient visit according to the design of the clinical trials, as well as the current clinical practice. So we had a large set of covariates, including patient characteristics, neurological and MRI assessment, laboratory measures, and our disease activity endpoint, which enter into the machine learning model during the training phase. Then we use covariates from unseen test data and input this into the trained machine learning model during the test to assess the prediction of disease activity. We trained and validated 4 models, the 3 months or T minus 12 weeks model and the 6 months or T minus 24 weeks model. And then we had phase 3 and phase 4 covariates to build the corresponding models. So in particular, in the phase 3 T minus 12 and T minus 24 weeks models, we use 57 independent covariates, including various baseline and time-varying covariates related to patient characteristics, neurological and MRI-based assessment, laboratory measures. But on top of this, we also wanted to establish a workflow that could leverage a number of phase 4 trials which are ongoing with data that are being generated. And for this, we constructed the so-called phase 4 T-12 and T-24 weeks model, which were based on a set of 25 independent covariates after excluding the laboratory measures. This was to mimic really the scenario of routine clinical practice or phase 4 trials where laboratory covariates are not usually collected. Let's move now to see our analysis framework. So first, the available patient data is split into 80-20% ratio using a stratified random sampling strategy for model training and testing. We employed the gradient boosting tree-based ensemble machine learning algorithm, XGBoost, which has been demonstrated to be highly effective in a variety of problems. During the training process, we used 10 times repeated tenfold cross-validation to optimize the model hyperparameters, which generalize best the model performance on unseen data. Then the trained model was applied to a completely left-out test data to get the unbiased estimate of the model performance on future unseen data. So this training and evaluation protocol is quite standard in data science and results in a good balance of preventing overfitting while ensuring generalizability of the model performance on future unseen data. It's also the so-called bias variance trade-off in machine learning. We know that there are some difficulties sometimes in interpreting complex machine learning models, and that the prediction of this model might be limited, as well as the confidence and trust into the outcome. In particular, in the clinical setting, it's crucial to understand not only the global aspects of the covariate dynamics or at the population level, but also to understand specifically for each individual patient how some covariates are impacting a prediction. For this purpose, we use SHAP, Shapley additive explanation, to explain the model prediction for each patient by computing the contribution of each covariate to the individual prediction. And I will cover this within the next slides. But first, let's have a look to the performance of our models. So both the phase 3 and phase 4 models and the 3 months, 6 months models perform equally well, with a consistent selection of the covariates, or like top ranking of predictive factors. We can see here in the table that we had the balance accuracy of 80%, and also the AUROC was quite high. The training and the validation performance are close, implying no model overfitting. But let's look now at our explanation model with SHAP. So the goal of SHAP is to explain the model prediction for each patient by computing the contribution of each covariate to the prediction using cooperative game theory. So in the example here, you can see the contribution of each covariate to the individual prediction of a mortality risk score. So by starting from a base rate, there are some factors that are increasing the mortality risk scores, while others decreasing it. So this provides us with a local explanation of prediction at the individual level. But a global ranking of important covariates can also be obtained by ordering covariates by their mean absolute SHAP values for all patients, thus giving the results at the population level. But let's have a look at our use case. So you see that the local explanation are based on assigning a numeric measure of credit to each input feature. So if you want, each feature value is a force that either increase or decrease the prediction. In our case, the risk of disease activity. And the prediction starts from a baseline, where the baseline is the average of SHAP values across all predictions. So in these following two figures, we see SHAP explanation for plots for two individuals where the risk of disease activity was correctly predicted by the model. So patient A has high predicted risk of disease activity. And when looking at the covariates, we can see that while treatment weeks equal to 8 was decreasing such risk, we had a high number of combined unique lesions and new T1 black holes that are increasing the prediction towards disease activity. Differently, for patient B, we have a low predicted risk. And when looking at the covariates, we can see that mainly the number of treatment weeks and almost absence of combined unique lesions are driving the prediction towards no disease activity. So these are really explanation for individual prediction. But Shapley value, as I was mentioning, can be combined into global explanation, which represent then in a global structure the ranking of the feature while retaining the local faithfulness to the original model. And let's start here with sharp feature importance. This represents a global view at the population level of the important covariates. So here the top 20 most predicted covariates and their relationship to the predicted output by the model. And these are represented for the phase 3 T minus 24 weeks model. So the features are sorted by their mean absolute sharp value. And you can see that in the top predicted covariates, we have the number of treatment weeks, new combined unique active lesions, new T1 lesion counts, as well as other measures like age-related MS severity score. So this represents and provides us with a global view at the population level of the important covariates, where covariate importance is really understood as the contribution of the covariate in the model to discriminate between the patient who in the future will have disease activity for those who will not. But another way to visualize the feature importance with SHAP is the summary plot. This combines feature importance with feature effects on the prediction. It's not very different, if you want, for pharmacometry forest plots, which are typically generated during the covariate assessment. So each point on the summary plot is a Shapley value for a feature and an instance. On the y-axis, we have the features ordered according to their importance, and on the x-axis, their Shapley value, with the color code being blue for low values of the feature to red for high values. I-sharp drive the model prediction towards disease activity, while low values of sharp towards no disease activity. So let's take as an example our first feature, which is the number of treatment weeks. You can see that a low number of treatment weeks, so blue, clearly drives the model prediction towards no disease activity, while high number of treatment weeks drive it towards no disease activity. So here we start having a first indication of the relationship between the value of the feature and the impact on the model prediction. But in fact, we can see the exact form of the relationship by looking at the sharp dependence plot. So the sharp dependence plot here, it's shown for the number of treatment weeks, where you can see that the model-derived predicted probability of disease activity of a patient is decreasing with increasing number of treatment weeks. And in particular, it's a minimum number of 4 treatment weeks, which decrease really the risk towards no disease activity. And while this can seem expected or obvious, because we say, okay, the drug is having an effect, and so it's driving the prediction towards no disease activity. In fact, this result is really outstanding because it perfectly reassembles the exposure-response relationship, which we derive with the repeated time to event model for relapse rate. Here you can see the exposure-response relationship with overlaid the ranges of the exposure corresponding to the 2 cladribine doses. You can see that the 3.5 milligram per kg recommended dose, it's really at the shoulder of the exposure-response relationship. Increasing the dose and then the exposure will not substantially increase the effect, while decreasing it would result in a drop in efficacy. And here, with machine learning, no mechanistic modeling, no assumption on the relationship between treatment and our endpoint, no inclusion of prior knowledge on the mode of action, a different and expanded endpoint, which is a composite endpoint going beyond qualifying relapses, we actually came to the same finding. Well, this is quite impressive and supportive of the application of AI machine learning to address model-informed drug development and clinically focused questions to integrate multimodal and heterogeneous data as well as multiple endpoints. Of course, this kind of dependence plot can be derived for any covariate in the model, here for the top 6 predicted covariates as an example. So to conclude, we have defined disease activity as a composite of clinical endpoints, and we could see in cladribine trials higher prevalence of disease activity in the placebo arm. The 3-month sustained EDSS progression was the most informative criterion in detecting patients with disease activity. All the development models performed quite well. The phase 3, phase 4, as well as the 3 months and 6 months models provided 80% balance accuracy. And we could see a strong overlap of the top predicted covariates. Among these, we had cladribine treatment durations, new combined unique active lesions, new T1 lesions, as well as other well-understood prognostic factors of MS disease activity. By using machine learning, we could exploit a 9-dimensional data set looking at integrating multimodal and heterogeneous data. Thanks to the application of SHAP, we could have interpretable predictions from our model. At the same time, results could highlight the importance of quality MRI evidence in the management of MS disease progression and patient monitoring, in line with MS guidelines. Indeed, this kind of detailed information regarding counts of various new lesions are difficult to collect in routine clinical practice and usually available in research or during drug development, but they are really important predictive covariates for MS disease activity in patients. On top of this, right now, we are further validating our model by using data, integrating data generated from few phase 4 trials. So we are further assessing also the contribution of other neurological measures for the prediction of disease activity. Of note, the current model treats the task of predicting future probability of disease activity as a binary classification. But we are currently investigating dynamic prediction models like recurrent neural networks, which take as an input the longitudinal covariates and then can predict an updated probability of the risk of disease activity. So with respect to the focus of this session today, this work clearly shows how interpretability methods like SHAP make the decision process of machine learning models more transparent, increasing trust in the models, while ensemble methods like XGBoost can enable improved predictions by capturing nonlinear effects and covariates interaction in a dataset with a high number of diverse covariates. Now, with these acknowledgements, I'm happy to take any question, and I thank you for your attention.
Thank you. Thank you, Nadia, very much for that clear and very informative presentation. So on behalf of the audience, I'm just going to ask a couple of questions. I think we have time for a couple. So first, can you comment on the imaging data? How was that data coded and how was it included in the model?
Yes. So we had so the MRI-based data that we use in the as input features and also to determine the composite endpoint. So these were collected in the clinical trials according to the study design. We had the number of volumes of T2 lesions, T1 gadolinium enhancing lesions, black holes, combined unique, but this was all available as part of the design of the clinical trial, so readily available in our clinical database. So if the question is about whether we went back to the MRI scans and tried to extract with machine learning any kind of other feature, so this was not the case for this work, but definitely it's a great area of interest also for our group. In fact, we do have collaborations on that in the oncology field. Where together with the university hospital, we are segmenting tumor lesions and extracting radiomics to assess the relationship with tumor sites parameters in the model. And in fact, we presented this at PAGE European conference last June. So it was not done for MS, but definitely it's an area that we are exploring as well.
Okay, thank you, Nadia. So another question is, the top predictive covariate that came out of the SHAP seemed like was the treatment duration, right? So was dosing information evaluated as a feature? And if not, do you think missing that information may have had a confounding effect on the treatment duration coming out as the most predictive? So in other words, can you comment on the potential interplay between dosing information and treatment duration? as a feature.
So indeed, for cladribine, it's really a special case. So somehow the number of treatment weeks, it's representative of the dose because of cladribine schedule. Because 3.5 milligram per kg, for example, as a dose, it consists of 4 treatment weeks. And in fact, we chose the representation of cladribine dose to the model as the number of treatment weeks received instead of the cumulative dose like the 3.5 milligram per kg, because these add a couple of confounders like body weight. So definitely in this specific case with cladribine treatment, the duration, it's really embedding the knowledge about the dose that the patient receives.
Okay. Yeah, thank you. And as far as predictability of the model, can you comment on the application of this model to other multiple sclerosis data sets that use different treatments? I don't know if you did that here. I think I only saw the MAVENCLAD, but maybe I missed that. Also, you mentioned applying the model to phase 4 clinical trial data. Can you share this model's predictive performance on that data?
Yes. So in this work, we did not apply the workflow of MS disease activity to compounds other than MAVENCLAD, but indeed it's a great question and point, because to generalize eventually the model and give this to the physicians in a way that they can predict and use this as a tool for disease activity in MS, definitely the model should be trained and then tested on a variety of treatments. And this will be great. We know that, we are aware, and we are finding the opportunities, right? And it's great because somehow it's by having various treatments on top of placebo, which is already included, we can also better understand which factors are really related to the drug, to the treatment, and which ones are related to the disease. So prognostic predictive, right? So we can really isolate the nature of the features that are going to be predictive. And so with respect to your question about phase 4, so I would be glad to share results, but in fact the analysis is ongoing. So we are really excited to test the model really on external validation, because these are completely unseen data and phase 4 data. And on top of this external validation, we have also the opportunity to include the number of biomarkers that have been collected there, most recent biomarkers, as well as patient-reported outcomes. So as a team, we are really excited, and we are looking forward to sharing this as soon as possible with the community, but I cannot share more today.
Yeah, of course, I understand. So yeah, thank you so much, and we are looking forward to those results too in the public domain when they do come out. But thank you for your time today, and with that, I'm going to hand it over to Mohammed to introduce our next speaker.
Thank you very much, Sujatha, and thank you, Nadia, for this wonderful talk and excellent discussion. Our next speaker is Dr. James Liu. He gave a very nice presentation this morning, and we are happy to have him in this session as well to talk about explainable machine learning. In brief, Dr. Liu is a principal AI scientist in modeling and simulation and clinical pharmacology at Genentech, where he currently leads a team of AI machine learning scientists in applying and developing advanced algorithms to clinical data. I'm going, for the sake of time, I'm going to turn things now to James to present his talk, which is titled Explainable Deep Learning for Tumor Dynamic Modeling and Overall Survival Prediction. James?
Thank you, Mohammed, for the introduction. It's my pleasure today to present to you in the session of the IQ workshop some of the recent work that we've been doing in explainable deep learning for tumor dynamic predictions. Next slide, please. So first of all, why are we interested in AI algorithms? A key reason being that we are facing a data challenge in this digital age. And this is especially profound in the setting of oncology. As a result of the current trends in technology and digitization, firstly, we are seeing a huge increase in the number of patients available, including from real-world data sources. Secondly, through non-invasive measurement methods such as liquid biopsy, we are seeing an increase in both the potential number of longitudinal measurements as well as their dimensionality. Finally, we see that data is becoming multimodal, and this can span from clinical to radiological and genomic and so on. So the question comes, how do we cope with the challenge of modeling a deluge of data. Next slide, please. One way to deal with this challenge is to evolve the methods that we use to build predictive models. The modeling of tumor dynamics for the prediction of patients' overall survival has become well established over the past decades or so. For instance, with a paper on the left from Claret et al. In 2018, shows an application of tumor modeling in around 1,000 patients, where a parametric survival model is used to link explanatory variables consisting of TGI matrix and other based on covariates, and linking them to OS via a parametric survival model. Since then, there have been a number of different machine learning efforts in survival predictions, and this includes a 2019 paper that utilized a stochastic differential equation model for tumor dynamics for more than 7,000 patients collected across many different clinical trials of non-small cell lung cancer patients. And the machine learning models that were used include Cox, random survival forest, and multi-layer perceptron. And subsequently, a number of different machine learning approaches have also been demonstrated. Next slide, please. Explaining how nonlinear machine learning models use explanatory variables to predict survival is an important task when the models are used to make decisions. As we have seen in Nadia's presentation previously, the use of sharp analysis is an attractive methodology that has desirable attributes. In this paper, we've shown how sharp values can be used to explain how variables influence the hazard rate. In particular, suppose we have a machine learning model that takes in the explanatory variables for patient I, namely the vector x sub I, and computes f of xi. Then the hazard rate h of t can be expressed as an exponent of f. And what the sharp analysis does, as Nadia has illustrated, is to decompose this nonlinear function f as an additive contribution of terms phi sub j, sum over the set of input variables j. And this expression below shows that the exponent of the sharp values quantify in a precise manner the contribution of each of the variables towards the hazard. And this allows us to draw conclusions that include, for instance, how much is treatment impacting the patient's hazard rate. We show that by appropriate sampling with replacement, we can also compute the confidence intervals. In fact, in this particular example, the results are in good qualitative agreement with that obtained from the Cox proportional hazards model. Next slide, please. So we believe that given the recent technological and computational advancements, we are headed towards the next generation of oncology disease modeling. The current paradigm on the left of TGI-OS is very well established, while the causal relationship between dose, exposure, and tumor growth inhibition is expected to be drug-specific. Much work has shown that there is a link between TGI matrix and overall survival, which is only disease-dependent. That is, once such a quantitative relationship has been established with an existing drug or set of drugs, a statistical model can be used to predict the OS from TGI for a new drug of interest. And I argue that we can augment this approach with machine intelligence. First of all, rather than having to pre-select the right tumor model, we can use the existing data to generate data-driven interpretable new ODE models. Secondly, rather than testing the right statistical model to use, one can use nonlinear machine learning models to make a link between tumor dynamic matrix and OS. And such an approach is also more amenable to the use of high content data, such as omics. And what we have shown here is a rather surprising finding, that we can link TGI to OS in a disease agnostic manner. So you may ask, what are the potential benefits of using machine intelligence? Firstly, this can lead to improved predictivity, both at the patient level as as well as at the trial level. While using the traditional TGI-OS approach can be labor intensive, in the machine learning approach, one could compile comprehensive data sets and perform cross-molecular learnings. And when we have high content data, there's also the potential to increase our disease understanding across different tumor types. Next slide, please. Here, we show some promising results of leveraging a single machine learning model to predict OS across many different cancer types. To train the model, we've compiled a data set consisting of more than 7,600 patients across the 5 cancer types you see at the left. And we've built a machine learning model that is indication independent. The result shows that the use of the data across tumor types enables better predictability. The algorithm that we use can handle missing data, hence patients who are so-called TGI non-evaluable can also be utilized. Our work suggests that it has the potential for better extrapolation to novel 10 cancer types. With sharp analysis, we have identified indeed that KG, or the tumor growth rate, is the most predictive driver in predicting survival. In particular, we also see that the machine learning algorithm has made the inference that patients who are TGI non-evaluable, because they only have baseline measurement but no subsequent tumor measurements, should also have higher hazard rate. And without making additional assumptions, the machine learning model has made that inference. Next slide, please. So to sum up this work, we have a single model that can predict hazard ratios across all of the 11 arms over 10 clinical trials that represent 5 different solid tumor types. And on the right, we show that the predicted hazard ratios and the 95% prediction intervals line up well with the observed hazard ratios in the test set. And this was a somewhat surprising result for us. As we know, the current way of doing TGI-OS is to model the 5 different tumor types individually. And this result shows that one single machine learning model can be used to make predictions across different tumor types. And this result raises the question of whether such a model could better generalize to new tumor types that have not been used in the training set. We argue that by leveraging a much larger number of patients across different tumor types, the machine learning model could in fact be more generalizable than a TGI-OS model that is built from a single tumor type. And while a machine learning model using traditional TGI matrix can improve OS predictions, we believe that there are several additional opportunities for further leveraging machine intelligence. Firstly, while TGI modeling works well when we have long follow-ups, our one current challenge is to enable precise predictions from earlier data cuts, and this is an area for further improvement. Secondly, and this is related to the first point, in making tumor size predictions from early data, there is oftentimes a bias, and how might machine learning help to improve that? And finally, while an increasing amount of multimodal data is being generated, it is not clear whether existing methodologies can fully leverage that information. And based on these opportunities, we've embarked on a deep learning approach to tumor dynamics modeling. Next slide, please. And the approach we've taken is to merge deep learning with dynamical systems at a fundamental level. On the one hand, we would like to leverage the ability of deep learning to excel in approximating high-dimensional nonlinear functions. In doing so, that improves as the data increases. On the other hand, we would like to build in the concepts of dynamical systems that we are already leveraging in tumor dynamic modeling. In addition, just as TGI matrix provide very useful abstraction, of longitudinal tumor data that can actually predict patient survival, we similarly want the neural network to come to a low-dimensional abstraction of the data P. The solution we propose is tumor dynamics neural ODE, which intertwines the two complementary concepts. And the result is an autonomous dynamical system that learns from tumor dynamics data and enables extraction of metrics that can predict patient survival. Next slide, please. So what are the key components of a TD-No model? First of all, the longitudinal tumor SLD data at the patient level is passed to an encoder, which has the task of performing data abstraction. And the encoder in turn delivers a low-dimensional vector denoted here as P for each patient. And this is what we call TD-No matrix. The vector P then goes into the decoder, which is an ODE system with the right-hand side represented by a neural network. And the aim of the decoder is to perform predictions using a single system of ODEs and taking in as input vector P. And in analogy with the existing TGI paradigm, we believe TD-No matrix can predict OS, and we'll be looking at that later. And there are several benefits to this TD-No architecture. Firstly, this neural net provides an explicit abstraction of each patient data in a low-dimensional latent representation, which has the desirable attribute of parsimony. Secondly, we are representing the observed tumor dynamics as a time-invariant dynamical system, which may also help in generalizations. Lastly, we try to interpret tumor dynamics metric P and can then use it also to make survival predictions. Next slide, please. One common issue with deep learning models is that they tend to be data hungry, and hence ways of augmenting limited data is of the key importance to improve accuracy and robustness. The plots illustrated here show how we go about doing that, using here a set of 3 different observation windows. Here we are looking at a set of 3 representative patients where various amounts of data have been fed into the encoder. In this first case, the panel on the left, observation data up to 16 weeks is used, and the model is asked to predict tumor size after that window. In the second case, data up to 24 weeks is used, and the model is asked to optimize prediction beyond that time frame. And as we can see from this data augmentation scheme, the model is definitely encouraged to do as well as it can to use early observation data in predicting future unseen data. And this aspect of optimizing predictions for future time is an aspect that we'll come back to. Next slide, please. In our work, we've split all the patients into 80% trend set and 20% test set. In the top left plot, we see the model captures the data well in both the trend and the test set. And in the table below, we assess the predicted performance of the model using RMSE and R-squared as performance metrics, both showing promising results. And as we previously mentioned, many of the existing tumor dynamic models are known to give rise to biased predictions when the observed data is truncated. As we've previously mentioned, the TD-No model is by design optimized to make future predictions. And we show that it provides minimal bias in the results shown here. We believe that this is a highly desirable attribute of this methodology. Next slide, please. There are certainly a number of different parallels in the methodological approaches between the pharmacometrics, TGI-OS modeling, and TD-No in how they use longitudinal data to make OS predictions. On the left, with the pharmacometrics approach, one chooses a tumor model of interest. Then the model parameters are estimated using the population approach. Subsequently, the key TGI matrix are chosen. Oftentimes, it's this parameter Kg. This is then used in a parametric survival model where different structures are explored. Then, based on the statistical model, we compute OS curves and the associated hazard ratios. So how do we go about doing this using TD node? We first feed the data into the neural network architecture, and the right ODE model is discovered from the data via supervised machine learning. Then the encoder output corresponds to the TD node matrix of interest. Subsequently, we feed this matrix into a machine learning model of survival, which by sampling with replacement, we can get prediction intervals for both the OS curves as well as the hazard ratios. So the benefit of TD node is that there's an almost one-to-one analogy with pharmacometrics, but just using a different set of machinery. Next slide, please. It turns out that TD node matrix can accurately predict patient survival at the individual level. In particular, while TD node matrix have only been trained on tumor dynamics SLD data, by providing a highly useful abstraction of data they can use to predict patient survival. Here we show how the model has been evaluated. Namely, 20% of the patients have been held out as a test set that none of the machine learning models have seen. And in each of the 5 folds of the training set, the neural network tries its best to predict tumor size data. And for each patient, we obtain the value of the TD node matrix. The TD node matrix then go into a tree-based model for OS, and then we then compare the equivalent approach, namely instead of using TD node, but use the existing set of TGI matrix, which consists of the 3 parameters, Kg, Ks, and TTG. In comparing these 2 models, we use C-index as the performance measure, which is a number that goes between 0 and 1, the higher the beta. And we see that the TD node OS machine learning model performs better both on the training set as well as on the test set. Next slide, please. Furthermore, we demonstrated that the TD node matrix can also be used to predict survival curves and hazard ratios. Here, we are only looking at 200 plus or so patients in the test set. On the TD node matrix and using XGBoost model, we show that in each of the 3 arms below, where we have around 70 patients, the machine learning predictions shown in dashed lines and blue shaded regions are in good agreement with the data shown in solid lines. Furthermore, the predicted hazard ratios of arm 1 versus arm 3 and arm 2 versus arm 3 are also in good agreement with the survival data in the test set. Next slide, please. As the theme of this session is explainable machine learning, so it's important that we are able to interpret the results. As we have previously discussed, sharp analysis provides a great way to understand how features drive ML predictions. In this left sharp summary plot, we see that of the 4-dimensional TD node matrix P, the OS prediction is primarily driven by only one component of it. And because this matrix P enters the ODE model explicitly, as we see on the top right, we can try to visualize how this key component changes the system dynamics. And to do so, we've picked 3 representative patients across the spectrum of sharp values. In each case, we vary this parameter component that has been previously identified. And we see, for instance, in the lower left panel, what happens when the variable increases, as shown in red, versus when it decreases, shown in these blue lines. And we see that in the leftmost panel of the 3, that varying parameter, this parameter corresponds to simultaneously changing the steady state as well as the slope. And this result certainly makes qualitative sense. Basically, the model is showing that the lower the tumor size becomes, the longer the patient should survive. Next slide, please. So far, we've seen how TD node can be used to model the longitudinal SLD data, but that's just the starting point. We believe that it's a foundation upon which that we can incorporate multimodal data. And this includes ctDNA and other data modalities that are high dimensional and difficult to work with in the traditional paradigm. From these multimodal data, we wish to extract basically a latent representation of the patient, from which we can also aim to predict patient survival. So this general approach opens up many possibilities for the future. Next slide, please. So I would like to end this presentation with some conclusions. Firstly, the rise of multimodal data increases the call for AI algorithms, and TD-Node provides an AI-based paradigm that in many ways mirrors the pharmacometrics TGI-OS paradigm. Lastly, this approach provides a principled foundation for combining multimodal data with the longitudinal data. Finally, I would like to acknowledge my colleagues at Genentech for their contributions, and I will be happy to take questions.
Thank you so much, James, for this very interesting presentation. So I think actually one of the things that I think we have some time, so we can ask a couple of questions. So one of the things that you mentioned in your presentation is that deep learning algorithms are data hungry, and as you also mentioned, one of the limitations of using deep learning is overfitting, especially with relatively small sample size data sets like we have in PKPD. So can you comment on the deep learning model you developed and how you think this can be mitigated? I noted that you talked about data augmentation, maybe one of the strategies to improve accuracy and reduce bias. However, one of the limitations of data augmentation as well is that it may arise from the data bias, right? That the augmented data distribution can be quite different from the original one. So can you comment on that?
Yeah, thanks, Mohammed, for the question. Certainly, we are faced with limited data in a lot of the time, and data augmentation have shown to be influential. However, as we know, bias is an important aspect that may come in the process. So I think there may be different approaches of data oversampling to kind of create a richer space of data where we currently don't have that many patients. And I also see the potential for approaches such as GANs to generate virtual synthetic data to enrich the model. And furthermore, often, for a particular drug treatment, we may not have a lot of data, but at least as many institutions have a lot of historical tumor size data, and how best to leverage that information so that almost in a Bayesian approach, if you will, that we have certain priors to start off from, maybe one approach to go. So I think there are a few methodologies that could be utilized. This field, of course, is rapidly evolving, and we'll see whether or not there are additional approaches that people can come up to deal with these limitations.
That's great. Thank you much, James, for sharing this very informative feedback. And I'm sure that this topic actually will come again in the discussion, and see if data sharing or synthetic data, as you mentioned, may be one of the ways to overcome this limitation. So another question is, for explainable machine learning methods, do we have the ability to interpret the statistical and clinical significance of the explanatory variable relative to the clinical outcome?
Yes, thanks for that question. It is a very important one. So in a lot of the work that we do, we try to not only give a point estimate of the prediction, we actually try to give the whole 95% prediction interval. Even though the meaning of parameters between machine learning models and statistical models are very different, machine learning models we often have these weights, so there's not a one-to-one in that sense. However, being able to kind of use approaches such as sampling with replacement can address many of these statistical issues of model uncertainty in model predictions. Secondly, the clinical significance. Yeah, I think we want to perhaps identify model mechanisms or parameters that are sufficiently large and kind of statistically significant before we draw too much conclusions from the results. So kind of understanding some of the uncertainties, and then there may be biological knowledge graphs that can be further employed to make sure this is linked to the existing knowledge. Maybe one way also to further address that.
All right, great. Thank you much, James, for sharing that. I think for the sake of time as well, and having more discussion in the panel with the panelists that we have, I think I'm gonna turn things over now to Sujatha. This concludes the 3 presentations we have. I would like to thank so much the presenters. And Sujatha, please go ahead and start the panel discussion.
Thank you, Mohammed. A very big thanks to all the 3 speakers. We just heard from all 3 of them very excellent talks about the applications of explainable ML. So now we'll just start the panel discussion on this topic. In addition to our 3 speakers that you just heard, we are also fortunate today to be joined in the panel by Dr. Yanning Wang, who's quite well known. Dr. Yanning Wang is the CEO of Createrna Science and Technology, and as many folks may know, he was until very recently the director of the Division of Pharmacometrics in the Office of Clinical Pharmacology at FDA. That was until September 2021. There he oversaw reviews, research projects, and policy development within the Division of Pharmacometrics for all disease areas. Before joining the FDA in 2003, Dr. Wang received his PhD in pharmaceutics and a master's degree in statistics from the University of Florida in 2003. He served as a board member of ISOP and is also a fellow of ISOP. He's a member of the advisory committee for the Chinese Pharmacometrics Society and a member of the editorial advisory board for the Journal of Pharmacokinetics and Pharmacodynamics. Dr. Wang is also an adjunct professor in the Department of Pharmaceutics at the University of Florida and the Clinical Research Institute at Beijing University. He has published over 110 papers and given over 320 presentations at various national and international meetings as an expert in new drug development and regulation. So thank you, Dr. Wang, for being here. Now going on to the panel discussion, let's start with a general question first. So today we've heard of examples of AI ML models to go beyond the traditional modeling strategy, as well as the development of explainable machine learning methods to better understand machine learning black box models. Given each of your scientific expertise, which is unique for each of you, what is your perspective on the importance of explainable AI in the MIDD setting? We'll start with Dr. Wang. From a regulatory perspective, what do you think is the role of explainable machine learning in informing decisions within the drug development paradigm? Is it likely to be beyond supportive evidence? Is it likely to be core as well? So we'll start with you, Dr. Wang.
First of all, thank you for having me at the panel. Like you said, I was the director of the Division of Metrics until last September. In fact, I'm very glad and excited to see all the excellent examples presented today. Before I left, I spent the last few years working on expanding the scope of Division of Pharmacometrics to include all the new tools, in the machine learning world to see whether, we can solve some of the most challenging issues, like the compounding issues, the causal inference, by using some of these new tools. And today I saw some excellent examples, especially the explainable AI models, because in the last 18 years, whenever we can explain some of this model-based analysis to our medical colleagues, to the decision makers at FDA, that will make the decision-making process much easier versus some models they don't understand. So therefore, I think if we can turn some of these black box models into white box models by using sharp or any explainable techniques to make it easier to understand, especially make it more intuitive, for the medical decision makers to see, yeah, this makes sense. Once you translate this hundreds of covariates to a few that makes medical sense, I think naturally they will be more open to use this kind of more complicated models, even though they may not still understand the full details of modeling. Therefore, I think making all this model explainable is a critical step. If we want to use this kind of more, I would say, complex models in any regulatory decision-making process, because we learned, even through the traditional, let's say, relative to the AI-based models, the traditional pharmacology-based modeling process, whenever you make it easier to understand and translate to something that makes medical sense, then the decision-making process, everything goes smoothly. Otherwise, you meet with resistance, doubt, or basically all the typical caveats on model-based analysis. You made this assumption, I don't understand the output, or the covariates you come up with don't make medical sense. So I think all the example presented today will move this new field forward within the general MIDD paradigm. We already laid a foundation so that the overall FDA medical divisions, they already have some exposure to the general model-based drug development or model-informed drug development. So the acceptance level is already there. And we just need to push this a little bit further by saying that, okay, now we have a new toolbox of applying this neural network model or any type of machine learning based models, and then continue this journey of translating all this complicated modeling into something that our decision makers can easily understand. I see a great future, for this field, and I'm sure, the current reviewers and the leaders within the division of pharmacometrics will push this forward, push this field forward in the next few years.
Thank you. Thank you, Dr. Mohammed.
Yeah, sure. I think actually following on the same point with Dr. Kozlowski, I just want to get your perspective as well on the importance of explainable AI in the MIDD setting, but perhaps you can cover it as well from bringing across to the decisions made by in digital twins and virtual twins, and how much details we really need to get trust in those models.
Yes, thanks. Thanks, Mohammed, for the question, and thanks, Dr. Wang, for your perspective on this important question. IBM has a large focus on ethics in AI, starting with the white paper we wrote in collaboration with the World Economics Forum, and this extends, beyond drug discovery, MIDD, and I don't want to go into every aspect of it, but explainability is a big part of it, whether it's in removing bias from our AI in terms of how categories are determined, to, in the case of explainable or accountable AI in drug discovery, in order to provide the regulatory bodies that exist a better understanding of what we're actually determining with the AI in terms of biological phenomena. And so, in terms of digital twins, I think it's a great, actually, way to approach the problem of explainable AI, because every explanation needs to be in terms of something else. And so the challenge AI presents to us is a very, very high dimensional parameter space that itself needs an analysis in order to to even begin to understand how it's making the categorizations or the determinations that it is. This is particularly important for categorization, I would say. In terms of digital twin, what we have is not only a categorization that's being provided by the AI, but it's a continuous mapping. And we saw in James's talk a very nice example of how you can continuously vary a tumor property within the parameter space of the model. This type of variability or mapping one distribution onto another gives you something in terms of explaining the AI's determination in terms of something else. And that's really why we took the approach we did in order to make the AI not about prediction or about categorization directly, but use the latent space of a mechanistic model's parameter space as sort of the representation layer, such that when it comes time to talk about how the categorization or prediction was made, you could do it in terms of those latent variables, which themselves are very much related to the biophysical mechanisms that the model is capturing. And in the very lively discussion we had, of course, you need to be able to trust the mechanistic model, which is a separate issue altogether. It's not the AI, it's the mechanistic model. So that's where we begin with an assumption that a good model exists either in the mathematics or in the individual's head who's using the AI.
Thank you much, James, for this excellent feedback. Sujata?
Yeah, so along the same vein, Dr. Nova, can you, given your broad, your breadth of expertise that ranges from QSP, pharmacometrics to AI, what's your perspective on the importance of explainable AI in the same MIDD setting?
Thank you. That's a great discussion, and I really appreciate all the perspectives and concur with what has been said so far. Indeed, I mean, I cannot agree more in saying that explainability is really one of the keys to unlock the value of AI machine learning for drug development. We talk about this a bit over the first session, but indeed, the interpretability of a model is crucial, right, to ensure scalability and also build the confidence into the model output. So we should ensure, we need to ensure indeed that these models are interpretable, reproducible, generalizable, and can enable these biological insights, whether it's the drug, the disease, so that we can extract directly from the model, or having something that then can go into a more mechanistic based setting that is closer to the conventionally used method. So I will be also a bit provocative in saying that, okay, this is about trustworthiness. We talked about this morning, and then you have interpretability and generalizability. So it's a trade-off. For example, if you have a model that is performing very well, but it's not interpretable, right? So would you give up in explainability to ensure the generalizability of it? I mean, that's really a chapter and a very hot discussion in the field. But focusing on interpretability, yes, indeed, there is a lot of research going on in fundamental computer science and machine learning. There are really smart people that have developed methods, indeed, for interpretability and visualization of the results. So trying to overcome the black box or making it less black, right? We have seen with my presentation, and also James, SHARP. It's not only about SHARP. We have many other methods that are being developed by great groups with respect, for example, to similarity classification, or try to provide some level of risk equation. So those that it's coming much closer to what we call like a probability of having a special event. So there are efforts going on. I think for us within translational medicine, model-informed drug development, really our job is be aware of these methods and really try to use them in a fit-for-purpose manner, really validate then the outcome to then build this trust within our community. So definitely education, context of use, domain knowledge, and the best practices with the clear communication of the results are going to be key, right? And interpretability can be different also with respect to stakeholders. So that communication, it's really important as well.
Thank you so much. Mohammed?
So I think we are getting a consensus here. From what we heard until now, we see that explainable machine learning methods can really help unlock some of the secrets within this black box, right? and which is needed to build trust in decision-making capabilities of any AI system. And this trust enable wide adoption of this tool for different applications. But I know that there are still some challenges, really. Like, that sounds really simple, very easy to implement. But I think, there are other challenges, and I actually want to ask Dr. James Liu about this, like moving to him. What do you think, beside explainable machine learning development, like, having those methods developed and implemented, do you think there's a need to, there's other things that we need to think about to achieve wide adoption of AI in drug development?
Yeah, thanks for the question. So I think one reason, for instance, we have trust in human models, human generated models, is that we believe that whoever is modeling that has experience of working with similar type of data, and there are certain concepts encapsulated in these models. This person knows about dynamical systems, the concepts associated with it, pharmacological concepts, causality, and that this person will not likely to kind of mistake correlation for causality. So I think ingraining some of this knowledge that we may take for granted, but machine is kind of naive to this world, and many of these notions and concepts are not present when models are constructed. And if we can come up with ways of encoding that knowledge somehow, or using certain knowledge base to make sure whatever model is built conforms to many of our existing knowledge, I think that would definitely go a long way to kind of instilling more trust. So I think it's not only explainable, but that is relying on concepts that we as a community agree upon. Right? That may also help to instill more trust. Also, we have seen examples where, if you, in machine learning, typically you are dealing with a lot of data, and there may be some spurious correlations between variables, and also different missingness patterns. And there have been cases where we, just feed a raw data into the model, and then we see, oh, it does surprisingly well. And then when you look deeper with explanatory tools, and you realize, oh, okay, it's using the fact that patients are missing this variable, that there's something peculiar about these patients, and using that fact to make predictions. So knowing what is driving your model prediction, is it like clinical variables, or is it lab measurements that may not be very reliable, and so on. I think this is also goes a long way towards kind of instilling confidence. Yeah. So
right, and I think yeah, and I think yeah, you are spot on. Like definitely understanding the model, understanding what the drivers of this prediction model is very important. Like especially like for the modelers who are doing this to be able to convince the regulators or the clinical team later, right? Like when they are presenting that. So so thank you, James, for this excellent feedback. Sujatha.
Yeah. So thank you, Mohammed. So circling back to Dr. Wang, and I know some of this we touched on some guidance aspects in the session 1 this morning, but in your opinion, we just wanted to, we do know some regulatory guidance does exist for medical devices to use AI for medical devices for the development of those. When do you think we may see formal regulatory guidance for the same in drug development? And also along the same lines of the, you did mention about the seeds of the adoption of this type of technology is already well planted into the agency at the FDA. But beyond that, you think the FDA, or as well as other regulatory agencies, what they may be doing for the wider adoption of such methodologies? Can you shed some more light on it, some more detail if you have it?
Absolutely. I think, especially in the field of AI or machine learning type of model, our CDRH colleagues are clearly leading the effort, by publishing, white paper or followed by guidance. But if you look at the history on the drug side, the CDR side, in fact, we already have guidance on the traditional models, like the POP-PK models or the exposure response type of models. This is only a different type of models. Whether you can use model-based analysis for regular decision making, that was already accepted, within CDER, across the entire CDER, across many different disease areas. Whether, in fact, even in that field, if you talk to the decision makers, whether they truly understand the details of what type of models, whether it's a simple statistical linear model or a more complex pharmacology Emacs model, they may not know the details. But as long as you present the results in a more clinical oriented way, they will buy the result, buy the outcome, and accept the conclusion. Therefore, in that sense, I would argue, the CDER side already has multiple guidance supporting, the traditional model-based analysis. Whether this new classes of more machine or data-driven models rather than mechanistic-based models will also get the same level of trust and acceptance within the CDER, the drug field, will depend on, whether we will come up good examples that can solve challenging issues where, let's say, the traditional or simpler models couldn't solve. And the confidence or the trust level will eventually build on real examples where you can demonstrate the value of applying this special class of models in the real review cases. And of course, in the CDRH, in the device field, they directly started with this type of models, right? Either imaging, identification, or classification. They don't have the traditional model to even as a starting level. Now all of a sudden, all this machine neural net model based device or software submissions came in, and in fact, our CDRH colleagues were pretty open-minded. They did not argue or require you have to make this fully transparent, make us understand everything before we can prove this. They use risk-based approach. It's just like what they did with the traditional device. If the risk is low, you don't even have to convince the FDA, this need to be reviewed. But if the risk is high, then they will put certain requirements on the, we call it software as a device these days. So I think, the two centers will eventually work together. Whenever we come up with a real example, we have to use this, neural net or model-based analysis to drive the decision. Otherwise, I would say they will always prefer the simpler models or simpler approaches where it's easier for the decision makers to understand. So I think that's the general direction, because even in the traditional mechanistic model-based world, we try to avoid too complicated models. Let's say if we can solve with a simple linear model, we'll not go to a complicated PK/PD nonlinear model. If you can solve that with the simple exposure response model, you don't want to go to a QSP model to make the things too complicated. So I think that's a very simple principle.
The law of parsimony. Yeah, thank you.
Thank you very much. So, yeah, I think the name of the game here is simplicity, right? Simplicity with precision, right? So returning back to Dr. James Kozlowski. So I really like your presentation, that you make it seems very simple, very easy to do, but I'm sure that you may have several challenges as well as opportunities, really, to implement this model, the virtual twins and digital twins for different purposes in different therapeutic areas. So I'm just out of curiosity, can you share with us what are the challenges and opportunities in implementing this really in real life? You have the model development, then validation, then after that the adoption. So just wanna get your feedback about how you can really move this forward for wide adoption, and what are the challenges and opportunity there to do that?
Okay, so looking back, the biggest challenge, and expansion to my knowledge that came out of this work with my team was really understanding whether it's a digital twin or a mechanistic model, that there is a certain amount of bias that the model induces in the problem. And so undoing that bias, much like histogram equalization can undo a certain bias, but in this case it's in a very complex high-dimensional model that was constructed because it can actually generate data that looks like the observations. But we can't rule out that there's going to be some intrinsic bias in the way in which it does that to sort of morph the distribution of parameters to outputs in a way that is not really biophysical. It doesn't have anything to do with the biophysics. So if you go back to my talk, we're really thinking about this in terms of models that are imperfect, and then use AI to remove their imperfections, if you will, in order to provide a way to sample and give rise to real-world useful digital twins. Looking ahead, and I don't want to talk too long, but looking ahead, in terms of what the challenges are moving forward, I just wanted to emphasize that the ability to construct digital twins and to understand that model-induced bias, a large challenge is computing the data. In other words, computing with the model enough samples from the model to reveal that bias such that AI can learn how to undo it. And so, we at IBM are rising to that challenge, and we're building systems to scale out that type of database construction for parameter to model output sort of transformations, compute it many thousands of times, and then ultimately used to train the AI, which can be applied to very small data sets in the way that I described. Thanks.
Oh, that's pretty exciting. Thank you very much. Sujatha?
Yeah, sure. So coming back to Nadia, from your experience working across different stages of drug development, right? So do you think AI would provide most value at more of the early stage or the late stage in clinical development?
Thank you for the question. So yeah, indeed, initially AI methods were mostly used in drug discovery, right? But now we are seeing indeed many opportunities and applications in drug development. We have seen, so the short answer is I do see many opportunities across the value chain of drug development. We have seen, for examples of how we can automate process pipelines. Today, I think another big opportunity comes from clinical trial design, so making them more efficient, the clinical trial operation design with projections really of the technical success, site selections, patient-centric, so ensuring diversity and inclusion in the clinical trials with representation of diverse population. I think AI machine learning can do a lot there. In fact, there are solutions already available. I mean, we have talked about digital twins of virtual control arms and data augmentation, of course, are going to be really relevant or open up for many opportunities. Disease progression modeling, right? So by including AI, machine learning application, we can really integrate all the large multidimensional, multimodal data that are being generated. We have omics, we have novel biomarkers like liquid biopsy, including circulating DNA. We can leverage priors coming from various data sources, so including real-world data. We can mine literature. So I think indeed this is yet another tool, but that can really leverage the kind of data that are being generated nowadays in the digital age of medicine, really with the totality of evidence mindset that we should have to build the confidence into the drug and into the disease. For example, by mining literature, we may initiate already first human trials with a very strong disease model, where then we can plug in data on a specific drug to isolate and better characterize the drug effect. So somehow building confidence and also accuracy into the prediction of the probability of success. We can inform combination therapies as well as potential patient subpopulation. We've seen some example, for example, how we can assess predictive prognostic factors, indeed by integrating multidimensional data baseline and also longitudinal covariates. On top, I think we need to pay and maybe invest more into safety prediction, because we have the opportunity here to integrate data with respect to compound properties in vivo, in vitro, up to human, and really to support forward and back translation. So, and this can be done in various ways, also with transfer learning. On top, we are going, we are having decentralized trials. We have telehealth technologies with remote patient monitoring. So, and this is, yes, it's used to monitor patients, but in fact, we are going to collect a vast amount of data that eventually can inform us and support clinical trial decisions as well. As we touch base a lot on GANs, yes, we talk about virtual control arms, data augmentation, but on top, synthetic data generation can also support data sharing with the privacy-preserving solutions, right? So, and I think this is really important because we say trustworthiness data and also the assessment of the cost-benefit threshold of these approaches with respect to conventional ones, right? So the cost is very high. Now we really need to, I said this morning, demystifying the hype of AI machine learning. There is a lot of expectation, but we really need to assess the value and the cost-benefit threshold of these techniques with respect to conventional ones to really build the confidence and have a successful adoption.
Yeah. Thank you so much. Yeah, the sky's the limit with the amount of information that's available, and of course, asking the right question at the right stage seems to be the most challenging part of this. So thank you for that. Just back to you, Mohammed.
Thank you, Sujata. So back to you, James Liu. I actually have a question, from your daily today interaction with different study teams at Genentech. I'm sure that when you build a model, most likely you will share with them how can we utilize this model for maybe building a prospective study to utilize this model. So just wanna get your perspective on this convincing step. What's your experience so far when you have a traditional interpretable model versus a model that just a black box? And did you feel when you added the explainability part to this model that the team became more convinced and really moved with your proposal, for instance, to have a prospective study to utilize this tool?
Yeah, thank you for this question. So before you convince the study team, you need to first convince the modelers, right? So because they have been doing this, for a long time, and they know their models very well, as well as their gaps and biases and so on. So traditional parameters like KG has a very easy interpretation. Is the tumor regrowth rate. But we also know some of the limitations, right? You may have to observe the patients for a longer time to be able to estimate that well. So I think so far these explanatory tools, while we still need to kind of come up with kind of terminologies to describe what we are seeing reflects the kind of what concept we use to describe what we're seeing in the dynamics as we vary these two number of parameters. I think it definitely has removed some concerns regarding whether or not this model is doing something sensible or is it doing something that is just purely spurious, right? If you were looking at longitudinal tumor size data, you just take the time of the last tumor measurement, use that to predict survival. Maybe that could work well in a limited setting, but in settings where you have different enrollment rates and so on, that could be very limited. So I think not only explainability, but doing additional simulations, like looking at the operating characteristics in the practical trial enrollment setting. When you at this particular time, you have this data and you want to make predictions for the future. I think these are all very key part of the process. So yeah, definitely, I think convincing the modelers would be the first step. And after that, we can involve the other stakeholders.
Thank you very much, James, for that. I know that it's not an easy job for sure, to go through the entire process, but I think this is most likely part of the development of those methods, in the entire industry. So So hopefully, we'll have more wide adoption of those methods moving forward. I really wish that we can stay for 1 full hour, but we have 4 minutes to go. So I'm gonna be turning over to Sajatha to ask our last question. And thank you so much for this fruitful discussion, really. Sajatha?
Let's see, we had 3 more for Dr. Kozlowski. So I'm just going to ask the first one that's showing up on my screen. So does the GAN method require a large data set, or would a small sparse data set be sufficient? What will the performance of such models when data are smaller sparse be? This is for Jane.
Sure. Thank you, Sujatha, for that question and for the discussion around it. I did want to mention that the GAN in our approach is not generating the virtual patient. So this is an important point, and the reason is that if we use the GAN to generate the virtual patient, it would be subject to the sample limitations from the real world data that would be used to train it. And while people are doing that productively in imaging, for example, where there are large numbers of data to train such generative models, we've taken a slightly different approach. Largely because my team is a long history in mechanistic modeling and building large models of neural systems and cardiac systems, and more recently cancer and pharmacometrics. But the reason is we think that the prior that comes with the systems of ODEs and very sophisticated, refined mechanistic models that are computed in an iterative time step manner on regular CPUs, as opposed to AI models, which are computed, in new types of architectures on GPUs. We think that prior can be exploited in new ways with AI. And so, to answer the question in a nutshell, we don't have any limitations on the amount of clinical, preclinical, biological data that we use. In fact, we published a paper on omecamtiv mecarbil with an N of 21 data points that we ran through the inverse model in order to infer the mechanism of action of the drug. What we're challenged by is how to fully sample the mechanistic model's parameter space in order to get a good picture of that bias I was mentioning in an earlier answer, and then train the AI to undo that bias in order to give a coherent representation of those small numbers of data, precious data from biology, that we want to project back into this mechanistic model parameter space and thereby derive a biophysical explanation for what we observe. So that, fortunately, is just a computing problem. When you're from IBM, it's nice to have that. And we are, computing these models many thousands to 1 million times in order to train the AI. Computing these mechanistic models, I should say. Yeah. Thank you.
Great. I think
thank you for the great questions.
I think we are almost at the end of the of this panel discussion.
Thank you, Mohammed. You can close it.
I'm sorry.
All of the panelists' time today. Yeah.
Okay. Looks like my internet connection went wrong. So apologies for that. But I would like to thank all the panelists for this excellent discussion and for the speakers for their great presentations. I also would like to thank you, Sujatha, for your help moderating with me this session, and thank for all the audience for attending and raising excellent questions for the discussion. This concludes our session 2 of this workshop. Thank you very much, and have a great rest of your day.
Thank you, everyone, and thanks to
Thank you, everyone. Bye-bye.
Bye-bye.
We'll be back at 4:05 for session 3.
Hello, hello, hello. Hi.
Thanks everyone for joining. We'll get started in a few moments. Good afternoon, everyone. Welcome to session 3 of the workshop, NLP, Natural Language Processing and Quantitative Pharmacology Modeling. Before we begin, I'd like to remind you of a few recurring housekeeping items. All attendees will be on listen-only mode and hand raising is disabled. Time permitting, we'll take a few questions after individual presentations, but we'll save the majority of questions for the session panel discussion. Enter questions at any time in the question module. or click on the question mark at your control panel. If possible, please preface your questions with speaker name and the slide number. This workshop is being recorded and links to the recording will be available after the workshop. Click the handout icon if you want to view and download the connection and audio tips handouts for possible solutions to any audio disruptions or delayed slide transitions, as well as the speaker biographies for your reference. Note that any questions submitted but not answered during the session will be sent to speakers and panelists offline, and we'll figure out a way to do some sort of post-meeting follow-up with them. Thank you all for joining this afternoon's final session of the day, where we'll be focusing on natural language processing and quantitative pharmacology modeling. James Liu of Genentech and I, James Liu, you've met in sessions 1 and 2, and I will be moderators for the session. I'm Tom Tensfeld, the Senior Director of Pharmacometrics in the Clinical Pharmacology Department at Pfizer Global Product Development Division. Our first speaker this afternoon is Jenny Ding from Eli Lilly and Northwestern University. Jenny received her bachelor's degree in microbiology, immunology, and molecular genetics from UCLA and her master's in healthcare analytics and information technology from Carnegie Mellon University. She had prior research experience building clinical decision support tools to predict disease prognosis from electronic health records and developing neural networks to understand viral divergence from sequencing data. In her role as a data scientist at Merck's Quantitative Pharmacology and Pharmacometrics Department, she used natural language processing algorithms to automate literature selection from meta-analysis and machine learning models to reduce the dimensionality of Parkinson's disease digital biomarkers data. Most recently, she'll be joining Northwestern University's Biomedical Informatics program, PhD program, to develop multimodal models, that's a mouthful, to understand disease subtypes and aid in the development of precision medicine. Jenny, we're going to switch to you and your presentation now.
Yeah. Thanks, Tom, for the introduction. Okay.
I've made you presenter.
Okay, thank you. Okay, thanks everyone for attending, and it's my pleasure to represent our work at Merck to present on using natural language processing to streamline literature selection for meta-analysis. So before we going into more details, I wanted to explain what meta-analysis is. So what it is that it uses published evidences from databases like PubMed to inform discovery and clinical decision making. And on the right, it's a Prisma flowchart for a typical meta-analysis attempt. So we would start with about 1,000 literature in this example. And upon initial abstract screening, we may be cutting down about 4/5 of them. And then upon more scrupulous examinations of full text and specific search criteria, we might cut down more of it and retain slightly less than 10% at the very end. So as you see, this is a very lengthy and laborious process that's also very costly. So what we're presenting here is that instead of using humans at every step, we wanted to try if we could use the natural language processing algorithm to automate the process. So for example, we wanted to test on whether natural language processing could help with the initial filtration of abstracts and more refined ranking instead of having human intervene at every step. We also proposed that we could use name entity recognition to automate the extraction of entities like patient, intervention, comparison outcomes. So as you see, this using computer-aided process here, it cuts down a lot of processing time. It would only need a few minutes of runtime and very low computing costs in comparison with 6 hours FTEs months. So we actually tried a few NLP methods to compare the results. So on the left, it's a search from a raw PubMed search by typing in the keywords on the PubMed search bar. So using the green cutoff box shown on the left, only 32% of abstracts selected are deemed to be relevant, which means that they are indeed included in our Merck's meta-analysis database. And so we also compared with a few other methods, which includes Facebook spelled Synthwek, which is embeddings trained on both the PubMed database as well as Mimic3's clinical database. And then we also compared with Google's Universal Sentence Encoder, which is a general domain embedding, meaning that it's trained not only on biomedical text, but also on other texts from other areas. And both of these methods are able to provide slight improvement. However, with the results not as satisfactory. We also tried the TF-IDF method, which is a term frequency-based approach by comparing term word frequency. And so we were able to achieve 55% relevancy using the cutoff. So you can see more red documents are focused on the left. However, this result is still not as satisfactory, so we went on to search for more alternatives. And so what came into picture are transformer models. So in 2017, transformers are published in the paper Attention is All You Need for sequence-to-sequence classification tasks. And then in 2018, using the transformer structure, BERT came into the picture to process long sequences like languages and now DNA sequences. In 2020, PubMed BERT is published by Huifeng Poon's team from Microsoft, and Huifeng is actually one of our panelists today. So, and the side note for 2021, we all know that AlphaFold became a big hit and it's capable of predicting protein 3D structure, and that's actually also a BERT-based model. So going more into our model and the training process. So looking at focusing on the left orange, we have all search results from PubMed BERT once we enter our query of keywords on the search bar. And so out of this big amount of literature that got returned from the search, only a small percent of them are included in Merck's meta-analysis database. And we label those literature as relevant in comparison with those irrelevant ones that are included only from the search, but not ended up to be in Merck's meta-analysis database. So having those documents will feed them into the PubMed BERT tokenizer shown on the bottom right. And so this PubMed BERT tokenizer, so this PubMed BERT is trained by Huifeng, and it's trained on PubMed, both the full text and abstract. So after we tokenize our documents and we transform them into embeddings, we classify them using a densely connected neural network classifier. And then from there, we are able to achieve the relevant versus irrelevant labels predicted by our model. And so this is going into the explanation of transformer models. So what transformer models have is that they have attention mechanism that can learn semantics. So looking at the graphic on the left, if I zoom in on the first cut, we could see that words like rabbit, quickly, hopped, they're all connected to other words in the same sentence, and they assign different weights to different words in the same sentence sentences. And so each color band here represents one neuron head, and the color intensity represents the magnitude of connection, and the hue of the color represents the sign of connection. So for example, hop would have a more positive connection to rabbit versus turtle. And so lastly, looking at an example of crawl, we see that there's higher intensity for crawl to be connected to turtle, slightly less for slowly and crowd, and there's not much connection from crowd to words like rabbit, quickly, which is not that relevant. So there's less attention there. And so the reason why we used attention mechanisms in comparison with word frequency ones is that if you look at the Tisney visualization in the middle, this is a 2D visualization of words from relevant and irrelevant abstract. There's quite a lot of overlap, so we can't really differentiate words that are in relevant abstracts versus those that are irrelevant. And the word cloud on the right shows very similar results. So looking at it from our initial look at the word cloud, both of them have words like patients, treatments, and the amount of them are pretty similar. That's why we needed to understand semantics here. And so using our trained model and added classifier structure, we tested our model on two different tasks. So the first task is whether our model could predict diseases that are not included in the training data set. So we call this task leave one disease out cross-validation. So in this example, our test data set is asthma, shown using this purple box on the top. And our training data set is all the diseases from HCV to endometriosis listed in the blue box. And so we were able to achieve 100% recall with this task set and 82% precision. So a quick note on recall versus precision. Recall is the percent positive that are able to be retained out of all true positives. And precision is the percent of true positive out of all those predicted to be positives. So having a precision of 82% means that 18% of what our model predicted to be positive are not actually selected by Merck's experts to be included in the meta-analysis database. And since this is a cross-validation task, our next move is to test on the HCV test dataset. So in this case, we train a completely different model on datasets from asthma to psoriasis to endometriosis. And so our result for HCV is shown again in the purple box. So we were able to achieve another pretty high recall of 93% and slightly lower precision of 28%. And this is because we wanted to capture as much positive literature as possible. And we repeat this process of retraining the model for all the other diseases in the data set. And this is our final result. So overall, we are able to achieve an average recall of 85%. So this means that 85% of all those that are included in our meta-analysis database are able to be retained by our NLP model. And our recall is slightly lower at 31%. This means that we are capturing a lot of literature that potentially might not be relevant to us. And so we also tested this task on another task. This is to test whether our model trained on historical data can classify new publications. So in this model, we trained a pan disease model, which means that it has all 14 disease areas. And our training data set is the previous 3-year data for each disease. And our test data is the most recent 3-year. So for example, for things like asthma, we would train on the 2002 to 2006 data, and our test data would be 2007 to 2010. And we have the same for all the other disease areas. So the results are shown on the bottom. So looking at the graph on the chart on the left, we see that most of the red literatures, and red here represents relevant literature deemed by Merck experts, are concentrated on the left, which means that our model gives them a pretty high inferred relevancy score. And this is what we are hoping for. And the confusion matrix is shown on the right. So we are able to, so we have a recall of 77% and a precision of 28%. And so this is, we also tested on our model on, and so also tested our trained model on a pilot task, and it's pretty relevant on the endemic coronavirus. So our initial pilot, so this is our first pilot, and this pilot is for this MBMA goal is both clinical and non-clinical. And if you recall, our training data was only clinical, so which gives a limitation. And the outcome for this MBMA attempt is to include viral replication, titer, antibodies, hospitalization data. And the purpose for this MBMA is to build a model that can use animal data to predict vaccine success. So this flowchart shows the process for this MBMA attempt by human. So we start with about 800 literature, and then using a rule-based filter, we were able to cut down about half of them, and then only 16 of them are finally retained by Merck's experts. And this process is very lengthy. And so we did this head-to-head comparison with our NLP model, and the confusion matrix is shown on the bottom left. So out of the 16 literature selected by Merck's experts, our NLP model retained 14 of them, and only 2 of them are missed. And then, however, it also included 60 other literature that are not selected by Merck's experts, but was included in our model. So what we noticed is that using our NLP model, we are able to cut down to 74 instead of 46 by rule-based filter by our vendor. And that's a 95% reduction versus 44% by a quick scan. However, we know that we still need to reduce the false negative ratio, and because the tool literature that was not selected by our model could potentially be very helpful in information. So that's what we are needing to work on. And so our next step is to carry out an entity extraction for enhanced flexibility. So in this example, we hope to extract features like patient data comparison, which is liraglutide versus sitagliptin, and treatment, and also outcome, which is which drug is more effective in this case. And so the conclusion here is that our BERT model can outperform traditional NLP models like PF-IDF, and it offers a potentially cheaper and quicker alternative to human labor. And we leverage the state-of-the-art PubMed BERT in this attempt. And so in the test set so far, we carry out 2 validation tasks. We're both able to generalize to unseen diseases and also future publications. And our future efforts include to conduct more testing over different therapeutic disease areas, reduce false negative ratio by fine-tuning, and then potentially expand on functionalities such as entity recognition. And with that, that concludes my presentation. So thank you all for listening.
Thank you, Jenny. I don't see any questions that came through. I think what we'll do is, if you're okay with it, we'll hold some questions that we have until the panel discussion.
Sure, that works.
Okay. Our next presentation is with Frank Kloprog. He is, so get my notes up here, the Wellcome Trust Sir Henry Dale Fellow of University College London. As a clinical pharmacology modeler, Dr. Kloprog is interested in parameterizing the relationship between drug levels and drug effect. At the University College London Institute of Global Health, his main research focuses around 3 basic themes. The first theme is in vivo PK/PD properties of anti-tuberculosis drugs and exploring candidate biomarkers for cure. Frank designs and runs population PK/PD studies and observational cohorts in the UK and abroad. Secondly, he also investigates the biomarkers for bacterial subpopulations that can survive treatment. He has set up in vivo mimicking in vitro experiments, hollow fiber experiments of infection at UCL. And finally, he is one of the co-founders of PKP the AI, an initiative that brings together quantitative pharmacometric knowledge into a single open source platform, making it amenable to the application of machine learning. PKP-DAI has been leveraging natural language processing as due to the size of publicly available data. Manual curation is not a viable option. We'll switch to your presentation now.
Thank you.
And Frank, please take it away.
Okay, you can hear me, I guess.
Very well, thank you.
Okay, thank you for the introduction, and thank you for the organizers for inviting me to speak tonight. I will present our work on automated approach to extract pharmacokinetic parameters from scientific publications. Next slide, please. The slides are gone now.
Frank, are you seeing the slides now?
No, I can see myself.
How's that?
Not good.
All right, I have a problem locally. Give me just a moment.
I can try it. Yeah, they're back again.
Very good.
Next slide, please. Yes, thank you. So I'd like to start with acknowledging all the people that are involved in this project. First of all, Farhan and Vicky. They are PhD students working on this project, and all the results I will be presenting are actually their achievements. Wati and Joe, together with them, I do supervise Farhan and Vicky, which is a great pleasure. Very clever, it's an amazing project. And obviously, a very big thanks to all our contributors and the collaborators, because as you will all know, machine learning and artificial intelligence algorithms only get as good as the data is labeled. So we have an immense amount of pharmacologists that voluntarily help us labeling the enormous amount of data. Next slide, please. So the overall aim is actually to centralize and structure a repository of PK parameters from scientific literature, and do this using machine reading. And how we envisaged such an open source database would look like is something alongside what we are having here. So in the ID column, you would have basically your PubMed ID, for example. In your drugs column, you would have the drugs that the study is relating to. In the drug descriptors, you could think of in vitro or about in vivo. In intravenous or per oral administration. Parameter data, that's what it is about. It's about AUC, clearance, volume of distribution, demographics, whether it was healthy volunteers or patients, the drug treatment, in case it was a product that was given, for example, and the modeling approach, non-compartmental analysis, population pharmacokinetic analysis, and additional information. Next slide, please. So with that in mind, why would you want to have such an open source big database? Well, as we all know, and we are all pharmacologists by background, or we work in the field, and we all know that it is actually very valuable, the various pharmacokinetic modeling approaches. For instance, in the early drug development stage, QSAR, SAR, PBPK models of great value. They mainly rely on a reductionist approach in the sense that the whole body is being simplified in a series of ordinary differential equations. And this allows us to make first-in-man predictions, special population predictions, which is all great, but there's also various situations that it's actually not performing as we would have liked to. The next situation is the development of population pharmacokinetic models. This is usually much more precise and accurate, although it comes with a lot of knowledge and labor, which makes it an expensive exercise. And lastly, for instance, TDM predictions, such as Bayesian and AUC forecasting, which is gaining more relevant with more computational power recently. There, obviously, the more models, whether it's in a model averaging framework or in various other hierarchical frameworks, it doesn't matter, but various models need to be considered. Well, all these approaches are good in a sense, but they each have their own disadvantages. And one way to overcome it, in general, could be machine learning or artificial intelligence. And whereas in other fields this has already gained relevance, in the field of pharmacokinetics, the achievements are relatively limited, and that's all due to the lack of a big data source. Because as we all know, we really need big data in order to develop good ML and AI algorithms. Now, there have been efforts to actually develop and make these databases publicly available, although they're all manually curated. And that's a big disadvantage, because there is approximately 500,000 papers containing PK information laying around that still then would need to be transposed into tables. And we have an influx of a couple of thousand papers each year. So it's very difficult to keep up with the influx as well as to work out the backlog, basically. So next page, please. Our approach is different. We propose to use a machine learning approach, an NLP approach, and we have chopped it up, our pipeline, in 3 different elements. First, we identify which papers are likely containing PK information. When we have determined which ones these are, we go into those papers and we use name entity recognizers and relation extractions to actually pull out those pharmacokinetic information. And then lastly, as we all know, most of the pharmacokinetic information, parameter estimates, are in tables. So that's the last, but maybe also a very important exercise is the table extraction exercise. Next slide, please. So first, binary classification of papers in order to see if they are likely containing PK information. So what we did, we needed to build a corpus, in fact, too. So what we did, we typed in pharmacokinetics on PubMed and made a random selection of papers. And we built a training corpus and a test corpus. The training corpus was labeled by 2 independent experts, pharmacologists in the field, and a referee third independent labeler was, in the case of disagreement, reaching a consensus between. So there was actually various meetings. So it was a laborious process. We did this on 4,000 articles. Then we had a test corpus. We wanted to have more confidence in that it was right. So we decided to go with 3 independent labelers on these 800 articles. The training and the test set actually all had a title, little under 90% had abstracts. We had authors, journal, publication type, keywords, mesh terms, chemicals, affiliations available at the following distributions. And this information we used to actually train our binary classifier. And most important, we saw the relevant versus not relevant distribution was 20 over 80% in both data sets. Next slide, please. Subsequently, we used extreme gradient boosting and unigrams using a BioBERT mean pooling approach to develop these models. So a little bit background, we did 200 bootstraps and we did split our training data set in a training split, a development split, and a test split. And we did this because we wanted to avoid overparameterization. So we ran the first epoch or run of the XGBoost trees on the training data set, and then we predicted how it was performing on the development data set. Subsequently, we retrained it again by adding another tree, and we tested it again on the development until we reached the asymptote. And then we stopped the experiment and we tested it on the final 20%. That's the training data performance, what you see on the top right. The best performance, as I already mentioned, was using the unigram and mean pooling approach. And we had an F1 score of 81.7%, which is basically the mean of the recall and the precision. And this is the readout I will use for the rest of the presentation. What we then did, we reran the model as it was on all the data, training data together, to basically update the hyperparameters. And it was used to predict out the test data set, which actually achieved a slightly higher F1 score. And this is obviously due to the size of the data. This work is now published in the below publication. And next slides, please. Subsequently, we knew which papers are there, so we needed to actually parse all the information for those papers likely containing PK information. But first, you would need to develop a name entity recognizer and a relation extraction. So what we did, we parsed those sentences and we ran them through Prodigy, which is a labeling platform, an online labeling platform. It looks like as displayed here. Sentences are presented to the labelers. They can select spans and label it as PK relevant or not. This rendered us with a training set of 2,800, a development set of 500, and a test set of 1,500 sentences. As you can see, the development and the test set actually represent a true distribution of what you would see in PubMed. And using active learning, we actually upped the percentage of sentences with PK mentions in the training set in order to achieve better training performances. For REX, so relation extractions, we also use Prodigy. we didn't only label the spans for pK, but also for units, for numeric values. And obviously, we labeled the spans or the relations between the different entities. And this yielded another 2,100 training, 500 development, and 1,000 test sentences with the following entities: pK, units, value, range, compare, and the relations between them. Next slide, please. The following models were developed: neural network. I mean, we use SPACY, convolutional neural network, eventually turned out to be the best. Using a BERT model that was pre-trained using PubMed abstracts, makes it BioBERT. It was already explained in the previous presentation slightly, but the whole idea of BioBERT is actually to predict out the entities using the following layers as displayed here. In order to make the relation extraction model, it was a bit more complicated because it became a multi-learning task. The architecture is the same, but instead of just predicting out the pharmacokinetic entities, the units, the numeric values, etc., also had to be predicted out. And if we really made it multitasking, we also tweaked it to do the relation extraction simultaneously. Next slide, please. And the following results could be achieved using a BioBERT model. We are looking at strict F1 scores and partial F1 scores. Let's take oral bioavailability as an example. The strict F1 score relates to if the model was able to predict out oral bioavailability as a PK term, and partial, for instance, if only bioavailability was labeled as a PK entity. The relation extraction, MT versus no MT stands for multitasking. Multitasking, as I said in the previous slide, relates to it does everything at the same time and also the relations, and no multitasking refers to you first do the NER, and on the back of that you do the relation extractions. And what we saw was that actually there was a slight improvement when you did everything together, which was hinting that the algorithms could benefit from the structures around the entities in the sentences. So the whole network on its own was more predictive and benefiting from all the information surrounding. Next slide, please. So we had developed our algorithms now on text, abstract text, and this allowed us actually to run our pipeline. We had identified 120,000 papers likely contain PK information. Of those papers, 113,000 had an abstract that was accessible through PubMed. Of those abstracts, approximately 65,000 had actually PK estimates in it. And using our NER and REX models, we pooled those estimates, and that was well over 250,000. We deposited that in a tabulated form on pkpdai.com. Please do take a look, and did the following QC tests with it, and among it was the following case study. Can we go to the next slide, please? We took actually a review from our colleague, Joe Standing, and his PhD student, Dagan Lonsdale, which was on scaling beta-lactams, and they just scanned the entire literature for amoxicillin, piperacillin, and meropenem. Obviously, they read it manually, but we were just interested, okay, on the basis of the abstract data, which what our models were validated for, how much of the abstracts actually contain clearance or volume parameters? Well, we noticed that it was somewhere between 40 and 50%. So what would be the recall rates for those abstracts be? And we found that it was actually very high, up to 100%, and certainly not lower than the higher 80%. So we were very satisfied with that. Next slide, please. Obviously, there is also false positives. So we are looking at the top panels before cleaning and before cleaning for clearance and volume. We removed the animal studies, for instance. We normalized all the units and ended up with, after cleaning, But obviously the whole process of running this abstract and doing the cleaning was in the order of hours, as opposed to a real Prisma review by 2 independent labelers going through thousands of papers, which was in the order of weeks or if not months. So there was a massive reduction. Next slide, please. So far, what I've presented all relates to abstracts. Work in progress is food text machine reading. So we have parsed well over 7,000 XML PK papers. They contain well over 1.1 million sentences, and of those sentences, 260,000 are in result sentences, and of those, 200,000 are containing numerical values. Machine reading is ongoing. Once that's ready and we have done our QCs and case studies, we will add this information in a tabulated form to the database that is already available and currently only contains information abstracts. Next slide, please. Lastly, the table extraction. This is also still work in progress. We still use Prodigy to label table cells. So, for instance, in yellow, a table cell is indicated, and then the labeler has to say whether it's PK or numeric cell. This rendered us with 2,500, 1,000 training, development, and test table cells. And amongst those, we have well over 2,000 PK unit and numerical cells. And using this data sets, next slide, please. It was, by the way, labeled by 2 independent labelers. We developed again an NER model, which was based on transfer learning on the previously presented. It was run on text. And this is still work in progress, but the confusion matrix actually shows that the predicted labels are surprisingly accurate compared to the true labels. And the performance for units, numerical values, and PK parameters are very high in the 90s. F1 scores over 0.95. And we compared it to annotator F1 scores. So what is the agreement between one versus the other annotator? And we actually showed that the model in its current form for those entities is actually similar as a single labeler. Obviously, there is various entity labels that are not at all nicely performing, but as said, this is work in progress, and we work hard on optimizing the model. Next slide, please. Well, resting me to say, please all use pkpdai.com. Visit it on pkpdai.com. Please report bugs or come up with any suggestions on info@pkpdai.com. We are very keen to know what you find of this creation. Thanks.
Thank you, Frank. A very interesting presentation. I'm not seeing any questions that came through. I have a quick question myself. One of your comments was around the cleaning phase of the data, where you were pulling animal data out of the data set. Was that something that could be automated, or was that something that had to be done manually, or was it part of your pipeline?
Well, the algorithms as they currently are, they exclude reviews basically. So it is including animal and human data. For this particular case study, as we wanted to actually benchmark that review, which was in-house, and that review was only done in human data. So we couldn't, there is no functionality as of yet that can actually distinguish. It's a relatively easy job, I agree, but there are so many things to do. And it's certainly on our to-do list, but yeah, we have to take it step by step.
Very good, thank you. I don't see any other questions coming through. We have some other questions that we'd like to ask, but we'll wait until the panel session. Good. All right, thank you. We're going to shift to our next presenter. So I'd like to take back this slide. Okay, I'm hoping now that you can see my screen.
Hey, Tom.
Yes.
I will present myself.
Yeah. Let me introduce you,
Okay.
Jinping.
Yeah.
Yes, I understand. So our next presenter of the afternoon is Dr. Jingfeng Zhang. Jingfeng is is the CEO of Insilicom LLC and professor of the Department of Statistics in Florida State University. He obtained his bachelor's degree from Peking University in Beijing. In 1997, he came to the US to pursue graduate studies at the University of Illinois in Chicago, where he obtained a master's degree in chemistry and a master's degree in mathematics and computer science and a PhD in bioinformatics. He received postdoctoral training in the Department of Statistics at Harvard, and then he joined the Department of Statistics at Florida State University as faculty in 2007. His research lies in three major areas: biological information extraction, integration, and mining, genomic data analysis, and computational structural biology. Dr. Zhang began a leave of absence from Florida State University in the summer of 2022 and is now working full-time on his startup company, Insilicom. Insilicom provides solutions enabled by advanced AI techniques to harness the power of big data to accelerate R&D efforts in industry and academia, specializing in document retrieval, information extraction, knowledge management, and knowledge discovery using graph-based AI algorithms. It's working with pharmaceutical companies to accelerate the drug development and discovery processes using modern AI techniques. Fang, I'm going to make you presenter.
All right, thank you, Tom, for the introduction. And also, I would like to thank the organizers for the invitation and for the opportunity to share our work. Can you see my screen?
Yes.
Okay. Yeah, I will start with a brief introduction of the background. We live in the so-called information explosion era, and we have to deal with a lot of text data. In pharmaceutical research, we have to read a lot of literature, and also health information can also be found in electronic health record and even social media posts. And this figure shows the increase of our publication from PubMed. So to make effective scientific discovery or make solid decisions, we have to use all the information we have. However, most of the time we cannot, because it's just impossible to read all the text manually. And actually the gap between all the available information and the information we can actually use is widening. So what should we do? A reasonable approach is to use information extraction methods with a computer to extract information automatically. Of course, there were many different information extraction tasks. For example, the one Frank just talked about. But I will just give a general framework for extracting information from literature and then convert that raw text into a structured format. Usually, it can be divided into two steps. The first step is extracting entities. This is also called named entity recognition, NER. For example, for this paper, we can recognize diseases, chemical compounds, genes, or proteins. And the next step is to extract relations among these entities. And that will give us this so-called knowledge graph. And then we can build applications on top of knowledge graph, because this is now become structured information. But the problem is actually quite challenging. So first, we have very large volumes of literature and also some other forms of biomedical text. And then the speed of their generation is actually increasing. And also, biomedical knowledge inherently is very complex, and people use very complex languages to describe such knowledge. And there are a very large number of entities, for example, diseases or chemical compounds, or tens or even hundreds of millions. And there are many synonyms, and also aberrations are commonly used, and the same aberration can mean different things. Of course, not even mentioning people publish conflicting information. So basically, understanding natural language is a very difficult problem. So it's not surprising that although deep learning has made a lot of breakthroughs in many other areas and beat humans, but cannot kind of beat human in understanding natural language, because it's really very difficult problem. So to facilitate the methodology development and also identify the most effective measures, the community has organized many open challenges. Basically, those are competitions, everybody can participate. And recently, NIH, NCATS of NIH and NASA organized a challenge called Litecoin LP Challenge. It actually has nothing to do with several currency, they just call it Litecoin. And this challenge has 2 stages. The first one is to perform entity recognition, and there are 6 major entities: diseases, species, gene or proteins, mutations, chemical compounds, and cell lines. And the second stage is to extract relations among these entities, and there are multiple relations among them. We participated in this challenge and won the first place. So it was really intensive 3 months. We worked really hard. At one point, I kind of even worried that my student may file complaint to the school, because I call meetings after work to organize our team, discuss urgent issues. But fortunately, we win the challenge, and which solved all the problem. Now they all put this on their resume and tell everyone that you have to work really hard to win challenges. So what did we do? So at the beginning of the challenge, we thought we have to develop a really innovative approach to win this challenge. And we tried some ideas, but given the time for only 3 months, so yeah, we couldn't kind of fully explore, investigate these new ideas. We kind of started. We are continuing work on that. But at the end, our framework mainly contained just fine-tuning of large pre-trained language models. But it turns out that do really well on fine-tuning these large pre-trained language models is not trivial, and it really make a significant difference. So we started with trying all these pre-trained models. Yeah, there's actually many different variations. We tried every one of them, and then different variations of each one. And then we also tried many different things in this whole fine-tuning pipeline. For example, different loss functions, data augmentation strategies, and different setting of label smoothing, and different way of ensemble learning. So at the end, here is our final set of components we have. For both NER and RE tasks, we first developed our own script for data processing. We started with these open source packages, but it turns out all the data have their own noises and corner cases, and you have to really write your own to fix all these smaller issues. And then at the end, we use the Roberta Large and PubMed BERT as our baseline models. PubMed BERT is very good because we deal with PubMed abstracts. Roberta is very good because it has very large training data and covers scientific and biomedical text as well. So we kind of covered both spectrum. And we also use ensemble modeling for both NER and RE, and we use label smoothing for some of the models, not for all the models. So part of that is also used in ensemble. So for NER, because there are a lot of aberrations, we have to handle that. We use a program called AB3P for getting the full names of aberrations. And also we assume in the same document, including full text part of the document, the same aberration has to be tagged with the same type and have to point to the same unique ID. Well, this is reasonable assumption, so we check this consistency. And for relation extraction, we develop a multi-sentence model for predicting relations. Because for Litecoin dataset, the annotation was done at document level. So that's why actually it's a quite challenging task. Document level relation extraction has been a very difficult problem. And because Litecoin also use a kind of a special score method, we also modified our classification rule. So how good are the latest deep learning model, including what we find from Litecoin and other challenges we have been doing? So in fact, we participated in many different challenges. I think that's actually very good. We work hard on these. We develop the best method we can. So the good news is for many AR and RE problems, NLP methods now have reached human or near human level accuracy. And this actually happened very recently. But what's the cost? So you may ask, well, if we have to annotate a very large number of documents, and it's still not very feasible. But in fact, another good news is we only need to annotate a relatively small amount of text. For example, in LateCoin challenge, the organizers only annotated 500 PubMed abstracts. 400 were used for training and 100 used for tests. Another example I took from a different challenge called Biocreative challenge. So this challenge has been going on for many years, and they have many more specialized information extracting tasks. For example, for protein chemical relation extraction, in the one before last, the Biocreative 6, the best FN score for protein chemical relation extraction, at that time, everybody already using deep learning, but the best FN score achieved was 69%. And the last one, because pandemic, there was a gap, and Biocreative 7, the best FN score actually reached 80%. And the organizer can declare, well, for this task, right now, the deep learning method actually using these pre-trained large language models has achieved human level accuracy. Yeah, so with this recent development in deep learning in NLP, so we believe it's a good opportunity to develop tools so that many researchers can actually use. So we can really translate the latest discovery to real applications. So we are developing this platform. We call it Biomedical Knowledge Discovery Engine, BioKDE. So what we do is we'll take an abstract text. So far, we are working on all the PubMed abstract and the full text articles. And then we plan to get some more text into our database, and we'll convert all these texts into structured form in terms of in the form of knowledge graph, and then we'll build applications on top of these knowledge graphs. So far we have 6 entities. We plan to expand our set of entities to include not only knowledge kind of more specific to pharmaceutical industry, but also in some other areas. Oh, yeah, by the way, so although we have only 6 entities, but they're really important ones in pharmaceutical research. So for example, if we just take some specific relations, and they're actually very important. The disease mutations, they're very important for genetic testing. So in genetic testing industry, they need the information to generate reports for patients. And then between disease and chemicals, there are actually 2 kinds of relations. One relation is a chemical cause disease, and that's a side effect or toxicity. And this kind of information is very important for pharmacovigilance. Another kind of type of relation is chemical can cure disease, and that will be very useful for developing new treatments, for example, drug repurposing, and also gene disease relationship is important for target identification. So we have built this platform, and we just released a beta version for public testing. So far, we have incorporated the ER part of our results from all PubMed abstracts. Then we're adding the relation extraction part. So right now, the relation part, actually, we use just coherence. If 2 terms occur in the same sentence, we draw an edge, and then that form one edge in our knowledge graph. But we plan to finish incorporation of the RE part of LateCoin results within a month. So yeah, check back. Okay, so that's our landing page. We also have a search engine we developed 2 years ago. We also use a deep learning method to develop a search engine, but today we'll focus more on this relation search part, which is built using the knowledge graph data structure. So here you can search relation among 2 entities. For example, we can search all the chemicals associated or related to COVID-19. So this has been actually challenging because you want to know all these chemicals related to COVID-19, but you may not even know the names. For example, you want to know the drugs can be used to cure COVID-19, but you don't even know the names. So traditionally, it's very difficult to search. And if you just search COVID-19 treatment, you'll get a lot of papers and you have to read them. But with knowledge graph, because we already tagged all these entities, and then we can extract their relations, we can actually just extract such information, and then we can display this as a graph. Yeah, here we have COVID-19, and then these are all these chemical compounds or drugs. You can click edge that will show you the sentences that describing the particular relation, and also that's a PubMed abstract link. You can click to see the abstract. You can also search all the genes related to a particular disease, for example, all the genes related to COVID-19. And then we will get this knowledge graph, and then you can read these relations. And then if you find, okay, one protein or gene is quite interesting, you can actually do additional search on this knowledge graph by just search relations related to a particular gene of interest. For example, this ACE2. What you can do is you can read something about that, and then you can right-click this node, and then there's a little window pop up, and then you can click more search, and then in this window you can search chemicals. Basically, maybe you want to find out some chemical that interact with this gene, which may be useful for treating COVID-19. So then you will get this knowledge graph. Again, you can read these links by clicking them, and then quickly read the literature. So next example is you can also search diseases related to a particular chemical compound. In fact, yeah, you can search all different kind of relations. I will just show some interesting ones. So here you see we have knowledge graph with aspirin and all these diseases. But here there are two kinds of diseases. One is the side effect, basically aspirin will cause that disease, and the other is a disease that aspirin can be used to treat. So as I mentioned, so far we have mixed all of them because we have not incorporated our latest Coherent results. But once we incorporate that, we'll allow users to search two kinds of relations. One is positive correlation, basically aspirin will cause that. That's kind of toxicity of aspirin. And the other is negative correlation, basically aspirin can be used to treat that disease. And you can also use our tool to kind of navigate in the space of knowledge. So if you think about the whole knowledge, right? Basically, it's a huge knowledge graph, and then all connected. There's so many nodes, and then the heterogeneous nodes, and you can actually traverse this whole knowledge graph from one node to another. Because say you may be interested in EGFR, and you read these sentences related to a particular relation, and then you may find out, oh, this PD-L1 EGFR relation is kind of interesting, and then you can search that, and then you may find out, oh, this PD-1 and PD-L1 relation is interesting, and you may even search chemicals that actually relate to the PD-1, and then you can kind of keep searching, and then now this becomes very crowded, right? So you can actually remove all the nodes connected to the PD-L1 by right-click it, and then there's a function called delete this chain, and that gives you a cleaner version. So basically, starting from EGFR, you can traverse to PD-L1 and go to PD-1 and find out all these chemicals related to PD-1. And of course, there's no limitation. You can keep searching, and basically you can really navigate within the current knowledge base. I believe, as far as I know, this has been a very challenging task. And also, it's very important sometimes you want to search relation with certain contexts. For example, people have done research using many different model organisms, and then certain relational gene may be important for many diseases. But you're only interested in, say, EGFR relation with other genes for human beings, and then you are only interested in such relations associated with cancer. Well, we offer that function, so you can actually specify context, and you can specify some even complex context by adding rules, adding groups. And if you specify these and search, you'll get a subset of EGFR and other gene relations that are related to human and cancer. Yeah, as I mentioned, we also have a search engine part, so you can just search like PubMed or Google Scholar, And one unique feature here is there's a green button, display knowledge graph. You can click it, and then I will show you the knowledge graph for the top 20 articles. Of course, here you can change that and select whatever article you want to show. You can, there are actually many functions. Yeah, I forgot to mention. You can edit knowledge graph. If you find out some nodes, edges are wrongly annotated or wrongly tagged, you can edit, delete, or change them. And you can save knowledge graph, you can share with other people. You can do some other modifications. For example, we can remove all the chemicals and genes, species. All right. So I think my time is almost up. So I'll quickly talk about the future work. As I mentioned, we'll incorporate the whole LateCoin pipeline, and that's to be completed next month. And after that, you can search different relation types covered by this LateCoin dataset. And also, we plan to develop more specific knowledge graph. And here, we plan to perform additional annotation, because if you look at LateCoin, only 500 PubMed abstracts, but actually that gives us a pretty good model. We plan to perform some additional annotations for some specific domain, and then to construct build better models, construct knowledge graph, and then we will build applications on top of that. And so my time's up. It's up, Tom.
We're going to give you another minute.
Okay, I would like to thank all the people I have worked with. So we have been working on this field for right now it's 15 years. So there's too many people to list. And then I would like to thank NIH and SF for support. And then recently the LateCoin challenge really helpful. That's basically give us data and the model to build this tool. And finally, please try Biomedical. Okay, and of course, we just released that, so expect there are issues here and there, and I really appreciate and you will let us know, and I will fix them as quick as we can. All right, thank you very much.
Thank you, Jim-Feng. It was very interesting. I think we'll save any questions that came across for the panel discussion, which will start immediately. So give me a second. I'm going to take the screen. I'll try to take the screen.
Okay, so let me see.
All right. Okay, thank you, Jim-Feng. That was a very interesting talk, and thank you all for joining now the panel discussion. I'll invite all of the panelists and presenters to come forward. Before we get started, I'd like to introduce a few of our panelists. Let's see, we have everybody here? Yes, I think we're good now. Very good, thank you. So the first of our panelists, in addition to our speakers, is Dr. Jiang Bian. He's a professor of biomedical informatics with the College of Medicine at the University of Florida. His research is focused on data-driven medicine through application of informatics methods, including machine learning, as well as development of novel data sciences and informatics methods, tools, systems, supporting clinical research. Welcome, Bian. I'm really happy to see you here. We also have Yufeng Cao, Director of Pharmacometrics at Eisai Incorporated. His expertise and interests are in application of AI and machine learning to quantitative systems pharmacology modeling, developing and implementing mechanistic modeling strategies for model drug development programs. And welcome, Yufeng. And we also have Dr. Hofeng Poon, Senior Director of Microsoft Health Futures and affiliated professor at the University of Washington Medical School. Broadly recognized for his work in NLP and AI, and at Microsoft, he leads research on biomedical AI, focusing on structured biomedical data for precision medicine. His research is addressing a wide range of problems in machine learning, natural language processing methods, and applications. And welcome to all the speakers. Thank you for joining today, the last session of the day. Really appreciate everyone hanging with us today for the last session. We know how that kind of peters out, but we do have a few questions, I think. One, I think we can address to Frank to start with. There was a question I came across, Frank, concerning whether or not your methods would be extensible to other approaches, other types of data, more traditionally considered PD data. I think we're talking about a lot of PD data today, but we're wondering, for the things that we, normally call PK, is it extensible to those things, other outcomes, endpoints, et cetera, and the relationships associated with pharmacokinetic, pharmacodynamic relationships?
Yes. It's the short answer, yes.
Short answer.
We have had many discussions about it. We think it should be relatively easy, as we don't need to start from scratch. It will be a term called transfer learning. We use our systems that they are and retrain it on data sets labeled IC50 and PD. In fact, whilst we were labeling using Prodigy, actually, we already had the option in the most recent rounds to also label PD data. We just haven't had the time yet to rerun those algorithms, and that's to come at some point.
The algorithms are directly relevant, or they'll have to be fine-tuned further?
Fine-tuned, yeah, but not from scratch. So transfer learning, yeah.
Okay, very good. Thank you for that. So I also had a question around the knowledge graphs, Jingfeng, that you're developing, and I see that it seems quite valuable for elucidating potential relationships between entities. Is there anything there that helps you measure and pull forward the strength of those relationships so that you can rank them?
Yes, because we use model to predict them. So each predicted relation will have a probability associated with them. But since it's well known that deep learning model, they are kind of overconfident, so we need to kind of calibrate the probability, and then we can sort relations by calibrated probabilities.
Very good. Thank you for that. I appreciate that. Jenny, I wanted to turn to you quickly because I found your presentation very interesting and very relevant to the types of things that we do or we're trying to do at Pfizer. So I'm wondering, for the techniques that you're implementing, how reliable, are the techniques, or are the considerations that we have to keep in mind, relative to these models that you're generating and your ability to pull forward relevant literature elements?
I guess in terms of reliability, a few things we tried out is different palettes and validation tasks. And so far, I think from our training data set internally, they have a pretty high recall, which means that I think they are able to capture a lot of the things that humans are able to capture in our human efforts meta-analysis. So in that sense, I think it is reliable, that the method that we used, which is Huifang's team's PubMed. But I know that I think there will always be paper that get missed out, that were selected by experts for certain reasons, but not selected by models. So in that sense, it's not able to, it may never achieve like 100%, but it does, I think, brings a lot of added values in terms of we are able to potentially capture literature that missed out by human experts. So that's my, I guess, answer to that, but Yifan or Huifan, anything to add on?
Any other comments on the reliability piece, especially in sort of context of how we might use the results in maybe regulatory contexts or, other clinical development decision-making contexts?
Yeah, I think Jenny, you covered very well. I think one thing to add is that it's quite interesting probably to also look at those, even, you showed a pretty low the recall, precision. So actually a lot of the papers, I think, is probably due to that during the human evaluation, a lot of papers being omitted. So the machine learning potentially can pick up those papers, which is quite interesting. I don't know if you have anything to add.
Yeah, I echo this point, and I also want to tee up a few additional dimensions, right? So I think also related to a little bit of Tom, your earlier question, and also Frank and Jim Feng's point. I think the first point view is that we don't envision kind of NLP is sort of like go all the way automated anytime soon, right? So we actually see it as there is actually a very sweet spot of human-computer symbiosis where, human experts are very good at, validating if you have some candidate finding, but it's, I mean, multiple of the speaker have already mentioned, right? Like PubMed pop up 4,000 paper a day. It's really hard to catch up, right? So, but that's where computer actually can really easy to scale. So I think what Jenny, you alluded to, sort of like the recall focus, focusing on getting, not missing any finding, that's sort of like seem to be the first priority. But then also incorporate the NLP with the, like I think Tom, you raised in some of the question about human in the loop, right? What we tend to think about assisted curation, so that NLP identify the candidate, basically some candidate needles from the giant haystack, right? So human don't need to go through all the big haystacks. I think there is also another dimension, which is like, what type of knowledge are we extracting? For example, we work on, for example, gene regulation a little bit, and then we talk to some biologists, they joke about like any direction you go, right? Like gene A regulate gene B, they could find another cellular context when it's the opposite, right? So that means like, there is also, even saying whether a finding is true or not, there is a lot of nuances, right? And also there is a question about first step being, so like Frank, you mentioned about machine reading, right? So taking the offer at the face value, but then also once you have a population, the literature level knowledge graph, right? Then we can start to assess like which finding is more consistent with others and which ones seem to be, could be noise, could be errors, and so forth. So there are multiple angles which could be very, very interesting.
Very good comments. Thinking, sort of reflecting on the process a little bit, and the reliability question, is the state of the art such at the moment that it's critical to have a human in the loop relative to qualifying what we're doing? Is it so early days that we're not quite there for the machines to be just set free? Frank, please.
I can't speak for others, but for our project, yes. In its current stage, there is maybe in the future. Let's see.
I would echo that. I would actually think maybe not even in the future. It's going to be forever the case. I mean, I really think the performance trade-off needs to be think through for every single application. It's going to be very different, right? So yes, recall focus may be suitable for certain applications, but then when you actually get to where precision is important, then it's really important. I was just talking about another NLP project today where they did summarization task from literature and tried 4 or 5 different models. They all give different answers. And then the problem is which one is more trustworthy, and that gets into how you actually disseminate NLP results to the downstream users, maybe clinicians who's actually going to use it for clinical decision support.
It's a good question to have in mind as you're proceeding with these projects. I guess another question that comes to mind is around other types of endpoints. I didn't hear too much today, I don't think, specifically around safety, maybe in some of the earlier discussions in the workshop. Safety endpoints tend to be underrepresented, I would say, in the literature. Does the panel have any comments about, how these methods might extend to safety rather than efficacy and other, clinical outcome measures? There's a bit of a worry about publication bias, and thinking about that in terms of traditional systematic reviews.
So maybe I can take a first step. So my team specifically had looked at a little bit about ADE side, adverse drug reaction, adverse drug event, both from the literature side, but also from a real world evidence side, for example, from EMRs, right? So, and for the literature, exactly like you said, there has been actually shared tasks where people looking into extracting ADE, and we have some internal effort that we are seeing probably soon can actually share, but we have did sort of PubMed scale ADE extracting sort of ADE findings, but obviously the concern is, like you said, sort of like could be publication bias. And on that regard, sort of the EMR side, from the real world evidence side, actually might give you maybe perhaps a more unbiased sort of like review, presuming that you have access to a big patient population. The challenge there, of course, is to figure out the confounders, right? Like figure out the actually establishing the causal relation between the drug and actually the adverse event that may be non-trivial. So we separate the problem into extracting explicit ADE reference when the clinical folks think this is a reaction to the drug, versus sort of like implicit could be high correlation of the AE versus the drug, and then further try to figure out the confounder. So there has been some work, and I'm sure the other speaker probably have also done work in that space.
Any other thoughts in that direction? And I also wonder a little bit around the requirements for reporting. Have these methods uncovered relationships that were, previously unknown relative to safety? I'll just use that example again, because of the, regulatory requirements, for us to report those relationships wherever we find them. So is that either a concern, something that you're able to handle, and something that you're able to flag and sort of move forward on, with your research? I just wonder about that a little bit.
Yeah. So yeah, this is good kind of aspect when we do information extraction. And actually, for Litecoin data, they also annotated for each relationship whether this relationship is actually describing novel discovery in the article or just talk about a background knowledge. And then we were asked to predict this novelty. And it turned out the novelty prediction can be quite high. So in a way that we can actually scan the documents and identify that new discovery. For example, we actually find this chemical can cause that side effect or that toxicity. Yeah, so I think, yeah, this is, people kind of recognize this is important and this is being addressed. Yeah.
Thank you for that, Chief.
Yeah, just sort of quick comment. I mean, my research mostly real world data and real world evidence. I serve for the chief data scientist for your health system. So it's important for us in terms of safety issues. There are two levels, like what Hufang was saying. The causal relationship is really hard to establish. And that's not just an NLP task. And the NLP may be able to detect some of the adverse event signals. The causal part is really hard. I don't think there's a good solution yet. I don't think even a human reporter can be able to actually do that accurately. So again, human in the loop still is important.
Yeah, the causal part.
Go ahead,
Sorry. The causal part, say if we have another graph, right, we can actually extract the detailed relation type. Then it is possible to kind of infer it. For example, people have identified this chemical compound inhibit that protein, and then when this protein is inhibited, and then it causes certain disease, right, or certain side effect. Then that causal effect can be inferred from literature by building knowledge graph. Right?
Yeah.
From the knowledge part, yes. I think the real world data, the EHR data, that's more challenging. We really don't have a good way to even establish cause of death, right? Whether it's COVID or cardiovascular disease, that's a judgment call at the end.
Yeah, and actually, Jiang, we should chat offline because we're starting working a bit on the causal side. Love to pick your brain. But to echo your point, I think Jinfeng, your point is that like if we have a correlation, for example, in gene regulation, right, you often see the paper like they see some correlation and then they try to uncover the mechanism, right? So maybe they gene A control some mediator, which then control gene B, and then in that case, you establish a causal pathway. The challenge in that usually is that because the knowledge graph you get from literature, A is super sparse, right? Super noisy. A lot of edges may not be properly qualified, right? And also could have error and biases and publication biases, right? I think Jiang, you mentioned about the real world data. That's exactly spot on, right? So you see this drug often preceding abdominal pain or something, right? So, but is it really the ADE or is it just happenstance? So causal learning is not silver bullet, absolutely not. But it can alleviate some of the confounder, but it's still a long way to go. And then also probably at a bigger level, right? So some of that actually could be interesting for even, for example, like Tom, right? Like in pharma, right? Like could this be actually a hypothesis generation engine for you guys to look at, right? Like to see, okay, for example, we are looking a little bit at precision immuno-oncology, for example, right? So like immunotherapy is very promising, but response rate still the minority, right? Why is that, right? And also cancer drug right now working so well, people live much longer. We need to worry about ADE, right? So we need to worry about cardiotoxicity, heart attacks, and all that. And so the cost will become more and more important in that aspect.
Very interesting, and not a surprise, really. Let's shift gears just a little bit here. So I saw a bit today, quite a bit around, PubMed and using it as a source for abstracts, and then ultimately, full text extraction. I'm assuming from the sources that we're obtaining from PubMed. But is there any question about how you would extend this to other sources of full text content, or is PubMed somewhat self-limiting as a consequence? And if it is self-limiting, what kind of methodological problems will we run into because we're not getting the full story? Jenny, do you have any thoughts on that?
Yeah, I can jump in a little bit. I think that using only PubMed would definitely be limiting, but I think PubMed will always start with this because this is the most direct and easy to use database that we're able to just do a keyword search and we're able to get all the results. But I think both you, I think Huifan's team is also moving on to using like the full text train. I think the PubMed board is actually trained not only using abstract but also full text as well. So I definitely think there are more potential in our testing step that also test on full text. I think that will add on, that will definitely improve the accuracies, and that will definitely be a next step for us.
Thank you for that. And Frank, I think you had a comment.
Yeah, no, yeah. The other thing is the format. Obviously, it's easiest to pull HTML from PubMed abstracts. They're all HTML. Then in our case, we had 7,000 HTML formats in full paper, especially the older ones. They're just photograph PDFs. I mean, the newer PDFs, I'm not sure what the format is, how you call it.
The appropriate layers in them.
Yeah, then you can still parse them with existing packages. If you really go to photographs, then it's another level.
So Frank, to echo your point, our first conversation with some clinical genomic folks, they hear us about doing machine reading. They were very excited. They said, we have a lot of scan PDF. Can you do OCR for us, right? And actually in clinical, we learned that in many situations, half of the pathology report are still scan PDF. So we did need to do OCR anyway. But I think, Frank, your point is actually even more general than PubMed, right? So you are alluding to certainly abstract and full text. Full text have much richer information. Like, for example, a lot of variant detail you cannot find in abstract. But I think, Frank, Tom, your point is also what about like social media? What about conference abstract? What about press release or something, right? So I think probably there are two dimensions there. One is like how, as your question alluded to, how structured are those texts? How difficult? How formal are the language, right? Like social media are really crappy, right? So it's maybe a little bit more like clinical notes, but much worse probably. And then on the other hand, like press release, they are very formal English, right? Pretty nice English. Conference abstract probably not too different from PubMed. And probably another dimension is that what are the things you want to extract from those? For example, I have seen people working on like surveillance, like epidemiology, right? Social media sometimes can play a role, right? And complete landscape, certainly, I mean, some of the press release and all those. When we work with some collaborator on curating precision oncology knowledge, they constantly beating on us like, you also should go to ASCO or AACR, right? Grab the abstracts and so forth. And for that part, actually, it's relatively just an engineering resource issue because the texts are relatively similar. So.
I think you bring up a very interesting question. It's kind of a laddering of reliability across the various sources, right? PubMed is kind of, you know, the target that everybody seems to go to at the moment, and it has some structure to it and a bit of structured reporting associated with it. The full text is, you know, the Wild West in some respects, in that, you know, you'll never know with a researcher how they'll describe a result. It can be described in many different ways. You don't know what the statistical sort of relationships are. You know, they're just kind of, you know, very difficult. And then we have things like EPARs and summary basis of approval from the FDA and the EU authorities. And you mentioned, you know, social media, which is beyond the Wild West. You know, that's just completely random in some respects. You know, how do you make sense out of any of that without structure? It makes me wonder a little bit for scientific and medical reporting, you know, whether we'll get to a point in our future where it'll not only be an expectation, it might be a requirement to provide structured content rather than what we see today. Because the other thing, and this one thing I wanted to ask you fellows and ladies, is really around, I heard you talk about table extraction, very exciting, entity relationships from the full text. Of course, I wondered about, you know, the possibility of data extraction from graphs that are presented, et cetera. And how well do you do both table and graphs and things like that, you know, with the current tools that are available? Or is this primarily textual relationships that we're getting at? Yifeng, would you like to comment on that? Any thoughts on that?
Well, I think Frank showed some examples and trying to move to that direction is very exciting. I think it's quite important. I do think, you know, another very important aspect, we were touching on a lot of stuff regarding, you know, data extraction, relationship extraction. I think it's also very important to think about how do we use all these relationships and also data we extracted in informing our modeling or you know, pharmacometrics or even QSP models, built into you know, to use those knowledge, leveraging those knowledge and the data from literature that extracted using NLP to really help with drug development. I guess we don't have a lot of time, but yeah, so that's the question I think we should really think a little deeper. I think also. I think, you know, the current tools provided by you know, experts, we always have a lot of space to improve, but it's already been quite a good tool compared to what we are always doing, just using the human power, right? So something to think about, I think.
Thank you for that. Well, we've gone. Yeah, please go ahead.
Oh, sorry. I know we're running out of time, but I want to quickly mention the multimodal part that Tom, you alluded to. The table one that Frank described is very exciting. I think actually another one that tend to be underexplored right now is even supplemental and attachment. So lots of great points for meta-analysis there. Not very pleasant publication glory kind of thing, but very practical. I want to also mention another modality, which is the figures. So my team particularly did a little bit of work, tiny bit, just caption and figure, but there is lots of room to pull information out from figures. When we work on gene regulation, people keep beating on us like text. Actually, oftentimes they have a lot of important thing in the figures. And then when we start to work on other like variant, translational precision oncology treatment, again, like you see a lot of IC50 result in the figure, right? You may not explicitly mention in the text, so there is a lot of room, like Yufang, you mentioned, like lots of room to improve.
Thank you for that, Yufang. I think this was a very interesting session. I think there's room for many other questions. I think for the audience to sort of understand and how we use this information in discovery and development would be a whole other topic completely. I mean, we saw some of that today, but this is where a lot of it begins, extracting the content, from the literature, scientific and medical, and other sources, as we mentioned. James, do you have any other comments before I close?
No, that's all. Thanks everyone for your very interesting talk and participation today.
Thank you, James. And I want to echo those comments. Really, thank you for sticking through to the end, but it's not the end. Today is day one of a 2-day or day and a half workshop. Tomorrow we start with the last session of the 4-session series, where we're going to focus on artificial intelligence and machine learning and drug discovery methods. So this is going to be very interesting. We start again at 10 a.m. Eastern Time sharp. I wish you all, thank you very much again for joining us, bringing your presentations. I wish you all a good evening, and I hope to see you everybody tomorrow.
Yeah, thank you. And I much appreciate Louis and Maya Leia from the IQ team to make the technical side possible. And hope to see many of you again tomorrow.
Thank you.
Yep, thank you.
Bye all.
Bye.